at a time? Yeah. All right, so I need data collected for that. The first live is over. Guys, you're taking too long. The next thing I would want you to do is have 2 beakers. Mark Davis had mentioned 2 beakers, sodium chloride, and stirring rods for each group. There? The sodium chloride. Okay. What about the beakers and distilled water? Kwambie miguu zangu zinauma. Miguu zangu zinauma. Tuko massage. Simama sana. All other parts of my body are okay, but my legs.
No kitchen shoes.
That's what nivae sandals. What? No, I have energy. Okay, I don't mind glucose, yeah. Maalam ndio zinalia. Now, by now, I need you seated with 2 cuboids that are identical in terms of measurements. They may not be identical in terms of weight, but at least have a reference. Have a reference. Ukikucha ni hivyo. Ukikucha ni hivyo, hiyo feeling inaisha. Kukikucha, hiyo feeling inaisha. Yeah. The sleep hormone stops being produced. Yeah, you're being provided with the beakers. Now, in one beaker, you're going to place distilled water. In one beaker, you will place distilled water. And in the other beaker, you will place in one beaker, you will place distilled water. And in the other beaker, you will place a high concentration of sodium chloride. Mark, we need to be faster. Where are the beakers? Because we would have already distributed the beakers, and now they would be making solutions, but we are just... Huh? Wow. We need beakers. Marlene, we need beakers. Sorry, we need beakers. Beakers and stirring rods and... Why are there not enough beakers? Now, as you're seated, I'd like to hear the dimensions and the weights for your cuboids. Please, excuse me. If we keep working slowly, we'll have a lot of time lapses, and it's going to eat into our record, please. Let's work faster. Okay, I can see this group. What's the weight? 5.808, 5.892. Now, you'll place this one in distilled water and this one in concentrated sodium chloride. All right? So, can I have 2 beakers for that group, please? And sodium chloride and also distilled water. Yeah, so you need to time the length of time that those cuboids are staying in the different solutions. So I think we can agree on 5 minutes. Yeah, we can agree on 5 minutes. We are getting the timers just now. So once you have your 2 cuboids, raise your hand. You'll be given 2 beakers. You'll be given distilled water, and you'll be given sodium chloride solution. Then now you allow it to take a break. You can't sleep. You allow it to stay there for 5 minutes. So we only have 6 groups. Is it 6 groups? So we are giving you timers. Set the timer. Start the timer at the same time for both. Please put sufficient liquid. Put like even over 100 ml. A liquid that will cover the cuboid. So I like their idea of writing their findings on the filter paper so that they don't confuse the cuboids. All right, I'm looking for the group that has already started timing. We need distilled water. Use tap water instead. Use tap water instead, it is still hypotonic. Okay, start timing. You can use a thermometer to stir. Yeah, you can use a thermometer to. Oh, you have stirring rods? Thank you. Anyone requiring a stirring rod? Si mko na stirring rod hapo. But you're not stirring the one that is hypotonic. Are you timing? So we are all timing them at 5 minutes. 5 minutes. So your A is the one you have put in hypotonic? Yeah. Hypotonic. Okay. Are you timing? Yeah. Okay. I need my sandals. Sorry? Let me wear this. Let me wear this instead. I went with them to the yeah. Now, the next step is to, once the 5 minutes are over, is to measure again the length, the width, and the height, and then go and measure the weight again. Then see how to compare the two before being put in a solution and after. Can we now make the final measurements? 5 minutes should be over. Not yet. 5 minutes are long today. Where? Huh?
Like 20 seconds.
Can you stop playing with the plastics in you? Eh?
Ameingiza ya mati yake.
Now I need to start getting the measurements. Please come and clarify. Come and clarify to me. Yes. I came back.
This one you came back 22:34, so you took 4 minutes extra.
Extra.
The previous time.
So that's a time lapse. Okay. So rest time I have accumulated now is?
You finished your rest time. Negative 4 minutes.
Okay. So I came back at 2:45.
Yeah, this one is okay. For us,
I came back at
2:45. 25 to 45, but for the previous ones, when you were not there, you used 4 minutes extra.
The rest time I took was 15 minutes.
It was up to 22:34.
And the one I have taken, the previous one is? The one I've taken
But the
15.
previous one you took,
Okay.
they said you took 4 minutes extra.
Yeah, I did. Okay. Okay, let me have the final measurements, please. We need to move to another activity. You're going now? You're done? For now? Okay. All right, let me have the data. Is there any group that has the final data? Okay. All right. Okay. Let me listen to Fabian. Now let me listen to Fabian. Now, Fabian, tell me what you are doing and the figures you've been having. Now I need all of us to settle now. I know you really want to have your data, but time for that activity is over.
The sample that we placed in distilled water, the initial weight was 5.808 grams. The length was 2 centimeters and the width 1.5 centimeter.
Did you measure the height?
It was,
Continue.
so, after the experiment, the length was 2.3 centimeters and 1.5 centimeters. But now, on the weight, it was 5.941.
Wow.
941.
Quite interesting set of data.
In the concentrated sodium chloride, initial weight before the experiment it was 5.892 grams. Length 2.3 centimeters, width 1.5 centimeters. After 2.2 centimeters length and width 1.4 centimeters. And now coming to weight, it was 5.637 grams.
Perfect. Their data is very interesting and very correct. Yeah? Clap for them. That's amazing. Wow, I really like this. Can I listen to another group? I saw, is it this group? Yes. Who is representing or who is presenting? Yes, Faith. Bilha. Ona sasa nambuta Faith.
So, for the hypertonic solution,
You're starting with hyper? Yes.
the initial measurements, the length was 3.4 centimeters. Both the width and the height were 1 centimeter. And the mass was 4.0 grams. After the 5 minutes, the length was 3.3 centimeters. The width and the height were 0.9 centimeters. And the weight was 3.98 grams.
Wow. Then let's go to hypo.
The similar measurements before the,
Similar, yeah?
apart from the weight.
So hypo, let's just talk about the weight.
Yeah,
Uh-huh.
it was 4.4 grams. After the measurements, they both increased by 0.1.
4.5 now.
Yes.
Yes?
Yeah.
All right, let me use these two sets of data to discuss basically the behavior of cells when you place them in different concentrations of what? Of solutions. Are we together? Now, I really am happy that we've been able to do this investigation and found the data that we found there, because that data is very acceptable and very correct. All right? Now, it goes to say that when you place cells in a hypotonic solution, what happens? They gain water molecules because osmosis occurs and the water molecules move from the hypotonic solution to the hypertonic solution, which is the inside of the cell. And that's why you're seeing everywhere where there is hypotonic, there is an increase in weight. Like here, we increased weight from 4.4 grams to 4.5 grams. Now, for hypo, for Fabian and his group, we started with 5.808 and ended up with 5.941. So you account for the increase of that mass as a result of osmosis happening. So that is actually evidence that osmosis has happened within those 5 minutes. Are we together? Yes. Now can we fill our table? Now I'll wrap this data. You'll include your own data, hoping it is also acceptable as these ones. Now, before we fill that table, it's important I talk about the steps we use to carry out an investigation in the lab. And it's good we have done an investigation, then now we can check the steps that we have followed. Now, we were investigating the changes in volume and weight stroke mass of potato cuboids immersed in different solutions. Are we together? So the first question is the first step is to come up with a question or form a question or ask a question. Ask a question sounds better. Now, which question can you ask for this investigation? Which question can you ask for this investigation, Alvin? Yes, just speak. Don't be distracted.
To investigate the behavior of potato when placed in a hypotonic and hypotonic solution.
No, that's not a question. That's not a question. Yes, Haron?
What is the mass of the potato cube?
Here? Where? Where? Please clarify. Maybe I'm not understanding.
Of the specimen, of that specimen.
No. The question, what question can we ask from this investigation? Yes?
Can you say what are the different concentrations?
On what?
Plant.
Yes, you can mention that, or probably be specific and say, does the volume and weight or mass of potato cuboids change when they are put in different concentrations? That is a question, isn't it? So the first step is asking a question. The second one is coming up with a hypothesis. Yeah? Now, a hypothesis is like a conclusion that is made prior to the conclusion that comes after the investigation. So, formulate a hypothesis. Formulate a hypothesis. Now, in this case, what is the hypothesis? Try and make a conclusion out of the investigation. Yes, Imelda? Mm-hmm.
There's no significant difference between the potato slices that are put in hypotonic solution and that one that are put in hypertonic solution.
Yeah, she's saying that there is no significant difference between potato slices put in hypotonic and those put in hypertonic solutions. Can we try and get hypotheses that are not negating? Because like hers is saying there is no. Why don't you just say as if it's, you know, positive, and then prove it wrong by your data or prove it right. Okay? Now the third one is identifying variables. Identifying variables. Now we have several variables, and I'm glad we are at this point now. Okay? We have constant variables. We have dependent variables. And we have independent variables. Are you together? Now the constant variables are the factors that you're keeping the same so that you're able to get reliable data, you're able to get comparable data because you're using constant, you have put in place constant variables. Now for this particular investigation, what are the constant variables? Okay, time of investigation, which was how many minutes? Another one? Maitha? Is he Maitha? Yes, Maitha. Tell me something. Martin is also quiet. Jelimo is quiet. Deborah is quiet. Say something, say something. Constant variables in this investigation. No, me, I don't want you again. Size of potato slices. So I told you to ensure that the two potato cuboids are the same. So size of potato slices. Another one? What else have we kept the same? Yes, Alvin? Amount of water for? Have we kept it the same? I didn't see us measuring that. Yes, Fabian? Or you kept the same? So for in some cases, the volume of water used, volume of solvent actually. Now, the dependent variables. Now, are the dependent variables what we are observing or what we are measuring? Scientists, wake up. Sorry? Yeah, the dependent variables refer to what we are measuring. Are we together? Then the independent variables, okay, refer to what we are changing. Refer to what we are changing. So dependent variables are what we are observing, what to observe. What to observe. And then these ones are what to change. So the dependent variable in this case is change in mass and length, width, and height. So you can say mass, length, I should teach when bending like this, width, feel better, and height. All right? And then what to change? Good. Concentration of the solvent. So I think now we can go to the table. Now let's go to the table quickly. Now can you fill in quickly what happens to plant cells in hypotonic solutions, referring to the investigation we have just concluded on potato slices. So you account for the changes you have seen and give an explanation biologically. All right. Now, please also include how plant cells behave in terms of when you put the potato slices in a hypertonic solution. Now, the terms to use in this description are turgid, turgor pressure, flaccid. Are we together? Those are the terms I would want to see you using and hear you using. Now the case study of the red blood cell, I'll give you 2 minutes maybe to finish that. Oh, we already finished half a day. I didn't even notice. Now let's talk about animal cells. Put your pens down and look at me. I'll give you time to write again. Let's talk about animal cells. Now the process is the same. It's just that the behavior of the animal cells is different. Okay? The behavior of the animal cells is different. Like for example, when you put an animal cell in a hypotonic solution, it will absorb water. But because animal cells do not have the cell wall, what happens? Bursting happens, and that is what we call hemolysis. Hemolysis. The bursting of animal cells as a result of placing them in hypotonic solutions. Hemolysis. And then the same thing happens when you put a plant cell in a hypertonic solution. Same thing happens when you put an animal cell in a hypertonic solution. The cell shrinks. The cell shrinks. Alvin, right. Let me get for you the term that describes the shrinking of animal cells. Yeah, so the shrinking of the animal cell is plasmolysis, which is the opposite of what? Hemolysis. Are we together? Yes. Plasmolysis. Now let's look at the importance of water potential and osmosis in the uptake and loss of water by organisms. So why study osmosis and why look at water potential and the uptake, the uptake of water and loss of water by organisms? Now, as I've said before, that when plant cells gain water through osmosis by being placed in a hypotonic solution, they become rigid and firm. Why? Because the large central vacuole gains more water. Are we together? Yes. Now this is important for plants because it makes the plant to be firm and it also provides support and strength for the plant. These points you should be writing under that question. Explain the importance of water potential and osmosis in the uptake and loss of water by organisms. Yes? So, due to osmosis, plants are able to be provided for strength and support as a result of turgidity of cells. Now, if a plant is losing water than it is gaining water, that plant ends up wilting or withering. Wilting or withering. So that tells you that a plant should always have sufficient supply of water for it even to appear upright, because it is the water that gives the cell the shape of the cells. Are we together? Sawa sawa. Now let's look at where osmosis is applicable in animal cells. Now we know that animal cells also lose and gain water as a result of osmosis. Are we together? Now we should be very keen on the solutions that we expose the animal cells to, because animal cells, we said they do not have what? Cell wall. So that means if they are exposed to a hypotonic solution, what happens? They will burst, which is hemolysis. Are we together? Yes. Yes, so we should have the right concentrations within our bodies. Now, have you ever heard that you can drown yourself to death by drinking too much water? Ever heard of anything like that? I see nowadays Gen Zs, let me stop saying that. Okay, I see some people nowadays carrying water with very big bottles that look like jerry cans. In the name of skincare. If you sleep again, I'll spray you with water. In the name of skincare, but you should take the right amount of water. Are we together? Yeah, so that you don't end up drowning yourselves and getting yourselves hemolyzed. All right? So the third mode of transport is active transport now. Now I want to define active transport as the movement of particles. I'm defining active transport as the movement of particles through a cell membrane from a region of lower concentration to a region of higher concentration using energy from respiration. Active transport is the movement of particles through a cell membrane from a region of lower concentration to a region of higher concentration using energy from respiration. Now, I always tell my students that active transport is like trying to make a river flow going up. Okay? Can you imagine a river coming from Indian Ocean and then climbing and going back to Mount Kenya? It must be very difficult, isn't it? Yeah, so active transport is actually the uptake of substances or particles from a region of lower concentration to a region of higher concentration using energy from respiration. Now, I have provided for you a diagram there for active transport across the cell membrane. Now, we have substances called the carrier proteins that are found within the cell membrane. Now, the work of the carrier proteins is to transport or to ferry. The carrier proteins are for ferrying substances from where they are having a low concentration to where they have a higher concentration. Now, one thing you need to note about the protein carriers, Alvin, we are writing. One thing we need to know about the protein carriers is that they have a complementary shape to the particle that they are transporting, carrying across. Now, have you ever seen when it really rains, okay, and people need to move from one place to another? We have some people volunteering to carry the women and the small children across, like the floods or across the water. The reason why I have not mentioned men is because they don't agree to be carried. So I have not yet seen. Okay? Yeah, so I liken the carrier proteins to these people who carry people from one part across the floods to another part. So it's the same thing, only that the protein carriers have a complementary shape to the substance that they are transporting. Now, importance of active transport. Now, the first one is uptake of glucose by epithelial cells in the villi of the small intestine and by the kidney tubules in the nephron. Now, when they do a urinalysis, I'll repeat, when they do a urinalysis, they basically test to see whether your urine has proteins, so they check for amino acids, and whether your urine has glucose. Your urine should not have those substances. Why? Because the nephron, which is part of the kidney, should have reabsorbed. The nephron should have reabsorbed those substances regardless of the small concentrations that they could be in the urine. Are we together? Yeah, so that tells you that most times the uptake of glucose is done by active transport. Are we together? Yeah, so that means there are carrier proteins as well within this membrane. Another application. Have you written uptake of glucose by epithelial cells?
Yes.
We are looking at importance of active transport. So uptake of glucose by epithelial cells mentioned that. Then uptake of ions from soil water by root hair cells. Group 3, health break. Uptake of mineral ions from soil water by root hair cells in plants. So think about a scenario where we have a shortage of some ions in the soil. Yet, the plants need these ions. So how will these ions get to the plant? Through which process? Active transport. Now the carrier proteins carry molecules or substances across the membrane using energy from respiration. And that is why you cannot disassociate respiration when you are defining active transport. Then once they carry them, they release them into the cells. And the diagram is there. Can you see it? The phospholipid bilayer, that is the layer of the cell membrane. Then you find the carrier proteins there that bind the substance being transported, carry that substance, and then drop it, or rather deposit it inside the cell. Now the phospholipid bilayer is also another another topic by itself. Yeah, so let's relax. Okay, on to another topic still in biology. Okay, let's look at chemicals of life. Do you remember that topic? No? Chemicals of life. Now, chemicals of life are also known as biological molecules. Chemicals of life are also known as biological molecules. Now, let me go around seeing what we've been doing. Kindly account for your time during the lesson. Now, for the effect of plant and animal cells on immersion of cells, I intended for you to have a diagram on the second row. I intended for you to have a diagram. So when you get time, please make sure you draw that. I'm looking at everyone's work, please. One way of participating in a lesson is writing notes. So your table is empty. Yeah. Do something about it.
I've written the data.
No, we just didn't do the data. The data actually we were doing in the exercise book. Can you feel the effect? Yes. Let me see. Ni watu wa kuchapa story. Yeah.
Wanaongea loudly.
Okay. Msiandike negative 4. You're not supposed to write negative. It's time lapse. No negative. I can't owe you time. That's why nilikuwa nasema nichazwe kwanza na nini. Tunafanya draft. So you can't say I owe you time. Mtasema yes, I took the 20 minutes, but there was a time lapse of 4 minutes, and account for it like she was walking to the venue or something like that. But it can't appear that I owe you, because saying I owe you means that when now I accumulate my 30 minutes, I'll take 26 so that I pay you your 4. It's not supposed to be that way. So it's a time lapse.
It's a time lapse. It's a comment rapid and a comment.
But this is not the timekeeper statement. Time lapses are captured in the timekeeper statement. Yes. There was before that. What about the one that says total rest time available?
And then that you have that break, tunasema this is our kick off time, sindio?
Sina break. Nilikuwa tu nawaambia hii juu she came and showed me that I have negative 4 rest.
No, sasa nilimwambia.
No, it's not negative.
No, so how are we going to capture it? No, that's a question.
Angalieni kwa hizi documents.
Tumetafuta, that's why tunamwambia hivyo. Kwa kuna mahali tunaona. It's only the statement and itakaptariwa wapi? Ama
kuna mahali. All right. So we have 4 main chemicals of life, 4 main biological molecules. Can I hear what you have in mind? Biological molecules. Oh yeah? Biological molecules. Jelimo. Sarah. Dolin. Can I call you Dolly? Dolin. Doline. Wow. Sorry. Forgive us. Doline. Biological molecules. Yes? Carbohydrates. Nice. Can we write carbohydrates? Yes. Sorry. Carbohydrates. Now, if you feel sleepy, wash your face. Okay? Yeah, wash your face. You'll be okay. Yes, Sarah? Lipids. We have lipids, and for lipids, we have fats and oils. Lipids, we have fats and oils. What's the difference between fats and oils? What's the difference between fats and oils? Deborah, finally. Fats are from animals, so they're sourced from animals, and then oils are from plants. Any other difference? Fats are solid, and oils are liquid. All right. A third biological molecule, Jessica. Protein, good. The fourth one? This one, the one who gets. Aki, yes? No. Where? Jelimo? Nucleic acids. Good. A DNA, basically. All right. So these are the main biological molecules. Now I want us to look at the chemical elements that make up these biological molecules. Are we together? Now we start with carbohydrates. Now which elements make carbohydrates? Elements that make carbohydrates. I'm going to write down okay, I can see people's hands up. Who do I pick? Clarice? Carbon, perfect. Someone else? Let me write for you the formula for glucose so that you tell me which elements are found in carbohydrates. Staki chorus answers. Hatuko nini? Jessica? Hydrogen. Then lastly? Marston? Oxygen. Good. Carbohydrates, hydrogen and oxygen. Now, fats and lipids are also made from carbohydrates. Fats and lipids are also, lipids are also made from carbon, hydrogen, and oxygen, just like carbohydrates. Are we together? It's just that the bonding of the elements is different, and also the number of atoms present per element is different. So we end up getting different compounds. Some are carbohydrates, others are fats and oils. Tuko pamoja? Any questions so far? So for fats and oils, you'll indicate the three elements as well. Now let's go to proteins. So I won't really write for fats and oils. Let's go to proteins. Quickly. Hey, chill. We are talking about chemical elements that make the biological molecule, not the simplest molecule. You understand? Yeah. Yes, there's a difference. There is a difference. Clarice again. Nitrogen. Nitrogen. Jelimo. Oxygen. Yes. Hydrogen. And lastly, carbon. Now, some very unique, very unique proteins have phosphorus and sulfur elements in them. They are rare, that's why I'm calling them very unique proteins. Now, who has spoiled your nini? You didn't want to keep it nicely? Oh, yes. Okay. Now, the nucleic acids. I want to talk about nucleic acids now. Tumalizane. Sorry? Well, we are not doing protein. Reducing sugars. Okay. Nice. Okay, let's look at nucleic acid. Now, in the nucleic acids, we are looking at DNA. In the nucleic acids, we are looking at DNA. Now, we have two types of nucleic acids. We have DNA and RNA. Now, what is the full name for DNA? One person, please. Yes? Jalimo. Yes. DNA. Deoxyribonucleic acid. Now, what do you know for D? D means? D and bi means? Two. So that is where the double helix structure comes from. Are we together? Yeah. The double helix structure comes from the two strands. Now we have protein bases. We have protein bases that match, and you match them according to the specific protein base that is available at that time. Like, you're given the sequence, and you're supposed to use that sequence to match others. Now let me give you an example so that it makes sense to you. So we said that the DNA structure is a double helix structure. You can see it there. Okay. Now the DNA has instructions for growth and development of all organisms. Did you know that how you look is highly contributed by your DNA? Yes. Did you also know that there are some diseases that are spread from generation to generation through the DNA? Yes, so that tells you that the DNA carries instructions that influence an organism. Now, the double helix contains two strands of DNA that wound around each other. You can see that in your diagram. And this is what we call a double helix. Now, the individual units of DNA are called nucleotides that have bases. Okay? Now, we have four bases. We have A for adenine, T for thiamine, C for cytosine, and G for guanine. Now, the bases always pair up in the same way. Now, the easiest way to remember is just saying ATCG. Can we say it? ATCG. Yeah. ATCG. When you say it twice, you can't confuse and say AGCT. It's ATCG. So adenine matches with thiamine, and cytosine always pairs up with guanine. Are we together? So I want to give you a strand, one side of the strand, for you to match it for me with the other strand. All right? Okay. Are you done writing this? So this is one side of the double helix. Are we together? Now using the ATCG, can you match and give me the sequence for the next strand? So the next activity is food tests. We are testing for starch, testing for reducing sugars, we are testing for proteins, we are testing for fats and oils using ethanol. So that's what we are doing in 2.17. Now I had requested you don't get rid of the leaves because we would want to probably test for starch in some of these leaves. So we'll need tripod stands, wire gauzes, so that we are able to warm the leaves. We'll need ethanol. And now I had requested for white tiles. There are white tiles. Tiles. Tripod stands. At least the first activity is testing for starch in a green leaf using ethanol. Using iodine, sorry. Yes. Let's take like 2 minutes to receive the apparatus for food tests. Maggie. We are taking too long to set up practicals.
No, you know, we had not planned to set up because it's in the night. We don't have time. The leaf has not photosynthesized. So we were.
I told them when I gave them this book that they should be following me because I'm following it in order. So they should look a few things.
But the other food is ready.
So we do the others? Okay. Okay. So. The more you stay awake, the less sleepy you feel. The more you stay awake, the less sleepy you feel.
I can see this doable.
Yes.
But I have tried. I also be at
Okay, now we have several food substances that have been provided. We have iodine, we have glucose, ethanol, benedicts, and we have starch. Now the first investigation we will do is test for reducing sugars. Okay? We are going to test for reducing sugars. Now the reason why you're being given a Bunsen burner or a burner is because when you're testing for reducing sugars, we use hot water. We use hot water to fasten the process of the reaction and also to make sure that we observe a change. Let me adjust it for you so that it's non-luminous. How come it's luminous? Is it a gas? Yeah, because it's open. How many people are out?
I'm not sure, but not much. I think it's only the 5.
I need to ensure we are more than 10. Okay, can go. Can something be done about this? Bunsen burner is just producing luminous flame with smoke. Now, what is the procedure? We are testing for Benedict's solution. We are testing for reducing sugars using Benedict's solution. Now, the procedure is you take approximately half the amount of glucose you have been given, okay? Approximately half the amount of glucose you have been given, and a few drops. We don't have droppers, but you can estimate like 5 drops of Benedict solution. We are putting all these in the boiling tube provided. These are test tubes, these are boiling tubes. So we are putting all that in the boiling tube provided. Please make it a habit of rinsing your apparatus before you rinse your apparatus before you start putting reagents in them. All right?
Yes.
Yeah, so follow that procedure and then place the water. Just put, we see. I wonder why it has behaved like that. Ideally, it's not supposed to do that. Oh, surprised. Yeah, this is not viable. The other alternative is probably to add some more Benedict solution and see. You see?
Is it supposed to be bubbling like that?
Yeah. Let's see. So. Did you do biology in high school?
Yeah, and we have done to shoot when we were chemistry.
Ah. Biochem. Now I want the group that is having tangible color change to miss. The Benedict solution is reacting with the glucose, fizzing and okay, let's wait. Yes. Huh? Like half, approximately half of it. Then estimate a few drops of you need another reagent. It's not supposed to behave this way. Miss, this is the observation after mixing glucose and Benedict solution. Benedict solution. It's fizzing and turning colorless immediately.
I told you we hydrolyze, so they had forgotten to add sodium and carbonate. So the new one they are giving them is neutralized.
Okay, so we dispose of what we have, the Benedict solution.
No, Benedict is okay.
Glucose.
Yes.
He has changed for us.
Now, can you dispose of the glucose you have? You're being given.
No, your group is okay. So you see the other one is okay.
The other one is working. Can you? And they are droppers. They can share.
Damaris, you're very generous. You're just distributing.
I want them to be accurate.
13 hours down. So now you have glucose.
You are using it in the next step.
Yes. Naenda hadi 8:45.
Unaenda hadi 45. Tunasubiri time yenye tunaandika hapa start time yenye shift tunaingia. Not yours. That way, that is the only way he lapse haitapatikana. Because you see we are to deduct from accumulated minutes, taken time, carried forward. That that is the only way hatuta have kuulizwa hizi minutes zinapotelea wapi.
No, let me ask you. Total valid time available up to now. But it was 30. Then I took 34.
Taking the shift. Wengine yao si walikuwa na 4 hours. So you already had 20 minutes from their shift and 20 minutes from our shift.
No. The first shift, the first break I got. With the first one, I'm liking what I'm seeing. See? They'll just turn green and sometimes yellow. Are we together? If the concentration is lower. But for this one, this is brick red. This shows a high concentration of glucose. Are we together? So kudos. Now I need us to put the leaf inside the hot water. The purpose is to soften the leaf. Now remember that we are writing the procedure as we are doing it. I am seeing some people's spaces are empty, blank. We are writing the procedure as we are doing it. Can we put the leaf inside the hot water to soften it? We want to test for starch in a leaf. I've been requesting for white tiles, please. All right. Can we use forceps to extract? Do we have forceps? Can we have those phones on silent, please? Students, please. Please. Whose phone is that? Please, if you're not sure about your phone's situation, go and check.
Alarms.
Now, you're writing about the Benedict solution under test for starch in a full green leaf. It's supposed to be down here. Benedict's test. No, Benedict's test, right? So Benedict's test first and then Burette test. Please, the Benedict's test, you're recording it under 2.18. Yeah, not 2.17. Then now is when we are doing 2.17. Enoch, I thought we were drawing both cooling curve and heating curve. You drew both of them in one grid. The way I did on the board.
I think so.
Okay. Yeah. So if we could get the white tiles, kindly. So on what platform do we place the leaf? Filter paper. Now the purpose of putting the green leaf in hot water is to soften the leaf. Are we together? Now, I want you to look for a clean portion of your bench and place that leaf there. We also require forceps to extract the leaf from the hot water. We put the leaf there. Sorry? So you're going to sorry. Now, forceps. Lab guys, you're letting me down. I'm asking for things they are not being brought. Forceps, tiles. We are having a lot of lagging when it comes. We are having a lot of lagging. Where are they? Where are they? It's unfortunate. Now remember to be very cautious with ethanol because ethanol is flammable. So you should not heat the leaf in the ethanol on a direct flame. It is flammable. So once you extract the leaf, put it inside the ethanol so that we can extract the chlorophyll. And then place it with the boiling tube inside the hot water. Be very careful with ethanol and fire. Have you already put it in ethanol? Just put it directly in ethanol and put it in the hot water. Now, please kindly note the purpose of everything we are doing. I said the purpose of the purpose of heating the leaf is, number one, is to soften it. Are we together? The purpose of putting in the ethanol is to extract chlorophyll so that when we are now testing for starch using iodine, we can clearly see the color change. Now, unfortunately, the white tiles are not available, so I had said that you look for a clean part of your, when we place on a filter paper, the filter paper will influence the results because it already has starch. It's made of, from wood which has cellulose, and cellulose is a polysaccharide. Is that true? Science is everywhere. So you'll still observe the color change when you do that. Now using the forceps, I think once it boils, how will I remove? No, this cannot. It's hot. It's hot. It's not even working. There's no color change either. Huh? It's changing. Is it changing to blue-black? Mildly, very mildly. But that could be because from the time these leaves were plucked, they have not photosynthesized. Are we together? So they could be having very minimal starch in them. Now I need to see the findings for test for starch in a leaf. Is there ethanol still here? This is ethanol.
Yes.
You can actually pour the ethanol and leave the leaf in the boiling tube. And then put in a few drops of iodine. Now, what time did we pluck these leaves? Huh? How many hours ago? Over 6 hours ago? Yeah, so that is why it's giving us a negative result, because it has not been actively photosynthesizing. So can we repeat this later today, like at 2 p.m. Yes. with plants that are freshly from outside, that are just from photosynthesizing? Are we together? Yes. And then we are going to see same procedure, okay? Yes. But we do it like 6 to 8 hours from now, and then we see. So if there is any change, it's very, very mild. It's giving a negative result, and that can be accounted for. Okay, I think we can move on to the next thing. So the next thing is test for protein. We can switch off the gas taps. Thank you. Now we are testing for protein. And the test for protein is called burette test. And remember I said, I'm not designating time for writing. So you're writing as we carry out the procedure. All I want to see in your workbook is written procedures and observations. Jessica, okay? Written procedures and observations. I hope we have all switched off our fires, our gas fires. So if there's nowhere to support it, let it lie on the sink. It's mildly changing, yeah? Just small parts that have starch in them. Now the test for protein is called the burette test. You'll be provided with the food substance. I think, or you have it in the boiling tube. And then you have the burette reagent. Now the burette reagent is the blue. The sky blue reagent. So the burette test is very simple. Just take the protein sample or the food substance that you are suspecting to be protein and add the burette reagent. Just approximate. Are we together? Approximate like 5 drops, 7 drops, until you can see a tangible color change. Then we are going to record. So a positive one should give you a certain color. A negative one should give you another color. So I want to see what you get. Oh, you don't have it yet, yes? Yes, you may. No. The one you're being given now, sky blue, not the Benedict solution. Not the Benedict solution. It's a burette reagent. Burette reagent for a burette test. Testing for proteins. Wonderful. Perfect. Weka tena ingine. Hey, Damaris, please put it. Nice. So for the positive test, we see purple. Okay? Has everyone gotten a purple positive test for protein? Apart from this group, or you have it already? Yeah. So that's a good indication that we've been able to test for the food substances. Now, have you been recording the procedures and the findings? Now, please also indicate in 2.17 that we got a negative result as a result of having leaves that have not photosynthesized in the previous 6 hours. Yes, yes. Okay. Okay, okay, okay. Sorry? You want to? Go to? What was that? Oh. Yeah, yeah. Now, enough of biology, please. Group 4, please go for your rest. Is the other group back? We wait for the other group, please. All of them. All these are out. They are fine? Okay. Group 4, you can go for your rest. You look fresh. Now, let's put aside all biology for now. We go to structure and bonding. Yeah, structure and bonding is nice to do in the morning when the mind is fresh. Our minds are fresh, right? Yes. Structure and bonding. All right, we are in structure and bonding. Hey, you can. It's now theory for the next 2 hours. Yes. So for the next 2 hours, we'll be looking at structures and bonds. Are we together? I don't know why I get excited when it's structure and bonding. Huh? Fabian Omala. Good morning.
Good morning.
All right. Remove the hood. Take my advice. Just remove the hood. All right. Now, we talked about compounds. Are we together? All right? Yes. Okay? Now, we have different types of compounds. We have some that form from metals and non-metals, and we have some that are formed by non-metals bonding with each other, okay? So basically, for a compound to be formed, we must have element A bonding with element B, or even 3 elements bonding together. And I can see that we have so many structures that we are going to draw, indicating their bonds. Some have 2 elements, others have 3 elements, okay? So the first one is ionic bond, ionic bonding. Now, through ionic bonding, we obtain ionic compounds. Are we together? Now, of course, we know that a bond is a force. that is holding something together. Are we together? So let us define an ionic bond. Now, I need to hear your ideas. Yes, sir.
Ionic bonding is a bond formed.
Please tell them.
Ionic
Which are metal ions. And then we have, what's the opposite of cations? Anions, which are non-metal ions, okay? So the bond, the ionic bond is formed, by transfer of electrons. The ionic bond is formed by transfer of electrons. Are we together? From a metal atom to a non-metal atom. Now we cannot draw ionic compounds without drawing ions within the first 20 elements. So that's what I want us to do. Now go back to the table we did for electron configurations. So you are working hand in hand with this table, okay? Not the periodic table. The table we drew, yes. Subtopic 2.6. Subtopic 2.6. Now, how do we form positive ions? We start there. We start with the positive ions. I'll start with lithium. I'll start with lithium. Now, lithium is which configuration? 2, 1. Are we together? Now, what can we see here when it comes to the second shell? It has 1 electron, and how many should it have? 2. Gosh, 8 electrons. So is it easier for lithium to gain 7 electrons or to lose 1? Yeah, less energy will be spent to lose 1 electron than to gain 7 electrons. So in this case now, lithium loses this electron to give it a configuration of 2, but because now it has lost an electron, it obtains a positive charge. Are we together? It obtains a positive charge because now the protons are more than electrons since we have lost electrons, okay? So when you lose negativity, what do you become? Positive. Think of even in life, yeah? When you lose negativity, you become positive. So if we are losing electrons and electrons are negatively charged, then we get what? A positively charged ion. Are we together? Now the next example I'll look at is beryllium before I leave you to do two by yourself. Now beryllium is configuration 2.2. Again I ask, how much energy will we spend to gain 6 compared with the energy for losing 2? Which one is more, which one is less? Gaining is more, right? So let us lose 2 electrons to get and what charge do we have? Positive 2. Are we together? Now I challenge you to draw boron and sodium. I challenge you to draw boron and sodium. Ions. Remember, we are looking at the positive ions first. So don't mind about what I have skipped. I'm sure now we are moving to sodium. Feel free to refer to the table. Feel free to refer to the table. Have I tampered with something? Remove it. Okay. Okay. That. I'll be coming to check in a few. No. Remember, we have only lost 2. Boron is 2.3. That's where you got it wrong. 2.4 is carbon. Do corrections. So now, because you have lost this shell, rub it. So you have lost the whole shell. It was 2.3. Then now it is 2. So yeah, you have lost a whole shell. Rub it, then enclose whatever is remaining with. Are we set? You made. Now, how many have we lost? 3. So what charge will we have? Positive 3. Do you really want me to mark those diagrams that have been drawn with a pen, despite my many reminders over the past 13 hours? Please, let's redo the diagrams in pencil. Thank you. Let me see your boron and your sodium. Oh no, we are using this as a space, my dear. This. Leo uko na usingizi ya. Unasikia aje? Yeah, you can. It's gonna the same meaning. It has the same meaning. But plus 3 sounds mathematically correct. Let me see.
I've made a mistake here. In the last one.
Now, the electrons are on the shell. Yours are suspended somewhere in between the shells. And then, you don't draw the shell that has been lost. Okay?
Yes.
Yeah, because we had 2.3. Then we lost the whole of this shell because it was easier to lose it than gain 5. So that means you remain with boron with only one shell but a charge of positive 3. Yeah, and then draw your electrons on the shell like that. On the shell, not suspended, because that now will bring confusion when you are counting the electrons. All right. There are people who've decided that they'll just get everything right. You didn't want to draw in the workbook on the space provided. Draw ions within the first 20 elements. There is space provided there. Now, where is the innermost shell? The one that has 2 electrons. Yeah. You see now? Okay? Let me give you a half. Now, please proceed to draw magnesium, aluminium, potassium, calcium. No. How many have we lost for beryllium?
2.
2? Sema ukweli. Yeah, beryllium is 2. What about boron? 3? Good. Put it here, and then just write plus. Don't indicate 1, okay? When you just say plus, we know that we have lost 1 electron, okay? Thank you. Yeah, so same thing. Don't indicate 1. If it's 1, just write positive. If it's a negative 1, just write negative. Okay. Beryllium. Okay. So have I marked your boron? You need to draw boron. Remember, we are drawing all the positive ions of the first 20 elements. So why are you squeezing your work? There's a lot of space to write. A lot. Just use the book freely. Okay? I'm coming round checking magnesium, aluminium, potassium, and calcium. Calcium. Okay. It's okay? No, it was there. It's okay. All right. By the way, since we are doing theory for the next 2 hours, you can remove your lab coat. You see, I also removed mine. We are not doing a practical in the for the next 2 hours. Let me mark what you've done. So wrap this shell because it has been lost. Okay? Yeah, because we lost it, it doesn't exist anymore. Its loss has been accounted for by this positive. So, why can't you bring your books where I am, surely? Can you come one by one? Huh? Thank you. Now, what charge does boron have? Your boron does not have a charge. Your boron does not have a charge. +3. Okay? Come this side. Now, you are making very, very big mistakes. Huh? Now, how many electrons are here? 1, 2, 3, 6 electrons. And how many are supposed to be there in the second shell? Hey, you see now? So I can't give you a tick for that one. 2.8.8. Yeah, I think that's okay. So these ones I had told you to repeat. Okay. I'm okay now. Okay? Where is the first shell? Is it this one?
Yeah.
I think that's nice, Bilha. Let me have everybody's positive ions. Now, Clarice, I would prefer if you drew the ions on the shell. like if this is the shell, okay, I'll still mark it, yeah, because at least I understand what you mean. But if it was another examiner, probably it would. So what I'm saying is, if this is the shell, for example, if it's that, so draw the electrons on the shell, not somewhere suspended on a space. Okay?
Yes.
Okay. So where is the charge for this ion? Sawa. Charge for this is +2, and for this one is +2 as well. So in terms of number of electrons, correct. How they have been distributed, correct. All right. So we have said that if you lose negative, you become positive, okay? So losing electrons makes an atom a positive ion. Is that clear? Are we clear on that? All right, have I seen everyone's positive ions? Okay, great. Whose book is this? Okay. Some people, Deborah, Martin, are they here? Oh, I thought they were talking like they have been talking. Wow. Your potassium has 2 shells that have a pair of electrons each, because that is what I'm seeing. The inner one. Sasa, how am I supposed to know you're removing them? Huh? You're supposed to rub. You'd have just rubbed, eh? Assuming it was an exam, you would have missed out on something. Now, when I give you the exam on Friday, and you don't follow the things I'm saying, you lose a lot of marks. Please don't repeat some of these mistakes I'm seeing. Now, there's a shell you've left out for potassium, because initially potassium was 2.8.8.1. Okay? Allow me to put a wrong. How many people have I not marked their books? Only the ones standing? Hey, lakini your diagrams, man.
Not an artist.
Wait. Good. Please don't ignore any. Were we drawing silicon? I said you draw magnesium, aluminium, potassium, calcium. Hatufanyi silicon kwa sasa. So I'm seeing that you guys have grasped this concept, +3, because we have lost 3. Bramwell, are you not aware that we have lost 3? Wait. So this is? Okay. Which, okay, it's always good to indicate the chemical symbol inside.
Like the rest.
Yeah.
Okay, but I have a question here.
Yes.
The way you place this one is this. Ile uliandika huko umeandika ni kama oxidation number. Na iron na style kwa plus hapo mbele si normal.
No. It doesn't matter. Yeah, because +2, 2 plus, all means that it's a positive number, yeah? Yeah. So you write what you're familiar with. Now we have lost 2 for calcium, so it's +2, eh? Yeah, and that one just 1. Okay, can we now go to the negative ions? The anions, okay? So for the anions, we are looking at atoms that have more than 4. Now the ones that have 4, like carbon and silicon, those ones they share. Are we together? Yes. So let's not consider them. Let's look at the ones that have number of electrons that are over 4, yeah? And I think I'll start with nitrogen. I'll start with nitrogen that has an electron configuration of 2.5. In terms of energy, which one will I use more energy? Losing 5 electrons or gaining 3 electrons? Losing 5. Gaining 3 electrons. Are we together? Oh, yes. Sorry? The question is contradicting. Now, I asked, between losing 5 and gaining 3, which one will require more energy?
Losing 5.
Yeah, losing 5 I'll use more energy to lose 5 than gaining 3. So, I am gaining electrons. I'm gaining electrons, and so in this case, I become a negatively charged particle, an anion. All right? So can we draw this anion? So remember it was 2.5. Now it will be 2.8, but it will have a charge of -3 because we have gained 3 electrons. So we draw the first shell there, 2 electrons. Then we draw the first electrons that were there. All right? Then we use dots to indicate the 3 that have been gained. Are we together? Now, enclose with the box brackets and write a charge of -3. Imagine. Structure and bonding, very simple. Can we look at the one that is next to that? Which one is it? Refer to your periodic table. Or your table, yeah, oxygen. Oxygen has a configuration of 2.6. So we have oxygen, first shell, 2. Second shell, again I ask, between losing 6 electrons and gaining 2, where do I spend more energy? Losing 6, so I choose the easier option, which is gaining 2. So we gain the 2. We include the 6 that were there. Then we enclose it to show that now it is an ion with a charge of -2. Easy peasy, right? Wow. The next one is the 2.7. 2.7. So I'll want you to draw the 2.7. I'll want you to draw phosphorus, which is 2.8.5. Continue and draw sulfur, which is 2.8.6. So you're drawing the ions, the negative ions. And then lastly, draw chlorine, which is 2.8.7. So please refer to this for that. The concept is the same. Practice to perfection. So we got to 14 hours around 10 minutes ago. We are pushing on, eh? Yeah. So, who is done with the four anions? Remember, for anions, the rule is for you to show me that. The rule is for you to show me that the ones that you gained have a different representation from the ones you had before. I come. Okay. Oh, your space is full. Now, I said if you lose one, it's just negative. Sawa? Sirudie hiyo makosa tena. Hey, your circles are, hey, nowhere near circles. Huh? Where? They're like, yeah? 2.8 now. Okay. Is it? Okay. Now, fluorine is F. Peke yake. Bila L. F. Yeah, it's okay, don't worry. Where is the one you gained? It needs to be represented with something different. Because you gained one, yeah? If you check at mine, the ones that were there are represented with an X. Can I give you time to do that correction? Because I can see same mistake here, everywhere. Is this space full?
Yes.
Okay. Most of you have actually caught the... What's this? Oh, so, okay. Now, can we just proceed? Fabian, you're insisting I mark, eh?
Yes, the last one.
Oh, yeah. Hamna huruma? Hey. Hey. Now, how many have we gained?
Oh, yeah, it's 3.
Relax. How many have we gained?
We have gained 3.
Now, I can't see the ones that have been gained here because you have represented them all with X's. So I don't even know why I gave you a tick here. Please represent them differently. Yeah, see. Hey. So, the way you have squeezed it here, I'm supposed to see it and mark. See? Can I tell you something, you guys? All these learning materials are yours to use. Now this exercise book, if you don't feel it, where will you take it? Use them. Trish, have you seen this? Nice.
Explain what it is before I start giving is the solar system.
3. This one I already showed you. I repeat again and again and again. The ones that have been gained are represented using these dots of yours. They're very small. All right, so now let's go to dot and cross diagrams for ionic compounds. Now, I said that an ionic compound is an electrostatic attraction between a positively charged cation, of course from the metal side, and a negatively charged anion from the non-metal side. We are okay up to that point? So now, the first step we have taken is to draw the positively charged particles, the cations, all of them for the first 20 elements. And then now we drew the anions. Now we want to see these electrons that are being lost by the metal atom. Where are they going? And how are ionic compounds being made? So I have several examples here. Aki, surely. What is this you have drawn? This one? No. What is this? Now, where have you gotten that? Can you rub it? We are moving systematically together. Thank you. In fact, that is not correct at all.
Yeah, no, I was just trying to remember. I know I was wrong. I was just trying to remember.
Oh, thank you. Okay, so the first compound we are looking at here is sodium chloride. So I'll do like 2 with you, and then I allow you to do 2, and then we do the last one together, isn't it? Yeah, so the first one is sodium chloride. Sodium chloride. How many electrons is sodium losing? How many electrons does chlorine require to become a chloride? Very simple, yeah? So you just draw sodium here. First shell, second shell. Of course, we lost the third shell, so we are not drawing it. Then, next to it, not far from it, draw the chloride ion, which is 2.8. No, I should have used dots. Sorry. Two dot 8. So these are called dot and cross diagrams or Lewis structures. Are we together? Yeah. Then we gained 1 from the one that sodium lost. Now, between this and this is an electrostatic force of attraction, and that is what the ionic bond is. So if you want, you may indicate that there is transfer of an electron. Toehamwe. Are we together? So that is sodium chloride. Which one do you want me to do with you as a second example? Huh? I knew. Okay, let's do the fourth one together. Finish sodium chloride and then we go to the fourth one together. Okay, I think that's enough time to draw that. Now, what do you think is the name of the fourth compound? What's the name of the fourth compound, the one you want us to draw together? Now, I want others to try. The name of the fourth compound, which elements does it have? Yes, fourth? Fourth, not the KF. KF is 5. You have overthought. Okay, you're okay with the calcium and the nitride, but you didn't need to mention the 2, the 3, okay? It's just calcium nitride, okay? Now, that fourth compound is very, very easy to draw. Why? Because from the formula, you can already tell that how many calcium ions will you draw? 3, okay? Because it's Ca3. And you can already tell from the formula how many nitrogen ions or nitride ions will you draw? 2. So then now, whatever calcium will lose, the total of 6, because each calcium is losing 2 electrons. So that means in total, the calcium atoms are losing 6 electrons. And in total, the nitrogen atoms require how many electrons? 6. So we just distribute the electrons. Let's do it. So let's draw the calcium ions above and then the nitride ions below. The other alternative is to draw calcium, nitride, calcium, nitride, calcium, which I don't know if it will fit here, but we'll try. So I'm drawing the calcium 2.8 plus 2, 2 plus, still okay. So we have lost 2 here, okay? Now let me just draw the calcium ions above and then the nitride ones below. So I draw another calcium ion. Then the last shell. Then I draw the last one now. We are okay now? We have the 3 calcium ions. All have lost 2, 2, 2, totaling to 6. Now I'll draw a nitride here and another nitride here. So I'll have 2 Okay, let me use dots. For nitride, I'll have 2. I wanted to use dots. Then the 5 that it had, and then the 3 that it has gained to give us -3. Then we have another one here, 2. Now, if you don't know where these electrons are coming from, please ask. If you don't know where the electrons are coming from, why is she drawing? Some are X, others are dots. So now that is calcium nitride. Is it as hard as you thought? Ama, I let you draw first. Okay, now I challenge you to draw compound 2, which is magnesium oxide. And as we did earlier on, just ask yourself in advance, how many am I losing and how many should I gain? And then also look at the formula of the compound. It will guide you on how many, careful, it will guide you on how many ions you're drawing. Please draw compound 2 and compound 3. I'll be going round in 2 minutes. I'm breaking at 8:45 for 30 minutes. Kwa tuna confirm. Asante. Siwaeki. Actually, but you see in my head. In my head, nimejiambia ni 20. By the way, I want to appreciate you all for being such amazing students. You're very lovely, very positive. I can see how you're pushing yourselves. The first night is over. Huh? We have one more tough night. Okay? But tomorrow night will not be tough because we'll be knowing what we are waking up to, right? Yes. That's why I'm saying if there's any toughness, daytimes are not tough. Daytimes are not tough. I mean, like from when the sun came, I feel normal. So tonight we must fight to stay alive, okay? Yes. Martin, have you heard? Yeah. Okay. So keep drawing, keep drawing. We must fight to stay alive tonight. I know tomorrow night you'll be very enthusiastic because of what you'll be waking up to. How many had a rough time with this topic the first time you encountered it? Be honest. And what kind of experience are you having now? Or you can't predict yet? Huh? Where is that red pen? I'm marking magnesium oxide. I may not mark for everyone all the compounds. Let's agree first, especially here. They want me to mark everything. Now, have you realized that after the compound forms, they both get the same configuration, but they have different charges? They both become, like if it's 2.8, they all become 2.8, but the charge is what differs. I wish you drew this aluminium somewhere. Well, it's not wrong, but... Okay. Draw the KF. Good. At least I should see your magnesium oxide. So you see what you had drawn had nothing to do with what we are doing.
We make errors. We make errors, we can come back.
Yes, Indio. Yeah. High five. Good. Hey, his hands are cold. What? Good. 1, 2, 3. Oh, you have interchanged them. Magnesium has a charge of positive 2 because it has lost 2 electrons. And oxygen has a charge of negative 2 because it has gained 2 electrons. Please do the corrections.
They want to have the same configuration. For potassium, it will remain 2 straight, and for this, that's the same configuration.
Not same configuration. Not same configuration because initially this was 2881. Now it will be 288. And then this one was 27. It will be 28. So they won't have the same configuration. The only thing is that one will give an electron, the other one will gain it. So we'll draw 1, 1. Go to KF, Potassium fluoride. Hii tulifanya na nyinyi. What are these numbers here representing? Hey! Why are you lazy? Please, let me show you this. Did you see all the work I did there of drawing 3 calcium ions? Huh? Yes. And then I drew 2 nitride ions. So this guy decides to draw 1 calcium ion and then writes 3 before it to show me that he has multiplied by 3. Eh? Look. Can you imagine? This is a shortcut. Mbaya sana. Surely, take a photo of this, please. Thank you. Huh? Imagine. So what do you call this? Instead of drawing the 3 ions, he drew 1. Then he wrote a 3 there. Bramwell. You, Bramwell. Thank you. Now, if I've not marked, are you okay? Should I mark? I'm coming there. Now, you need to draw 3 chloride because the formula is aluminium and 3 chloride ions, okay? Now, you have drawn magnesium with an extra shell. Ideally, or rather, magnesium as an atom was 2.8.2. So, when it loses, you're just supposed to have 2 shells. The first one with 2, the other ones with 8. So please rub this, otherwise everything else is correct. You just need to rub that extra shell and then draw the other chloride ions. 2 more. Okay. Now if it gains 1, just write negative. Don't include this one. You just write negative. If it loses 1, write positive. That way we'll know it has lost or gained 1. Good. Group 1, please go for your health break. Is there a group that is outside? Oh, wow, you're all here? Amazing. These boys here are insisting I mark everything. Ama, that is my work. Whose book is this? Wait, let me mark hers first. Now, the formula says AlCl3. So that means you're supposed to draw 3 chloride ions. You've only drawn 1. You have also not drawn calcium nitride. You can move to the front for purposes of copying what is on the board and then come back. Feel free. You're at home. Yes. Yeah, so I need 2 done, aluminium chloride and calcium nitride. The formula will guide you on how many atoms you're drawing. Okay? How come you have 2 books? Oh. Now, your ions are too far from each other, so they look independent. They don't look like it's a compound, okay? That's why I insisted, draw them next to each other, all right? I think it's best if you do corrections so that I don't put a tick on something that does not look 100% correct. No, don't put this here. It's not a reaction. Sawa sawa. And then you can push next time, push this one closer. All right? But otherwise it's correct. You'll have to draw 3 chloride ions. Okay. Hey. My students. Aya, come here. There's confusion here. Sawa? Aya. Si ushajua? Uko sawa? Okay. Damaris, you've become a very good chemistry student. Only.
Can't believe it.
Okay, now don't indicate 1. If we lose or gain 1, just put the sign. Yes. At the ambulance. At the ambulance. Yeah, let me mark for you, then you go.
It should be visible.
Wow. Now draw the potassium chloride. So I think we are okay with Lewis dot and cross diagrams for ionic compounds. I'm not seeing anything that should make me panic. You can go. Have mercy on me now. Diana. And you've been quiet the whole night. Diana, draw the ions closer to each other to indicate that there is a bond. You see? When you draw them apart, it's like you're drawing ions independently. But when you draw them close to each other, you're indicating that there is a bond. Right? Yes? How many are we? How many are we? We should not be less than 10. Yes. How many are we?
Yes, 17.
Okay. For how long? You're sure?
Yeah.
Okay. marked 1. Lenny, have I marked even 1? I've marked 2. Okay, then we are safe. I don't like your diagrams. Who is this? Amol. So you draw nice diagrams like the way you look. Now, aluminium chloride, the formula is already guiding you on the number of ions to draw. You're supposed to draw 3 ions. All right? Yeah. So that one you'll do corrections, 2.8.8 plus 1 here. Good. I'll mark. Okay. Don't draw them apart from each other. Yes. Complete this. Alvin. Unaandika pole pole. Oh, okay. All right, now let's describe the giant lattice structure of ionic compounds. Now, what do you understand by the term? Actually, what do you understand by the terms giant first? Giant, think about it scientifically in terms of particles. Guys, tuchangamke. In terms of particles, when I mention giant in a chemistry class, what do I mean? Yes, just say what is in your mind. Yes. Uh-huh. Mm-hmm. Mm-hmm. Yeah, so basically means a big structure. Now also in chemistry, because we are talking about bonds, it also means many bonds, millions of bonds. Are we together? Many bonds involving many particles and many atoms, all right? So ionic compounds, we are writing as I explained. Ionic compounds have a giant lattice structure. I have provided you with the diagram for that. Now, lattice refers to the arrangement of the atoms of a substance in a 3D space. Lattice structure refers to the arrangement of the atoms of a substance in 3D space. Now, in a lattice structure, the atoms are arranged in an ordered and repeating fashion. Now, look at the diagram I have provided for you there in the workbook. The red atoms, or rather, sorry, the red ions represent the sodium ions, and the green particles represent the chloride ions. When you look at how it is, it's a regular arrangement of alternating particles that are negatively charged. True or false? Are you able to clearly see that from the diagram? Yes. So we are alternating red, which is sodium ion, and green, which is chloride ion, alternating. And then the interconnecting lines now are showing you the bond between those oppositely charged particles. Are we together? So that is the lattice structure of sodium chloride. Now I can see some people have not written much about the lattice structure, so I'll repeat the statements so that you write. Lattice structure refers to the arrangement of the atoms Lattice structure refers to the arrangement of the atoms of a substance in 3D space, three-dimensional space. In three-dimensional space. And again, I've said, they are arranged in an ordered and repeating manner. They are arranged in an ordered and repeating manner. So in an ionic compound, we have a regular arrangement of alternating positive and negative ions. So you're basically describing that diagram. In an ionic compound, we have a regular arrangement of alternating positive and negative ions. So with that explanation and the diagram there, that is sufficient enough. The last statement. In an ionic compound, there is alternating arrangement of oppositely charged particles. And that's just one example we have given of sodium chloride. So now the next part is about the physical properties of ionic compounds. Now, you should be able, by the end of this discussion here, you should be able to state the physical properties and the appearance of ionic compounds and explain that behavior in relation to the bonding of, please do what we are doing. You explain that behavior based on on your understanding of the bonding. Like when I say that they have a high melting point, are you able to relate that feature or that property to bonding? Are you together? Okay, so the first one. Ionic compounds are usually solid at room temperature. Ionic compounds are usually solid at room temperature. What explanation can you give me in terms of bonding? What explanation can you give me for that in terms of bonding? Yes, Clarice?
They have strong ionic bonds.
Yes, they have strong forces holding them together. When you go back to the particle theory and what we said about solids, you remember? We said that solid particles are held together by strong forces. All right? So the fact that we mentioned electrostatic forces here, it means these are forces strong enough to hold the particles together, and that's how we end up with ionic compounds being solid at room temperature. So it is the property with an explanation to do with bonding. Jelimo, are we together? Yes. Now the second property. Now we are writing the property and also the explanation in terms of bonding. So they are usually solid at room temperature because of the strong electrostatic forces holding the particles together. So you have not just said it, you have also explained it. Now the second property is that they have high melting and boiling points. Now, are you able to relate that to bonding? Someone share their findings? Yes.
electrostatic forces that
Speak up.
the ionic compound have a strong electrostatic forces which require a lot of energy to break.
Wow. Hey. Clap for him. They have strong electrostatic forces that require a lot of energy to break. So as we are trying to supply that energy to break those bonds, we end up melting these substances at very high temperatures. All right, property number 3. Ionic compounds conduct electricity only in molten or aqueous states. Allow me to write for you the spelling of aqueous before you write your own things. Molten or aqueous states. I know you have the answer. You are burning to tell us the answer, right? Now, ionic compounds, I've said, only conduct electricity in molten state or aqueous states. What is electricity? There is no electricity. What is electricity? Bramwell. Flow of current. Which current? Electric current through a conductor. Hizo zote, you're correct, all of you. What else? And also, for electricity to be there, we must have charged particles that can move to carry the charge. Are we together? Now, when you look at the lattice, do you think those particles are free to move in the solid state? No, and that's why we can't conduct electricity using a solid ionic compound. Yet, it has charged particles. It has positive particles and negative particles. The negative particles are the ones responsible for flow of electricity. But the moment you take that sodium chloride and melt it, when you melt the sodium chloride and it turns now to the molten state, it means in the liquid form, the particles can move. We said for liquids, particles can move over and under each other. Did we say that? Yes. So that means that in molten state or aqueous state, aqueous means dissolved in water. The moment you make them liquid, then there is flow of electricity, there is movement of the charged particles. In your own words, write that point. Conduct electricity only in molten or aqueous state because it is in these states that the charged particles can move. Simple. Then we move forward. Now, you should also have an explanation as to why they are poor conductors in the solid state. Why are they poor conductors in the solid state? Because the particles are compacted and held together by very strong forces. Okay? And so there's no movement of any charge. And that way we cannot conduct electricity. Now there are other properties like number 5. Number 5 or 4? Okay. Which number are we in? 4. Okay. Number 4, they easily dissolve in water because they are polar. They easily dissolve in water because they are polar. Polar meaning that they have a positive aspect, which is the positive ion, the cation, and the negative side of the compound, which is the anion. So polar compounds dissolve in other polar substances. So for example, water is polar. H2O is a polar substance. It is polar because we have hydrogen ions in water, and we have which other ions? Hey? No. I'm talking about the ions in water. Hydroxide ions, good. So does this make water polar? Yeah. And then something like sodium chloride is also polar. So polar substances easily dissolve in what? Other polar substances. And the ionic compounds are polar because they have cations and anions, and they so easily dissolve in water. Most of them, that's number 5, most of them have a crystal-like appearance. Most of them have a crystal-like appearance. Crystalline appearance. And I think that's it for ionic compounds. Let's breathe in and out. You can stand up. Breathe in and out. Stretch, bend a bit. Huh? How are you feeling? 15 hours on? Yes. Munaenda nje sana. Kati. All right, let's sit down and start on covalent bonding. Or we are not sitting to relax. Covalent bonding. Do you have energy to rub the board? Rub it for me. By the way, you can work while standing. Even if you want to work while bending like this, you are allowed. As long as we are working. Okay? Yeah, as long as we are working. Yeah, because it will reach a point I'll teach you when I'm like this. You'll bear with me. Okay, let's go to covalent bonding. I said we are doing theory, so you can remove your lab coats. All right, let's continue covalent bonding. Now, covalent bonding is achieved, or a covalent bond is achieved by sharing of electrons between nonmetal atoms. Sharing of electrons between nonmetal atoms. Now, when these atoms share electrons, they all achieve a complete configuration. Okay? So they share electrons in such a way that the final compound or the final molecule, all the atoms are going to have a complete configuration. Are we together? So you're going to see this as we do more examples. Place it on this tool. As we do more examples, but I've already explained that it is through sharing of electrons. So in this case then, if we are dealing with nonmetal atoms only, then we don't have cations and anions. We also do not have electrostatic forces of attraction. We don't have electrostatic forces of attraction because electrostatic forces of attraction are between oppositely charged particles. So we don't have ions in this case. We are just having atoms sharing what? Electrons. Again, remember we said for ionic compounds we are transferring electrons from the metal atom to the nonmetal atom. So I believe the difference is starting to be clear, isn't it? Now let's talk about single bonds, double bonds, and triple bonds. Teach me chemistry, teach me chemistry. What are single bonds? What are double bonds? What are triple bonds? Deborah, single bonds, double bonds, triple bonds. I agree. But what are they? What's a single bond?
Single bond is an atom and an atom.
You know, sometimes when you know too much chemistry, those simple questions, you are unable to answer them. Huh? Single bond. Yes, Clarice?
A single bond is a bond that is formed when a valency of one of an atom joins another atom with a valency of one. One electron in the outermost energy level, another electron.
No. Don't think about it that way. Don't think about it in terms of valencies, okay? Now, a single bond is formed when only one pair of electrons is shared. A single bond is formed when only one pair of electrons is shared, and it is represented by one line, like a hyphen, okay? So single bond. Only when one pair of electrons is shared, and it is represented by one line, a hyphen. Now, double bond, now it's easier to know what a double bond is. A double bond is when two pairs of electrons are shared and is represented by two lines. You see? You're moving on very well. Now write what a triple bond is. Write what a triple bond is. And what we use to represent a triple bond. Then after that, now we go to draw the exciting molecules. Are you excited? Are you sure? I'm so excited when I see the molecules that are awaiting for us to draw. I can't wait to start distributing the electrons. Can you wait for us? Please talk to this guy. Who is his friend? Bramwell. You, talk to your friend. Tell him to calm down. He's rushing and he won't finish before us. We are all going to wait until the clock goes stop. Thank you, Bramwell. All right. I'm sure now we have finished defining single bonds, double bonds, and triple bonds. Now let's draw some exciting, simple molecules. Say yay! Yay! The first one is hydrogen. Hydrogen molecule. I want to say something about hydrogen. Within this class, you will see me treating hydrogen as an nonmetal. Are we together? Yes. But because of some of its behavior sometimes, I will treat it as a metal. So there are times you will see me putting a positive charge next to hydrogen because as at that time in that context, hydrogen has behaved as a metal. Are we together? Yeah, so don't go saying, you know, she was even confused. All right? Yeah, I've said that to protect myself. Anyway, now hydrogen has how many electrons? Only one. So that means that if I am forming a hydrogen molecule, we will share between the first hydrogen and the second hydrogen. Each will contribute one electron. So that means that this hydrogen, are you seeing the way I've covered it? This one here. Okay. This one, it now has two. So does it have a complete configuration now? Yes. Now, when you look at it in terms of the second hydrogen, it also has how many electrons? Two. So does it have a complete configuration now? Yes. So this connection where they are sharing electrons is now the covalent bond, right? So if you want to represent it structurally in terms of the bonds, this is a single bond because we are sharing one pair of electrons. So it's hydrogen, single bond, hydrogen. Sawa sawa? Yes. Now, molecules that only have two atoms are called diatomic molecules. Diatomic molecules. Di means two. Are we together? And we have many examples. We have oxygen. I want to list them here for you just so you know them. We have oxygen, it's diatomic. Nitrogen. Fluorine. Chlorine. Why can't we say chloride here? My students in the high school say chloride. Chlorine, chloride. Huh? Why can't we say chloride here? Because it is a molecule. Chloride is an ion. Are we together? Yeah, so these are examples of bromine, yes, iodine. All these are examples of diatomic molecules. So sometimes in the examinations, I see students confusing the molecule and the atom. So sometimes when they are calculating, for example, the mass of oxygen atom, they end up calculating for what? For the molecule, which now gives them a double figure. because now they'll have to do 16 plus 16. Yeah, so those are some of the mistakes I see being made a lot. The second one, or which one do you want us to do together before you can attempt the others? I know you'll choose. I know the one you'll choose. I just want to see if it's methanol. Now, we'll do nitrogen first. Okay? And then I want you to crack methanol. Try and do it by yourself. See, you just share the electrons. It's nothing complex. Okay, let's go to nitrogen. So please pay attention here. Please pay attention. Now, there's a student I'm going to move from where she's seated. She knows herself. So, last chance. I'm going to move her to come and sit here. Anyway. Now, nitrogen, the electron configuration of nitrogen is 2,5. All right? So that's the first nitrogen, that's the second one. Now, if this one gives 1 and this one gives another one, how many does this one remain with? It each had 5, isn't it? Yeah. So they have all given 1, 1. So how many does it remain with? 4, then 5, 6. Is it now full? No. Can we try and contribute more? Yeah. So we give again 1 and another one. So we remain with 3 outside, okay? So it will be 3 plus 4, which is 7. Is it complete? No. Let's share again. So we remain with 2 now, because we have given out 3 and we had 5. So 2 plus 6 is 8. And on this other side we have 2 plus 6 is 8. So that shows you that nitrogen molecule has a triple bond, indicated like that because they are sharing 3 pairs of electrons. Okay? Thank you. Now please draw carbon dioxide, oxygen. My work will be to go around verifying your work. Now for purposes of, giving you reasonable work, only draw the outer shell. Are we together? Yeah. Because it's the outer shell that participates in the sharing. Okay? Miss Jessica, are you okay? Have you drank water? You need to. Okay? Mm. Please don't draw tiny things that will give me a hard time when I'm marking. Yeah, you can. As long as it's clear, it's clearly indicated. I'm sure some have drawn a few. Drawing, I see. Mm-hmm. Mm. Yes, yes. So that mtu akihesabu, when someone is counting, they count everything to indicate that now the configuration is full. Mm. Okay, can we do it differently this time? Okay, let's do it a bit differently. Instead of me marking, okay, we exchange books, all right, and correct each other. It's called peer assessment. Assess your peer. So finish drawing the, how many molecules? 1, 2, 3, 4. Yeah, finish drawing the 4 molecules, and then we assess each other. If there is one that we are unable to crack completely, we do it together as a class, okay? Yes. We assess each other. Where are the sticky notes? Yes?
I was explaining to
Oh,
my colleague.
okay. How can I help you? You okay? Imagine we are heading to 9 a.m. Hey, it's already mid-morning. Hmm? It's already mid-morning, so. Now for every atom you draw, please make sure you label it. Don't just draw circles, please. Label it. Label it. Now what are these? Which ones are dots and which ones are crosses? So just draw a nice cross and a nice dot. Now assess each other, okay? If you are not sure that somebody deserves a tick, call me, I'll tell you whether they deserve it. Okay? Assess each other in pairs. Feel free to discuss. Inaingia kwanza kwa wingi, inaingia Now we must make a model of a molecule. Are we together? And the molecule we are going to make a 3D model of methane. Are we together? We'll make a 3D model of methane. Actually, this side, make a 3D model of nitrogen. All right? Just show me the 2 atoms and the triple bond. Are we together? And then this side, make a 3D model of methane. We are going to supply you, I think you should be having some molding clay with you. Now, if it is a compound, then the atoms must be differently colored and most likely different sizes. Are we together? So you'll be provided with skewers. I don't know if we have toothpicks. If they are there, well and good. So the skewers and the toothpicks can be used to show the bonds. So one skewer can indicate a single bond. All right? So we are going to supply you with half a manila paper to use it as the base for your model. So the ones on my right, you're making a model of methane. The ones on my left, you're making a model of nitrogen molecule. Show me the covalent bonds. Show me how interesting and how creative you can be. Now, please carry the models of the atoms and place them on the benches we are not using, so that when we get visitors, they can see what we have been doing. Are we together? You dismantled yours. Don't dismantle, carry it. Carry it and place it there. It even has your names. Yeah, someone can enter and say, the ones who made these ones, I'm giving you 200,000 each. Amen. Yeah? Aya, nyi hamjui kutafuta hizi vitu. The president can come in here and say, hey, everybody who made this, come for a check. Anyway, that's not the aim, isn't it? Yeah, sure. But if it comes, yeah, please supply us with skewers, half manila paper. Enda. Now, I want to say something. From now going forward, I'm not giving anyone permission to go out. You're only going to use your break. Number one, you're not drinking anything in the lab, isn't it? Yeah. Secondly, I am staying longer than you, and I am older than you. Do your mathematics. If an older person can endure, you who is younger, is it a good reason to use as an explanation, really? Now, flatten that manila paper. Flatten the manila paper. Flatten the manila paper. Flatten it nicely. Start on your model. Now, it doesn't have to stick on the manila paper like the way we did for the other one. For the other one, it was easy to stick because it was a board. Okay? So for this one, you can just use it as a base, but it can be a 3D, something that we can look at all round. Okay? Feel free to use the resources that have been availed to you. And you can move from bench to bench, checking the resources they have, stealing a few skewers here and there. Okay, anything that is on your bench that you don't need, place it on the benches on the edge. Like the tripod stands. Just clear. Remove the Bunsen burners. That one is stuck, eh? That's okay. Let it stay. Can someone else do this, please? But they are student managers to do that. Sikuitana, like. But we don't have to call out their names. Juu inafanya tutumie time kufanya other things other than learning.
So we just let it pass.
Not let it pass, but student managers. Do you know the ones who are missing, who they are?
No, we do that. That's when we want the list. Because from 1 a.m. to less than 20.
Less than 20.
20, 21, 22. So there are 4, 5. So we don't know whether they disappeared, they're not coming back. We don't know.
But they can't come back.
Yeah,
If from 1 a.m. hawajakuwa. It's okay, they can fall off. It's just unfortunate that they are falling off with our merchandise. Oh, but yeah. Yeah. No, what are student managers one? Now, please don't start the models before you finish assessing each other on the molecules. Sorry? Just go around. You must find. Are there more plasters in bars that have not been used? In the other big carton? What are you making? Nitrogen. How many are you? Notes. Are you done? Yeah, so probably we can have stills of this. We can have stills of the models before we transfer them to. So you're done? I had looked at the previous one. I almost advised you, but how many are you?
1, 2, 3, 4, 5.
Okay. All right. So diffusion is still taking place, eh? It's still taking place, regardless of how slow it is. You're 5. So we can have one here. Write your names on those papers. Ensure that an image of the model with the names is that upside down. It's not. Okay, I had advised you flatten, but it's okay. Yeah, so I think most of you have gotten the idea. From your models, I can tell you have gotten the idea. Those ones on this side were able to make models of the diatomic nitrogen molecule that has a triple bond. So kudos, clap for yourselves. You're really trying. Or rather, you're doing it. Are we doing in the next few hours we won't have somewhere to even write on? Huh? Now, you can clear the plants as well. Anything you do not need, put in the sinks. Like anything that requires cleaning, like that, you can put in the sinks. The plants, dispose them in the dustbin. Dispose the plants in the dustbin. Any gloves, any used gloves, that is, dispose them in the dustbin. And then quickly we look at properties of simple molecular compounds. Properties of simple molecular compounds. So that when I come back, we'll look at giant covalent structures, okay? Properties of simple covalent compounds. Now, property number 1, covalent compounds have low melting and boiling points. Covalent compounds have low melting and boiling points. Low melting points and low boiling points. Low melting points and low boiling points. This is because minimal energy is required to break. This is because minimal energy is required to break the weak intermolecular forces. Minimal energy is required to break the weak intermolecular forces. Now, the weak intermolecular forces are also called the van der Waals forces. Now, I'll explain what van der Waals forces are. If this is a water molecule, and there are many water molecules, okay? Inside the water molecule between oxygen and hydrogen, we have covalent bonds. Are we together? In the molecule, one molecule, we have covalent bonds between hydrogen and oxygen. But in between a molecule and another molecule, we have weak intermolecular forces. Are we together? Yes. So it is these intermolecular forces that require minimal energy to break. And that's why you find we have some covalent compounds that even melt at negative something degrees Celsius. Are we together? Yeah, because they need very little energy to break the weak intermolecular forces. Actually, that is one of the main differences between molecular compounds and ionic compounds. Ionic compounds have high melting and boiling points. Molecular compounds have low melting and boiling points. All right? Now, most of the covalent compounds exist as gases or liquids. Most of the covalent compounds exist as gases or liquids. Most of the covalent compounds exist as gases or liquids. And this is still attributed to the weak intermolecular forces. So the weak intermolecular forces are not able to hold the particles tightly together for them to actually be a solid. All right? Now, they are all non-conductors of electricity, except graphite. They are all non-conductors of heat and electricity except graphite. They are all non-conductors of electricity except graphite. Now, what do we call substances that do not conduct electricity? Insulators. So you can also mention that they are insulators. So every time you're describing a property of a compound or a molecule, you're supposed to describe in relation to what? To the bonding. Are you together? Yeah, you're supposed to describe in relation to the bonding. It doesn't make sense to just say they have high melting points. So what is the chemistry behind it? All right? Now, as the molecules increase in size, the melting and boiling points generally increase. As the molecules increase in size, the melting and boiling points generally increase? Basically because there are more bonds to be broken. There are more bonds to be broken. So the more bonds there are to be broken, the higher the melting point and the boiling point. Especially when we start looking at organic chemistry, you will see short chain hydrocarbons and long chain hydrocarbons, indicating that the melting and boiling points are increasing as the size of the molecule increases. Now, is there any question about covalent compounds, simple covalent compounds, before I tell you what next? So I'll see you in 20-something minutes. Bye. Bye. It's been 6 hours since 2:45.
CPF is a financial services company that offers a whole host of solutions to the Kenyan public and across the globe. We have been recognized both locally and internationally as a trusted brand. And the following one of the solutions that we have developed internally as CPF is called Rukisha. Rukisha is a concept that we developed primarily to address people in the informal sector, people with small businesses, people with big businesses as well. And the intention is to help them save towards their retirement. We have incorporated a concept called Save As You Spend into the platform, where for every transaction you make, you're able to save a percentage of it. And over time, you're able to see your retirement funds accrue. One of the other benefits of utilizing Rukisha is you also have access to others in the CPF group of products, among them being investments through the CPF managers. You should be able to save towards a specific goal under the CPF asset managers. You should also have access to the CPF insurance products under the laser insurance brokerage services, as well as the real estate products within the laser property management services. For more information on Rukisha, visit www.rukisha.com. You can also access it through the short code *391# or download it in the Google Play Store as the Rukisha app. Talk to us, reach out to us, and we shall help you achieve your retirement goals.
CPF Financial Services, we've been able to package the solution for both for the individual, for the corporate, and also for groups. At the individual level, we've come up with products that are able to take care of the member both at before retirement and after retirement. For before retirement, we have training for the member to be able to prepare themselves to go out there and be able to save for themselves. Also at that particular level, they can be able to save individually on a continuous basis with as low as 50 shillings every day or 500 per month to be able to take care of themselves at the point of retirement. At corporate level, we have solutions where we carry out fund administration for corporates that have already existing schemes, or they can be able to join our umbrella DC schemes within this called Taifa. We also do gratuity administration to assist corporates be able to administer the gratuity plans for their different staff within the organization. We also have solutions for chamas where we can be able to open up opportunities for them to be able to save and invest as chamas and also enjoy welfare solutions which are always covered under our trust fund called the Momba Trust Fund. As part of our solutions under the management and training arm, we also do management consulting. Under management consulting, we consult for you to do governance audits, we do strategy development, among other consulting services that you might request. We also do trainings. Under trainings, we have trainings that are bespoke to the organization, or they can already be able to come in at best prices to attend some of the trainings that have already been packaged by the CPF arm. For more information, visit our website on www.cpf.or.ke, or you can be able to access the information on the CPF app on the Play Store or the iOS Store.
Prayo bwana, asante kutupeleka holiday bwana. Lakini na swali, uliniambia tunasave na CPF. Kwani ukisave unapata pesa wapi?
Boni, unajua first una save, halafu una spend. Nilikuwa nishaweka kitu CPF kabla sherehe.
Oh, okay. Na kama sina kitu this month nafanyaje?
Usistress. Ukiwa na CPF, unaweza convert Safaricom Bonga Points zinakuwa contribution yako kwa M-Pension. Na ni rahisi sana. Dial *289# au piga 0111 114 000 na CPF watakusort.
Hello, my name is Faith Wanjiru Kinyanjui. I'm a 4th year student here at Jomo Kenyatta University of Agriculture and Technology. I'm pursuing a degree in horticulture, and this is my final semester. So I lost my dad in 2018 when I was in first year, second semester, and it was a 1-week illness. After his death, my life was kind of difficult because I didn't know if I was going to continue with my studies because my mom is a housewife. And so when I joined second year, my first semester, my mom introduced me to Lab Trust after she received some phone calls from my dad's workplace. And so we went with her because I'm usually here in Nairobi. So when we went, they assured me that they were going to pay my school fees and I didn't have to worry. So that was like a saving grace for me. And since then, my school fees has always been paid. My upkeep is always in check and my house is always in check. I've never lacked anything since then in second year, and up to now I'm in my 4th year, final semester. And as you can see, due to Lab Trust,
welcome to Heaven Trinity Motors, your premier destination for quality cars and exceptional service. At Heaven Trinity Motors, we bring you a heavenly experience in the world of vehicles. Discover a world of possibilities with our range of fresh import used cars. Each vehicle meticulously inspected and handpicked to ensure you drive away with confidence. Whether you are looking to upgrade, trade in, or sell on behalf, Heaven Trinity Motors is your trusted partner. We offer competitive rates for your trade-ins and ensure a hassle-free process when you choose to sell with us. Explore convenient bank financing options tailored to suit your needs, making your dream car more accessible than ever. Call us today on +254 707 073 551. Heaven Trinity Motors. Your journey starts here.
Introducing Champion Security, where protection meets peace of mind. In an ever-changing world, security is paramount, and we're here to ensure your safety with unmatched expertise and cutting-edge technology. Whether it's securing your home, business, or event, our team of dedicated professionals is committed to deliver top-notch solutions tailored to your specific need. With 24/7 monitoring and rapid response capabilities, you can trust us to be vigilant guardians. Don't leave your security to chance. Choose Champion Security and take the first step towards a safer tomorrow. Contact us now to learn more about our comprehensive security service and join countless satisfied clients who have made the smart choice for their protection needs.
In an uncertain world, trust is everything. Introducing Tamka Security Services Limited, your trusted partner in security solutions. At Tamka Security Services Limited, we understand that security is not just about protection, it's about peace of mind. That's why we specialize in providing comprehensive security solutions for homes, residential areas, and malls. Our team of highly trained security guards equipped with state-of-the-art technology ensure round-the-clock surveillance and protection. From guard services to K9 units, from installing electrical fences to providing quick response in times of emergencies, we also offer bodyguards and event security. Tamka Security Services Limited is your one-stop solution for all your security needs. With our commitment to excellence and swift action, you can rest assured that your safety is our top priority. Choose Tamka Security Services Limited for security you can trust. We are located at Rimpa along Magadi Road, and you can contact us on 0202 595098. Tamka Security Services Limited, protecting what matters the most.
CPF is a financial services company that offers a whole host of solutions to the Kenyan public and across the globe. We have been recognized both locally and internationally as a trusted brand, and the following are some of the reasons why you should be working with us. Currently, we provide fund administration services to the largest schemes in the country. One of them is Lab Trust, which is the oldest. Having been established in 1929, it currently has a fund value of 30 billion Kenya shillings and membership of about 20,000 members. The County Pension Fund, which is also one of the largest in the county space, and the Public Service Superannuation Scheme. Overall, we have total assets under management of 100 billion. One of the biggest challenges in the social security sector has always been enabling people save towards their retirement. And one of the solutions that we have developed internally as CPF is called really to address people in the informal sector, people with small businesses, people with big businesses as well. And the intention is to help them save towards their retirement. We have incorporated a concept called Save As You Spend into the platform, where for every transaction you make, you're able to save a percentage of it. And over time, you're able to see your retirement funds accrue. One of the other benefits of utilizing Rukisha is you also have access to other solutions within the CPF group of products. Among them being investments through the CPF asset managers. You should be able to save towards a specific goal under the CPF asset managers. You should also have access to the CPF insurance products under the Laser Insurance Brokerage Services, as well as the real estate products within the Laser Property Management Services. For more information on Rukisha, visit www.rukisha.com. You can also access it through the short code *391# or download it in the Google Play Store as the Rukisha app. Talk to us, reach out to us, and we shall help you achieve your retirement goals.
At CPF Financial Services, we've been able to package the solution for both for the individual, for the corporate, and also for groups. At the individual level, we've come up with products that are able to take care of the member both at before retirement and after retirement. For before retirement, we have training for the member to be able of retirement. At corporate level, we have solutions where we carry out fund administration for corporates that have already existing schemes, or they can be able to join our umbrella DC schemes within this called Taifa. We also do gratuity administration to assist corporates be able to administer the gratuity plans for their different staff within the organization. We also have solutions for chamas where we can be able to open up opportunities for them to be able to save and invest as chamas and also enjoy welfare solutions which are always covered under our trust fund called the Momba Trust Fund. As part of our solutions under the management and training arm, we also do management consulting. Under management consulting, we consult for you to do governance audits, we do strategy development, among other consulting services that you might request. We also do trainings. Under trainings, we have trainings that are bespoke to the organization, or they can already be able to come in at best prices to attend some of the trainings that have already been packaged by the CPF arm. For more information, visit our website on www.cpf.or.ke, or you can be able to access the information on the CPF app on the Play Store or the iOS Store.
Brayo bwana, asante kutupeleka holiday bwana. Lakini na swali, uliniambia tunasave na CPF. Kwani ukisave unapata pesa wapi?
Boni, unajua first una save, halafu una spend. Nilikuwa nishaweka kitu CPF kabla sherehe.
Oh, okay. Na kama sina kitu this month nafanyaje?
Usistress, ukiwa na CPF unaweza convert Safaricom Bonga Points zinakuwa contribution yako kwa M-Pension. Na ni rahisi sana. Dial *289# au piga 0111 114 000 na CPF watakusort.
Hello, my name is Faith Wanjiru Kinyanjui. I'm a 4th year student here at Jomo Kenyatta University of Agriculture and Technology. I'm pursuing a degree in horticulture, and this is my final semester. So I lost my dad in 2018 when I was in first year, second semester, and it was a 1-week illness. After his death, my life was kind of difficult because I didn't know if I was going to continue with my studies because my mom is a housewife. And so when I joined second year, my first semester, my mom introduced me to Lab Trust after she received some phone calls from my dad's workplace. And so we went with her because I'm usually here in Nairobi. So when we went, they assured me that they were going to pay my school fees and I didn't have to worry. So that was like a saving grace for me. And since then, my school fees has always been paid. My keep is always in check and my house is always in check. I've never lacked anything since then. In second year and up to now, I'm in my fourth year, final semester. And as you can see, due to Lab Trust, I'm able to carry on my project. Here is one of them, and I'm studying the plant diseases, and I'm using a treatment known as silicone. Then I'm controlling the disease to see if it is going to be controllable. And this has become my passion, and I love agriculture so much. Right now, I'm in my final semester, and by next year in January, I'll be done with school. And my plan is, if I'm going to get a job, I can join the scheme, the Lab Trust scheme, and ensure even if when I'm going to have kids later in future, their lives are going to be catered for even in my demise, because CPF has done that for me, and I'm very grateful. Because if it weren't for them, I don't know if I could be even here in school. And thank you so much, CPF, for fulfilling our lives. I have two sisters at the moment, one in college and the other one is in grade 4 in primary school, and they have been paying for their fees as well. And we can say our life has never been the same, and we are so happy for CPF and how they have helped us. And thank you, CPF, for fulfilling our lives and making us smile again after everything.
In an uncertain world, trust is everything. Introducing Tamka Security Services Limited, your trusted partner in security solutions. At Tamka Security Services Limited, we understand that security is not just about protection, it's about peace of mind. That's why we specialize in providing comprehensive security solutions for homes, residential areas, and malls. Our team of highly trained security guards equipped with state-of-the-art technology ensure round-the-clock surveillance and protection. From guard services to canine units, From installing electrical fences to providing quick response in times of emergencies, we also offer bodyguards and event security. Tamka Security Services Limited is your one-stop solution for all your security needs. With our commitment to excellence and swift action, you can rest assured that your safety is our top priority. Choose Tamka Security Services Limited for security you can trust. We are located at Rimpa along Magadi Road, and you can contact us on 0202 595098. Tamka Security Services Limited, protecting what matters the most.
His Excellency Mike Mbuvi Sonko, the Good Deeds Ambassador, crowned for his generosity in helping the needy. Sonko's move to help the needy is his way of life and started way before he joined politics. Hundreds of needy people from all walks of life can attest to this. Mariam Abdallah has been in and out of hospital, but Governor Sonko has always been there for the family.
Namshukuru sana mheshimiwa, yeye na familia yake, anawaombea sana kila nikiswali, Mwenyezi Mungu awajalie kila la kheri.
Natoa shukrani zangu kwa Mheshimiwa Sonko kwanza. Mwenyezi Mungu amjaze kila la heri. Amuondoshe matatizo yake, mazito amfanye mepesi. Familia yake Mwenyezi Mungu aijalie salama. Amenifariji mimi na amenisaidia mtoto wangu. Sikupata usaidizi wowote Mombasa zaidi ya kwamba ni hao katika wale wataja. Hamna mheshimiwa yote alikuja kunisaidia. Lakini nasema Mwenyezi Mungu yuko pamoja na yeye. Tumekuja naye karibu vizuri. Alhamdulillah, na ubaguzi. Ni mwanamume ambaye sijui niseme nani. Anifadhili kwa kila kitu.
Tunakwambia rudi after 2 months, lakini umetupatia karatasi ile kule Mombasa. Unajua umeshona na chuma ile kutumia. So watamtoa.
Umeshakatia ndege?
Ndege kila kitu zile kupasha shughuli kabisa.
Sawa. Kidogo ya dawa na supu damu ile. Haya, shika tuenda kununua naye dawa. Sawa.
Kwa majina naitwa Mariam Abdallah Kombo kutoka Mombasa. Nimeshukuru sana kusaidiwa na mheshimiwa. Operesheni zile zangu za kwanza alinisaidia nikafanyiwa, na hizi za pili tukarudi tena pia amenisaidia yeye. Namuombea Mungu sana yeye na familia yake. Mwenyezi Mungu ampe afya, ampe umri, ampe nguvu na amlinde na azidi kuwa na imani ya kusaidia.
Not once or twice has Governor Sonko intervened when the elderly are being neglected by the community.
Wewe nani mheshimiwa?
Hapana,
hapana, wewe kuja mama. Wacha kufanya hivyo.
Hapana, mama, wacha mama ingia ndani yake. Hapana, hapana. Hapana, haiwezekani. Twende mami.
Sasa ile
hapana
hapa.
Nimembariki hapa. Nimempa baraka na mbariki zaidi. Apande juu. Amen. Ile yote tutaweza. Ubarikiwe na Mungu. Usikutane nangu.
Asante.
Asante sana. Baraka kubwa.
Hakuna mtu atakutoa hapa. Sasa uko na coach na uko naye.
Times society neglects elderly people. A 100-year-old granny in Embo was forced to spend nights in jail for contempt of court, unable to pay a fine of 100,000. Sonko came to her rescue. Same story for a 106-year-old granny who was evicted from her land in Pangani, but Sonko helped her get it back. Several other people have also gotten back their grabbed land through the help of Governor Sonko.
Itakuwa registered kwa hiyo title yako na utarudishiwa hii shamba. Shamba yako itarudishwa kwa sababu tu register hii imetoka kotini. Kwa hivyo itarudisha hiyo shamba kwako sasa. Yes. Please. Hiyo tutafanya mara moja na itarudi. Sawa.
What I have to say is that today
I have managed to have access to Mheshimiwa, which not many people can have. I think it was God's grace that maybe our dad had done something very good for us for today Mheshimiwa to be standing here and assisting me, because we just seen Mheshimiwa in the papers, we hear about him, but today I've seen it with my own eyes what work he does.
He has such a compassionate heart.
Ninaguzwa sana na moyo wake wa huruma.
You know there is politics.
Kuna siasa.
And there is a core.
Na kuna mwito.
This man is not a politician.
Huyu si mwanasiasa.
When I watch the way he handles the poor people,
ninapoona jinsi anawashughulikia watu maskini,
then I know
basi najua
there is something extraordinary in his life.
Kuna kitu kisicho cha kawaida ndani yake.
Governor Mike Sonko, through Sonko Rescue Team, has continued with his charity work, which he began even before joining politics. Meet baby Kyla, a 1.5-year-old baby. Governor Sonko met Kyla's mother in Westlands, who narrated her daughter's woes to the governor. Sonko was touched by the story of young Kyla, who had lost her eyesight at a tender age and vowed to help her seek medical attention in India. Weeks later, she is back after the surgery. Kyla was received by Nairobi County First Lady after landing at JKIA.
Wow, you can see salama. God is good. This is a miracle. Wow. Baby Kyla, God bless you. Wow.
The day she was born, I noticed that she had some problems with her eyes, and I came to meet Sonko, and I told him about my story, and he decided to help me.
Beautiful baby Skendi Chelangat, whose life was threatened by a cancerous tumor in her eye, Sonko took the initiative to pay for her medical attention in India. She is now a happy child in school. It is a story of joy.
Today, so thank you Sonko for everything you've given me. May God bless you. Thank you very much.
Late March 2019, Governor Sonko donated tons of relief food to the drought-stricken counties of Turkana and West Pokot. The governor traveled by road to Turkana, where he distributed the assorted foodstuffs and set a medical camp for the needy.
He has brought a lot of food. We have gotten maize meal, vegetables, fruits, water. He brought bowsers, water, rice, sugar, all different types of food. I want to also appreciate the quality of food because people who are hungry and weak are not able to chew hard food like maize. And so the maize meal that he has brought.
Unajua hatuna miles, usifanye hiyo iko. Nisikia? Sawa. Tutatumia peroxide na liver. Peroxide 5 volumes na 20 volumes. Okay. So unauliza wanafunzi they count the number of bubbles. Okay. So. Good morning. Good morning. Are we set? Yes. Are you sure? Yes. Okay, we move on. Or you want to sleep? Yeah? 16.5 hours. Who wants to sleep? I've not slept. I've not slept since yesterday in the morning. I know. Well, lazima ujikaze hadi mwisho. Now let's look at giant covalent structures. Are they being captured by the camera? These two? Yes? No. For real? Forgive me. Forgive me. Come, I hug you. Come, I hug you. Come. Just come. You're resisting my hug. Yeah, I'm surprised. Let's go to giant covalent structures. Giant covalent structures. Is there any empty bench from this side? And any empty sitting space? Huh? No, for the ones who have been there. And there? Ah. Would you like to come where the camera is? Come here. Martin, any space over here? Any empty space? Sorry? Okay, let's go to giant covalent structures. Giant covalent structures. Now we need to be settled. We need to be settled as much as possible. Now, as I had mentioned earlier, giant covalent structures, when you see the name giant in chemistry, we are referring to a structure that has many atoms and many bonds. Are we together? Now, in this case now, we are going to look at diamond and graphite, and we are going to compare and contrast them. So we have diamond on this side, and we have graphite. Okay, so did you know that both diamond and graphite are all made of carbon? Yes. You knew? Yes. So what do we call them? Allotropes of carbon. Now, what does allotrope mean for those probably who do not know what it means? What does an allotrope mean when we say allotropes? Now, allotropes are substances made from the same element, all right? Allotropes. So these are substances made from the same element, but the bonding of the atoms is different, giving them different chemical and physical properties. I repeat again, these are substances made from the same element, but the bonding is different. So it gives these substances different chemical and physical properties. So physical properties basically mean how it looks, how it behaves on the external, the melting point, the boiling point, and then chemical properties, how it reacts, and the kind of compounds it can form, etc. So we are looking at diamond and graphite in this case. Now, when it comes to diamond, give me that lid. When it comes to diamond, now diamond involves one carbon bonding with 4 carbons. One carbon bonding with 4 carbons. Are we together? Now I'm going to show you this bonding just so that you understand. So that when I'm talking about the inability of diamond to conduct electricity, you'll understand from this. So 1 carbon bonding with 4 carbons. Okay? And because it is a giant structure, each carbon also bonds with 4 other carbons. So like this one now, this one has bonded with 1, 2, 3, 4. Are we together? And this one has bonded with 1, 2, 3, 4. Okay? And this one the same, like that. That's how we end up with a giant lattice. But for purposes of this illustration, let me wrap these other bonds. And assume this is what we have. So we have a carbon bonding with another carbon, 1, 2, 3, and another carbon here, and another carbon here. Okay? Are you seeing? So it's this that I have illustrated. It is this that I have illustrated using the circles to represent the atoms. Are we together? Now, please focus on this middle carbon. Now, what is the electron configuration of carbon? Sorry, someone? Yes, Clarice? 2, 4. 2, 4. So that means that if this carbon is bonding with these other 4, it is contributing 1 here, 1 here, 1 here, and another 1 here. So is it remaining with any extra carbon? No. And this is what happens to every other carbon. All the external shell electrons are used to bond. Are we together? So that means that there is no extra electron that has not been used in the bonding. Now, based on what I have said, do you think diamond can conduct electricity? Based on what I have said, can it conduct electricity? No. Why? Why? Yes. And you guys, you have removed your tags. I still need your tags on, please. Thank you. But you can explain as you put it on.
Because they lack delocalized electrons.
They lack delocalized electrons, or they lack free electrons. All right? Yeah, because we said that what is electricity? Movement of electric current through charged particles. So in this case, we don't have any charged particles because all the charged particles that we had, which are electrons, we have used them, we have shared them out, all of them. So basically, there is nothing remaining in terms of charged particles. Are we together? So that is diamond. Now, all these carbons are held together by strong covalent bonds. Are we writing? All the carbon atoms in diamond are held together by strong covalent bonds. Now, I want you to underline the word strong. I want you to underline the word strong, because when I get to the physical properties of diamond, you will apply that aspect of the strong covalent bonds to help you explain one of the physical properties. Okay? Just underline it and leave it there. All right. Now, diamond forms a tetrahedral structure. Tetra means 4. Does it make sense, based on what I was explaining, to say that it forms a tetrahedral structure? Yes, because it is a carbon bonding with 4 other carbons. Are we together? Yeah, so it forms a tetrahedral structure of interconnected bonds, interconnected covalent bonds. So now I would want to talk about the physical properties of diamond. Physical properties of diamond. We are not yet on the uses. We are still on the physical properties of diamond. Now, diamond is the hardest known substance. Diamond is the hardest known substance. This is because the carbon atoms are tightly packed and held together by strong covalent bonds. You remember I told you you'll remember the strong covalent bonds? Yes. So diamond is the hardest known substance because the carbon atoms are held together by strong covalent bonds. So for you to say that this is a diamond crystal, those are millions of carbon atoms held together by strong covalent bonds. Are we together? All right. Now another property is that it appears crystalline. It appears crystalline due to the tetrahedral structure. Due to the tetrahedral structure and arrangement of the carbon atoms. Due to the tetrahedral structure and arrangement of carbon atoms. Due to the tetrahedral structure and arrangement of carbon atoms. All right. Now, diamond has a high melting point of 3,550 degrees Celsius. Can you imagine? That's very high, yeah? It has a high melting point of 3,550 degrees. Have you ever dipped your finger in boiling water? I'm sure you have tried to do this. I personally did try when I was young. Anyone who has ever tried dipping their hand in boiling water? How did you feel? Okay, not boiling water. Have you ever gone to open the cooking pot and then the steam burns you somewhere? Yes. How do you feel? Most likely that was 100 degrees. Now think about 3,550 degrees Celsius. That's a very high temperature, yeah? That's a very, very high boiling point. Now, who could tell me the reason for this? Why do you think we have such a high melting point for diamond? Yes, Sarah? Because of the strong covalent bonds that require a lot of energy to break. Let's write that down. Now, one thing you need to note about diamond is that diamond does not have intermolecular forces. There are no intermolecular forces in diamond. It's just strong covalent bonds. Right. So I have given you the diagram for the giant covalent structure of diamond and how the carbons bond with each other to form the tetrahedral structure. Now let's talk about graphite. Let's talk about graphite. Let me wrap this because it was for my own illustration. Now graphite is also made of carbon, and in graphite, one carbon atom bonds with 3 carbon atoms. Now look here. So it's a carbon bonding with 3 carbon atoms. Are you together up to that point? So in terms of atoms, I'll draw a carbon atom here, another carbon atom, and another one here. So carbon, 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 carbon. 1 carbon bonding with 3 carbons. Now, again, the electron configuration of carbon is 2, 4. 2, 4. So if this carbon gives 1 here, and it gives 1 here, and gives 1 electron here, what happens? Sorry? There is 1 electron remaining. Sawa sawa? So that 1 electron remaining is a free electron, and it is a charged particle that exists within the structure of graphite, and that is why we say that graphite is the only nonmetal that conducts electricity. Do you now understand why? Yes. Now, because of this kind of bonding, of a carbon bonding with 3 carbons, we get the hexagonal structure. Please look at the hexagonal structure in your workbook. So we form layers of hexagon. What does hexa mean? Hexa means 6, and 6 is a multiple of 3, right? Yes. So it makes a lot of sense for the hexagonal layers. And between a hexagonal layer and another one, we have weak intermolecular forces. Now, there goes one difference between diamond and graphite. We say that in diamond, we only have, what do we have in diamond? Strong covalent bonds. But in graphite, we have strong covalent bonds and weak intermolecular forces. All right? Now, because of these layers, and then a layer intermolecular forces, another layer intermolecular forces, these layers easily slide over each other. They easily slide over each other, and that makes graphite to be used as a lubricant. All right? So what I want to talk about now is the uses of diamond based on the structure. So if we are using it for jewelry, what chemistry is in that? Is it just because it is beautiful? Shiny? But we also say metals are shiny. Expensive. Show me the chemistry in that. Huh? All right. Now, I've already said that diamond does not conduct electricity. It has a very high melting point, and then it is extremely hard and dense. Sawa sawa. Now, diamond's hardness makes it useful, all right, because it is used in drill bits, tips of drill bits. That one I have to write. Tips of drill bits. Now, I wish I had a drill to explain with, but assuming this is a drill, are we together? Assuming this is a drill, we always fit the bits, different bits of different sizes. So the bit is actually the one that now rotates. Are we together? So the drill has a motor, and then we fit the bit there. Okay? So the tip of that bit, the tip of the bit, the one that will actually come into contact with the substance that is, or rather if it's a wall or a metal that is being drilled, they normally coat it with diamond. Now, why can't you use this pen to drill the desk? Can you try? Why can't you use this pen to drill the desk? It will break. Other than breaking, why will it break? Why will it break? Think science, think science. Now, you can't drill a substance with a substance that is not harder than it. Does it make sense? Yes. Yeah. You can't drill a substance. So this pen, or rather this tip, is not harder than the desk. Okay? So you cannot use it to drill. But if I come here with something that is harder than this desk, I can use it to drill the desk. So that's what happens. So when we want to drill steel, we use the hardest available substance, and that is what? Diamond. Okay? So the hardness of diamond, as a result of the strong covalent bonds, makes it to be used as drill bits. Now, diamond has a sparkly appearance. That's why I was a bit uncomfortable with shiny. Thinking about it, it's metals that we refer to as shiny, all right? So, due to its sparkly appearance, it is used for making jewelry. Now, diamond occurs naturally as an allotrope of carbon, and it is a non-renewable resource. So when you mine it, it doesn't form more where you have removed it from. So that makes it very expensive, okay? Because of the uniqueness and the scarcity of it. And even the resources that are used to mine diamond, those are reasons why it ends up being very expensive. Now let's look at uses of graphite. Now remember, we are talking about uses in relation to the structure and the physical properties, all right? Now I talked about graphite having hexagonal layers, and that those layers can slide over each other. I mentioned that, right? So that is why graphite is used as an industrial lubricant in engines and in locks. Graphite is used as an industrial lubricant in engines and in locks. Now, it is also used to make inert electrodes for electrolysis. Electrolysis is a whole topic that we'll cover. It is also used to make non-reactive electrodes for electrolysis. Sorry? Okay. Ikifika hiyo time. Forty. All right. Is there anything else we would like to say about graphite? Any contribution from the class? Any other use of graphite? Yes, Martin? Sorry? Used in making pencil leads. Used in making pencil leads. Thank you. I had forgotten that. Used in making pencil leads. Now, let's look at metallic bonding. This one is just a short topic. Now look at the diagram I've given you for metallic bonding. Now metallic bonding basically explains to you, okay, how a metal bonds within itself, okay? So most students confuse metallic bonding with, like a metal is bonding with another metal. No. Metallic bonding refers to the bonding that occurs within the metal. Are we together? Now, when you look at the diagram I have provided there in the workbook, you can see that because metals form cations, cations are positively charged. All right? Now, how do we form cations? How do we form cations? Sorry. How do we form cations? By loss of electrons. Now, when those electrons are lost, where do they go? Where do they go if there is no ionic bonding happening? When you look at that diagram, you'll know where they go, and you'll give me an answer. Any idea, Diana? Any idea? Now they become delocalized electrons. Thank you. Sorry, Sarah, I saw your hand too late. They become delocalized electrons, and they exist in between the positive particles, which are the positive ions, the cations. So then, metallic bonding is the electrostatic attraction again between the positively charged metal ions and the negatively charged delocalized electrons. I repeat, metallic bonding is the electrostatic force between the positively charged cations, between the positively charged cations and the negatively charged delocalized electrons. Delocalized meaning they are not located, they are just within the structure. We actually call them sea of electrons. The sea of electrons. So like for example, if I'm looking at something like sodium, okay? So we form a giant metallic lattice. Giant metallic lattice, to me, we are alternating the positive particle and the negative particle, the cation and the electrons, the cation and the electrons. So those electrons that are pending within the structure are the ones that are responsible for conductivity of electricity in metals. So metals conduct electricity because of the presence of delocalized electrons. Are we together? Can we write that? Now, I'll briefly look at properties of metals in reference to bonding. Properties of metals in reference to bonding. So, I'll give you a property, and based on what I've told you about the bonding of metals, you'll tell me why it has that property, why metals have that property, okay? Now, number one, we are filling in the workbook. Number 1. Metals have high melting and boiling points. Feel free to discuss with your friend why metals have high melting and boiling points and give me the feedback. Oh, drinking. Maybe usiku at night. Now I said that metals have high melting and boiling points, all right? High melting and boiling points. So what is it about the bonding of metals that causes them to have high melting and boiling points? Raise your hand, please, yes? There's a strong electrostatic force between the cations and the delocalized electrons. Perfect. Now, what do you say about it in terms of energy to justify the high melting point? What do you say about that, what he has said, which is very correct? Yes, Sarah? Yes, yes, yeah. So your answer has completed hers. Are we together? Yeah, so we require, your answer has completed his. Yes. Why? You need to be careful. Huh? Yeah, sorry, Bramwell. Yeah, so it is because of the strong electrostatic forces that we need to give a lot of energy to be able to break these forces. So for every property, you give an explanation. For every property, you give an explanation using the chemistry of the structure and the bonding. All right? Now, we go to another. So a lot of energy is needed to break these bonds. Let's go to another property. Metals conduct electricity. Property number 2. Metals conduct electricity. Now, in terms of structure and bonding, I'd like to hear your views about this. Jelimo, you're quiet on me. Yeah, because metals have free electrons that move through the structure and carry charge. Now, which charge does electrons carry? Which charge do electrons carry? Negative charge. And that is the charge we need for conduction of electricity. So that's amazing. So when electrons can flow, electricity is conducted. Always remember that. When electrons can flow, electricity is conducted. All right. Let me give you another property, and then I can give you a minute to discuss and tell me the chemistry behind this. Metals are malleable and ductile. Metals are malleable and ductile. Take a minute, 60 seconds. What is malleable? What is ductile? Give me feedback after this. Sorry? They didn't make, but we have made 2 models so far. I feel like making a third one. Which group is willing to give me feedback? Yes, Marston?
From our group, we have said that malleability is the ability to coil in the wire, and ductility, the ability to be hammered into sheets or reverse.
Isn't that explanation the same? Something that is foldable can be hammered into shape, right? So, Marston, both your explanations are talking about malleability. Yes, Bramwell?
Can say the malleability is as we are saying, but the ductility can bring the aspect of when it's being hammered, it will not break, no matter the extent it takes.
Thank you for trying, Bramwell. But that is not right. There is another description for ductility. Now, when we say that metals are ductile, it means they can be drawn into wires. All right? I can give you a metal, and we are able to extract wires from that metal. Are we together? Yeah, so please note that down. Now, what is the chemistry behind malleability? We have said malleability is ability of metals to be bent into shape, hammered into shape without breaking. What's the chemistry behind that in terms of structure and bonding? Any idea? Lucy, any idea? Sorry? Delocalized electrons. Okay, what's the relationship between delocalized electrons and malleability? Is there a relationship? Who can give us a relationship between Are you raising your hand? Yes. You know, I'm just seeing his hand doing like this near me. Okay. Haron, tell the class. Please tell the class.
So the relationship between the malleability is due to the strong electrostatic force that act. No.
But we want to hear. Just bring, I'll hold it.
Okay. Malleability it is due to strong electrostatic force of attraction. Force of attraction between the metal in the lattice. So when it is hammered, it cannot break easily.
Someone else with a different explanation? Someone else? Yes, Omar.
This is due to the ability of the electrons during hammering to slide against each other, giving them the distinct shape, like the wires.
Yeah, your answer makes a lot of sense, and it is actually correct. There are layers, but the layers are not just layers of delocalized electrons, okay? Remember, it is a layer of positive particles, the cations, and then a layer of delocalized electrons. That's how it forms a lattice. Remember, we said that a lattice is a regular arrangement, okay? And then we interchange the layers. So that is what makes them to be able to be bent into shape because of those layers that slide over each other. Are we together? Now, which words will we use to describe that? Can I give you the words for that? The wording for that point. All right. So layers of positive ions. Remember the property we are describing is malleability and ductility. So layers of positive ions can slide over one another and take up different positions. Now this makes it possible for metals to be hammered and bent into different shapes. This makes it possible for metals to be hammered and bent into different shapes or drawn into wires without breaking. Or drawn into wires without breaking. So I think I would want to stop on properties of metals there. Now, let us make a table. And I'd want you to do this in pairs or 3. Make a table comparing metals and nonmetals in reference to physical and chemical aspects. Work together. At least give me 6 differences per group. And then once you're ready to present, you let me know. Maximum of 4 in case we are not able to get 3. Sorry? Sorry? A health break now. That's what they've been told. Is that what they've been told? Okay, the student manager should advise. Please let's make the table for differences quickly. Welcome. 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 So now there's a lot of commotion, number 1. Number 2, there's a lot of people passing in front of the camera. Okay. So can you watch that? I think now our camera is even those St. John's ladies, they are just. Sorry? Okay. Sorry? Okay, but the student managers should manage that. You don't even have to follow up. They should do that. If it's one student, it's okay. Sorry? Sorry? Group 3, you have not gone for a health break? Since when?
Since 6:20.
Now, group 3, please take a health break. Group 3, please take a health break now. Whether you've finished the table or not. I understand you have gone for almost 6 hours without a health break. Please go. And please move faster. We need to continue with the class. Kindly. All right. Let me hear what you have gathered. Oh, you're still in the first property. Okay, let me see what you've been able to come up with. If you are in a group, draw closer to them. Yeah, so that you're able to discuss. Are you guys okay? Mko sawa? We'll break at 3:17. Clarice, are you in a group? Are you working as a group? Okay. Okay, I'd like to get feedback. Can we avoid passing in front of the camera kindly? All of us, please. Let's avoid passing in front of that camera, please. By all means. Can I get feedback from you concerning the differences between metals and non-metals? So from this group, who is presenting? You are? Okay. From this group, who is presenting? Yeah, so you just give us two from each group. So let's start here. Give her your attention.
The first difference, metal has high melting and boiling point, while non-metals have low melting and boiling point. The second difference, metals conduct electricity in solid state, while non-metals conduct electricity in molten state. Sorry. Am I clear?
Please say the second one again, kindly.
In metals, they conduct electricity in solid state, while non-metals, they conduct electricity in aqueous state.
Let's discuss the second. Let's discuss the second difference. So you're saying that metals conduct electricity in the solid state. Do we all agree? Are we in agreement with that part? Metals, yeah? Yes or no? Yes. Do metals conduct electricity in the molten state? In all states, yeah? So we agree with your first part. But your second part about non-metals conducting electricity in molten state, I want the class to give us their feedback, please, on that one. Yes? It's only graphite that conducts electricity for non-metals, because non-metals don't have charged particles. Are we together? Yeah, so you can amend that. Non-metals are actually non-conductors except graphite. Okay, let me go to the next group. Who is speaking on behalf of this group? Gideon.
metals are made up of
Let's listen.
metals are made up of metallic bonds, while non-metals are made up of covalent bonds. Metals have a giant metallic structure, while non-metals have a simple molecular structure.
You can say non-metals have both simple and giant, because we have the giant ones, right? Like silicon 4 oxide is a non-metal compound that has what? Giant covalent structures, okay? Yeah, so this group, you have majorly shown, or rather you have majorly talked about the chemical properties, all right? You have talked about chemical properties. Now can we have Clarice and Jelimo, probably one of them, commenting about physical properties, physical differences between metals and non-metals.
Okay, I'm not sure about this. I think metals are naturally occurring and non-metals are not.
Now, listen to her statement, those who are still discussing. Say your statement again.
Metals are naturally occurring and non-metals are not.
So are you saying non-metals are man-made or something?
Yeah.
Like oxygen is man-made?
Okay.
If you want to know
sure.
if it's correct, think about specific elements and you'll see where it's wrong. Okay? We say that elements are naturally occurring, whether they are metals or non-metals. Are we together? Yes. Why don't you just say the simple, you know, that's why I said when you know a lot of chemistry, these simple things, they can overwhelm you because of their simplicity. Who is speaking on your behalf? I? How do we know who is speaking? Okay, Martin. So are you in their group as well? Yes. Okay.
The metals form cations and non-metals form anions when they react.
Is it all non-metals that form anions?
Except the noble gases.
Ah, you needed to mention that.
And they have a shiny lustrous surface when freshly cut, the metals. While non-metals don't have
So we can say that metals are shiny and non-metals are dull. Okay? So we could have non-metals that are brightly colored, but when we are talking about the aspect of being shiny and not shiny, we can comfortably say that metals are shiny and non-metals are dull. Are we together? This is quite interesting. You know, when I gave you this exercise, I thought it would be so simple. But I'm hearing answers that are communicating something to me. Who is speaking on behalf of this group?
So all of our points have been exhausted, so
all your points have been exhausted.
if they have any that they haven't mentioned, they can add up, but we are also, unless you have another question.
Okay, I'll ask you another question as you have requested. All right. Well, here comes the real deal. Yes, my dear.
Metals gain stability by losing electrons, while non-metals gain stability by gaining electrons.
Yes, but not all again, because we have the noble gases. She's asking whether you can comfortably say that metals gain stability by losing electrons. That you can comfortably say, right? And then she's saying that non-metals gain stability by gaining. It is correct, but you have to mention except the noble gases. Thank you. Now, if there is a point that has been mentioned and you did not write, please note it down. Have as many differences as possible. Have as many differences as possible. Now, here comes the real deal. We are starting stoichiometry. I am smiling because of how people fear this topic, and it's my favorite. It's an amazing chemistry topic. Stoichiometry. Now, stoichiometry is basically calculations that involve amounts, okay, masses, volumes, all right, concentrations, etc. Calculations that involve, what did I say, the first one? Amounts, volumes, masses, concentrations, and the last one is percentage yield of reactions. Percentage yield of reactions. So within this topic, we are going to calculate all those things, and it's going to be fun. Okay? Yeah. It's going to be nice. Just wait and see. Now, we'll start by defining valency. Now, valency of an element is the number of electrons its atoms lose. For example, the valency of sodium is 1. Why? Because it loses 1 electron. And the valency of chlorine is also 1 because it gains 1 electron. Now, what is the valency of carbon? 4. You're right. Why? It either gains or loses? No. You know you're being watched worldwide. You can't say that. Yes, Master? Yes, yes. It shares 4 electrons. So, again, we say that valency is number of electrons an atom loses or gains or shares. Are we together? Yes. So when I say valency, you'll know what I mean. Now, I want us to come up with different formula for different compounds based on their valencies. Are we together? Different formulas for different compounds based on their valencies. And I'm not going to use complicated atoms. I'm going to use atoms within the first 20 elements. We've been interacting with atoms within the first 20 elements for almost 3 hours now. So we are okay with that, right? Now, the first one is calcium nitride. Now, remember we are referring to the valency. Now, what is the valency of calcium? Yes? 2. Positive 2. So the calcium ion has positive 2. What about the nitride ion? Sorry? What's the nitride ion? Negative? No. Please confirm. Go back to where we drew an ions, please. Nitride. Nitride. Please, if you know the answer, say it confidently. Eh? This here is for forcing. Force your answer to be correct. Negative 3, thank you. So nitride is negative 3. Now, it's so easy to get the formula for calcium nitride by knowing the valencies of the different elements. Now, how you get the formula is just by interchanging these valencies here. Are we together? Yes. So that the ratios balance, and you'll see why. So we have that. Are we together? Now, when you go back to the calcium nitride compound that we drew under ionic compounds, you will understand why switching the valencies balances out the ratios. You'll understand why. Because now you'll see that if each calcium is losing 2 electrons and we involve 3 ions of calcium, then how many electrons do we have to give out to share? Not to share, to transfer. 6, okay? Now, each nitrogen requires how many? 3. So do you think exchanging the valencies like that has balanced? Yeah? Yes. Very simple. So I want us to do a few more examples of coming up with different formula for different compounds. Did you think it was that easy? You knew it was that easy? Just a minute, I open my book here. All right. Now, you can know the valency of an element by knowing the group in which that element belongs to. Now, how do you know the group an element belongs to? How do you know the group an element belongs to? By?
Knowing its position in the periodic table.
By knowing its position in the periodic table. But I thought the group is the one that will tell you the position in the periodic table. Sorry?
If you know the number of electrons and its electron configuration, then you can tell the group.
Yes. If you know the electron configuration of an element, then you can tell the group, and as a result of that, you can now get the valency. Are we in agreement with that? Yes. So, I want us to write the formula for hydrogen sulfide referring to the valencies for hydrogen and sulfur. So the first one we'll do is for hydrogen sulfide. The other one we'll do is for aluminium oxide. Now all these are within the first 20 elements. So using valencies, I know you may be knowing it, but do it again using the valencies. So we are doing for hydrogen sulfide and now is there anyone stuck with valencies for the elements I have mentioned? Now if you're stuck with valencies, go back to where we drew the ions, the cations, the anions, and even where we drew the electronic structures. So that way, you'll be able to know the valencies and then write the formula of the compounds. Now, what is the formula of hydrogen sulfide? H2 S. Now, I'll just explain it again here. The valency for hydrogen is 1. What is the valency for sulfur? Okay. So we said to balance the ratios, you switch the valencies, and now you get that. What about aluminium oxide using the valencies? Yes? Al2O3. Where is the 2 coming from? From the oxygen's valency. And the 3? From aluminium's valency. Great. I'm going to write for you 4 formulas on the board, and then you're going to tell me which one is correct, which one is incorrect, and the one that is incorrect, you'll write it for me correctly. So I'll write for you the 4. We are still introducing stoichiometry. We have A as that. Please, can you try and crack this individually, please? Tell me which one is correct and which one is wrong, and the one that is wrong. Now, let me give you a hint. When the compound contains a metal and a non-metal, the name of the metal is given first, and the symbol of the metal is written first. That's a hint. Are we together? And if it is hydrogen, it's hydrogen that is named first. So look at the 4 and tell me what is exactly happening here. Let me hear your feedback. Let me hear your feedback. Damaris is very serious. She's trying to crack the matter. Yes.
I think all of them are wrong.
Marlon thinks that all of them are wrong. Please go to the board and tell us about the first one. Just the first one. Who else agrees that all of them are wrong? Huh? The valency for barium is positive 2. Are we together? Yes, probably some people did not know. Please pay attention over there. Tell us whether it's negative. Yes. So at least we have an explanation for the first one. For the second one, what's the problem? Huh? Omol. Yeah? No, go and write it the correct way.
When naming the compound, you should start with the metal and then the non-metal, which will be
all right. Thank you. Okay. Okay, someone come up and tell us about the third one. The third one has major mistakes. I think Bramwell can let us know about that one. By the way, if you feel like I'm not giving you an opportunity, let me know. Okay? Please. Eh? Am I favoring some people? Yes. I'm sorry. I'm trying my best to balance.
First of all, there is a metal and a non-metal. So we must begin with the metal, then the non-metal. But the calcium has 2+, and the chlorine has a negative.
Yeah. So what's the name of that compound?
Calcium chloride.
Calcium chloride. Who feels I have not been giving them an opportunity to go and show us the fourth one? Huh? Owegi, they have mentioned you. Huh? Owegi, go tell us about the fourth one.
So
use the microphone, please.
So this one was correct apart from the exchange of the valencies. No barrier mass valency of 2, then oxygen as of -2. So this will cancel out with this, then we find barium oxide.
All right. So when the valencies are the same, they just cancel out and you remain with a ratio of 1:1. All right? Yes, so I think that's it. So we can go to empirical and molecular formula. Now, we'll start by defining empirical formula and molecular formula. Now, what is the empirical formula? What is empirical formula? Somebody? Empirical formula? No one has an idea? Analytical chemistry students? Huh? Gideon, empirical formula. Yes, yes.
Empirical formula. This is the arrangement of different atoms that make a certain composition.
Arrangement of atoms that make a specific compound. Let me hear another one. Yes?
The composition of elements in a compound.
Where's the mic? Sorry, come up again.
It's the composition of elements in a compound.
Composition of elements in a compound.
Yeah, in
So
terms
like
of
when I
quantity.
say H2SO4, that's an empirical formula.
No, it's not an empirical formula, but we're going to have to find it through what we have.
Let me hear another perspective, another way of, yes.
It is a formula or an expression that shows the exact number of each type of atom present in a molecule.
Shows the exact number. What if you say it is the simplest formula of a molecule, showing the least number of atoms available for each element? Does that explain it better? So we can write it down, okay? So the empirical formula is the simplest formula of any molecule, showing the least number of atoms present for each element. Showing the least number of atoms present for each element. Now, from the formula of a compound, you can tell the combinations. You can tell all the elements that are in that compound. Like, for example, when I say methane is CH4, can you tell the elements that are in that compound? Yes. So a formula can give you the exact ratios to the simplest form for empirical formula. Now, what you told us was the definition for molecular formula. When you said that it is a formula that shows the exact number of atoms of different elements, that is molecular formula. So molecular formula shows you the exact number. Empirical formula shows you the simplest number. Have you gotten the difference now? So I can have several empirical units fitting in a molecular formula. And I think this will make sense when we do some practice, because some of these things you understand when you start practicing them, all right? So we'll start with calculations involving empirical and molecular formula. Now, I have provided a periodic table for you in section 1.82, or is it 1.86? 1.82. So put something on that page, the one that has the periodic table, 1.82. Behind. 1.78 there. Okay. So whatever we are calculating, we are referring to the figures in that periodic table in subtopic 1.82. All right? Now, in the periodic table, the higher number is the mass number. And then, what is the lower number? If I have 4He2, what does 4 represent? Sorry? Mass number. And then what does 2 represent? Atomic number. So, can you calculate using the data in that periodic table the mass of carbon dioxide? Refer to the periodic table and calculate the mass of carbon dioxide. So where will the amylase be? Oh, okay. Okay. Once it is set, just let me know. Okay, thank you. Ah, okay. Okay. This might take us. It might take us. But it will be here. So it is in the store that are okay, wow. Okay, what is the mass of carbon dioxide? Sorry? 44. Mass of water? 18. Now, is there anyone who is not aware of what is happening in terms of calculating these masses? Now, what is the mass of methane? 16. Ethene? Calcium sulfate? Oh, you have already done that. 121 point. Where is the 0.5 coming from? Which element has a 0.5? Because I can see that is 72. Why are you getting a 0.5 for calcium sulfate? Excuse me? Those who got a 0.5, how did you get that? Sorry? Oh, okay, okay. So what is the mass of calcium sulfate? 136. Damaris, are we together? Where exactly? Okay. Okay, so you see this large number. This is the mass number. So like if I want to calculate the mass of, the first one is carbon dioxide, okay? The mass of carbon here is 12, okay? The mass of oxygen here is 16, but the oxygens are 2. So it's plus 32 to give you 44. Now if I want to calculate for calcium sulfate, I'll have calcium with a mass of 40, sulfur with a mass of 32, it's here. Then oxygen we have 4 and each has 16. So that is already 72 plus 64 because 16 times 4 is 64. So we have 136. So it's very easy using the periodic table, and it's possible to calculate the masses of different compounds. Okay? Please attempt, we have done this, and we have done this. So this one is 136, and this one is 44. So using this data, here, calculate the mass numbers for the remaining ones. Very easy, you can use this as well. Okay, anyone else with a challenge in calculating masses? Anyone else? Now, as she finishes, we can look at the formula for calculating molecular formula. Now remember we said that empirical formula gives you the simplest form of a formula giving the least number of atoms of each element that is in that formula. Are we together? So, for you now to get the molecular formula, you must take the empirical formula and multiply by the number of empirical units derived. I repeat, I've written it there. For you to get the molecular formula, you must get the empirical formula and multiply it by the number of empirical units derived. Now, how will you know the number of empirical units? You take molecular mass over empirical mass. And that's why it's very key for us to be able to calculate masses correctly in reference to the periodic table. So most of the time, you will be given sufficient information to help you calculate. If they are requesting for molecular formula or they are requesting for empirical formula, you will be given sufficient information for that. But those are the formulas to apply. Now, it is important to be able to calculate ratios properly to get the right proportions for the atoms. Now, a mole is equal to the mass of 1 unit of an element or compound. In solutions, it can be used as part of unit of concentration. And I've said there to be looked at later. Yeah, we are going to look at the mole later, but I felt it's important for you to know that the mole is the mass of 1 unit of a compound. So what is the mass of 1 mole of carbon dioxide? Based on that definition of a mole, can you look at the definition of a mole and tell me the mass of 1 mole of carbon dioxide? 44. Because it is the mass of 1 unit of that molecule. All right? That's great. So I'm looking forward to us starting the examples because it is through the examples that we will understand. And I want us to look at example 1. So you can finish that later. Plus 48. It's 48. So that's 63. All right. Yes, it is. It is one of them. Okay, so now let's go to empirical and molecular formulas. Yes. Now, we are all in example 1. Example 1. Determine, are we all in example 1? Determine the empirical formula of a compound consisting of 55% potassium, 55.3% potassium, 14.6% phosphorus, and 30.1% oxygen. First of all, do these add up to 100? They add up to 100, so then we can proceed. So you first of all write them as a ratio next to each other. Okay? So we have potassium is to phosphorus is to oxygen. All right? Now, what is the percentage for potassium? Percentage for potassium, Mason? 55.3. Now, can you refer to your periodic table for the mass of potassium? Sorry? 39. So you divide this by 39. Remember, we are trying to get whole number ratios so that we can get the simplest formula. That's why the definition for empirical formula was very important. Now, what is the percentage for phosphorus? Sorry? 14.6. Great. Someone else check for me in the periodic table the mass of phosphorus. 31. Thank you. Give me the percentage for oxygen. 30.1. Mass of oxygen. Sorry? 16. Now, using your scientific calculators, I want you to give me the answers for all these divisions. We are still on our journey to getting a whole number ratio. Give it to me in 2 decimal places. 2 decimal places. Tuko pamoja kwa hizo ratios. Divided by 100. Point 3 divided by 39. Use this. Good. Okay, so I can see the first one is 1.42. 1.42. 1.42 is 2. 0.47 is 2. 1.88. We are still in the journey of getting whole number ratios. All right? Now, let's divide all of them by the smallest number. Which is the smallest number here? 0.47. So we divide all of them by 0.47. As I said, we are still in a journey of making this a whole number ratio. So can you give me your findings for 1.42 over 0.47? 3 points? So that is 3.00 is to 1.00, now that we have taken the direction of 2 decimal places. And then the third one is 4 points. Yeah, so then it means that the empirical formula for this compound is K3PO4. What's the name of this compound? Yes? Potassium phosphate. Potassium phosphate. Now, anytime you see 8, what does it mean? Anytime you see 8, it means? No. Hey, you have overthought. It has nothing to do with valency, please. Yeah, it means that there is not the number. It means that there is oxygen. Let us leave it at that. All right? Okay. Please attempt B. Now, are you seeing the ones in the formulas in brackets? Can you cancel that? Cancel that. Just cancel. The formulas in brackets. Cancel all of them, even this. Yeah. Okay, can you attempt B? Group 3, do you feel better? All right. Attempt B. Now, please come and consult if you're stranded. Come and consult if you're stranded. Come. Are you coming? Okay. Okay. Yes, come this side. Come this side.
I cannot round it off. So
To 2 decimal places.
So I just write 0.73.
No, you can round it off.
To
0.74. Yes. Aha, let me use this. Empirical formula is the simplest formula of a compound that gives us the least number of atoms that can be found in a compound, right? So the aim here now is to use these percentages and get whole number ratios, okay? Now when you look at the board, potassium, phosphorus, and oxygen, those are the elements that are in this compound. Now, potassium has a percentage of 55.3, and the mass of potassium is 39, all right? So you divide all percentages by the masses, all right? So you'll get these figures that are rounded off to the nearest 2 decimal places. So you'll get for the first one 1.42, 0.47, and 1.88. Then you divide all of them by the smallest number because we are converting them into whole number ratios to give us the simplest formula. You understand? So after I divided all of them with 0.47, I actually got whole number ratios. I got 3, 1, and 4. So then that means that in this particular compound, I have 3 ratios of potassium, 1 ratio of phosphorus, and 4 ratios of oxygen. And that gives us the compound potassium phosphate. All right? So for this one now, we can do B together and then you attempt C alone. 47.3 divided by, should be 64. 64. Okay. 64 is to 52.7 divided by 35.5. Assist me with that calculator. So we have 47. Are we together or there are people who are lost? If you are lost, please come to where I am. So this is 0.74 is to 52.7 divided by 35.5. That is 1.48. So divide all of them by the. You got it? That's nice. Ah, yeah. So this is 1, ratio 1, and this is 2. 74 times 2 is normally 148. So then we get CuCl2. And this is quite correct because the valency of copper is 2. Copper 2. So if you're looking at in terms of valency, you put there plus the chloride. You exchange like that to give you copper chloride.
You're making me feel I really missed.
So you see now, this has actually proved that our calculation. Yes, yeah. Now, what is the formula for B? Formula for B, guys. CuCl2. Can you attempt C and D quickly? Please, 1.5, actually not 1.5 hours. 1.5 minutes, 1.5 minutes. Now, group 4, please go for your health break. You're welcome. Four points?
37.
Okay. So it gave you automatic whole number ratios?
No, it's 0, 0, and then
then others, yeah. So those are automatically whole numbers. So this is the simplest you got.
To write here like percentage composition, relative, add the mass number, and the division, or it's okay
no,
just
no,
like
it's okay. It's okay. You have shown you're working. All right?
Yeah.
Are we doing C and D? I'm looking for people who are stranded. Huh? We move to the next topic.
Let's say those.
You have not done C and D and A. Group 4, please go for your health break. Now, group 3. How many pages have we done while you were away? Okay, I think can we go to problem 2? Skip the next page and go to problem 2. Now, what is the empirical formula for A? A. Clarice. What is the empirical formula for A? Darlene. What's the empirical formula for A? Or you're still calculating? Okay. Anyone? Empirical formula for A. Problem 2. You guys, which problem 2 are you doing? Yeah. Empirical formula cannot be a number. What is the empirical formula for C4O12? Now, considering that is a molecular formula, can you give me the simplest formula for that? Yes? CO3. Yes, because you're dividing all of them by 4. All right, the last one I want you to do individually is problem 3 in less than 2 minutes. Now, as you're doing problem 3, what is the empirical formula for C in problem 2? N2H4Cl. Good. I think now we're okay with that, yeah? So let's complete problem 3. I think 2 minutes are over. Can I have the empirical formula for problem 3? Someone give it to me. Yes? Is this what everyone has got? Perfect. So we can move on to something different. I hope you're not writing on my book. That's yours. Okay. Okay. Now let's look at simple chemical reactions and balancing of equations inserting state symbols. Remember, we are still on the foundation stages of stoichiometry, okay? Now we are looking at reactions of metals with acids to make salt and hydrogen gas. So we'd like magnesium ribbon with hydrochloric acid and test for hydrogen gas using a lighted splint. So we want to carry out that investigation of magnesium ribbon with dilute hydrochloric acid and testing for hydrogen gas using a lighted splint. Now, as they plan for that, I want you to give me rather, the three main acids that we use in the lab. So we are going to fill that table with the three main acids. Quickly. Yes? HCl is hydrochloric acid. Hydrochloric acid. Now, which salt does hydrochloric acid give us, Chebi? Chloride. All right. Another acid, yes, Marston? Sulfuric acid. Which salt do we expect from this? A sulfate. Now, it may look obvious, but it is not obvious. Are we together? Yes. The third one is nitric acid. That gives us a nitrate. All right. So as they're organizing for us to do that experiment, or rather to do the demonstration for reaction of magnesium and hydrochloric acid, we could write the symbol equation based on the word equation. Are we together? The symbol equation based on the word equation. Now, the formula for magnesium is Mg plus HCl to give you magnesium chloride. Now, how will you know the formula of magnesium chloride? Use the valencies for magnesium and hydrogen. We are here. Oh, chlorine, sorry. Thank you for that. Use the valencies for magnesium and chlorine. And then for hydrogen, write the diatomic molecule formula for that. Who are you interested in? Now insert state symbols. Insert state symbols. The state symbol for magnesium is S for solid. We are using dilute hydrochloric acid, so if it is dilute, it is in aqueous state. So HCl in aqueous state to give us, what formula did you find for magnesium chloride? And it's an aqueous salt plus hydrogen gas. Now we need to balance this equation. We need to balance this equation by ensuring that we have the same number of atoms on the left and on the right. So how many atoms of magnesium do we have on the reactant side? One. And on the product side? One. What about hydrogen and chlorine? 1, 1, right? And here it's 2, 2. So what do? We need to add a 2 here. All right? We need to add a 2 here to make sure that we have 2 hydrogens and 2 chlorine atoms, both on the reactant side and on the product side. So I want us to do it experimentally and test for hydrogen gas. Is it in order? Is the mic in order? Okay. You can move on to zinc and sulfuric acid. To give you zinc sulfate and hydrogen gas. So sulfate is the valency for sulfate is SO4. Okay? You treat it as a unit, the same way you treat the chloride as a unit. Okay? So if you're making a sulfate, you treat this as a unit, as a whole unit. Okay? Let's do this together. Zinc is a solid plus sulfuric acid. It's aqueous because we are working with it in dilute state. Okay? To give us that. Now how we find this is because the valency for zinc is 2. Sorry. Like that. Plus the sulfate, I said we deal with it as a unit. To give you, these two cancel now. To give you that. So this salt plus hydrogen gas. This salt is aqueous, forms a solution, right? So the equation is balanced because we have 1 zinc, 2 hydrogen, 2 hydrogen, 1 sulfate, 1 sulfate. So it's a balanced equation. Okay. All right. Where are they? So we are demonstrating or giving them as groups. I want them to do. You can just do in 3 groups. 3 groups. So we are going to provide you with magnesium ribbon and a dilute acid. And we'd like to see the production of hydrogen and the tests for hydrogen gas. How many are we able to give? 3. 3 groups. That's okay. Now we are going to supply you with 2 types of metals. The zinc ribbon, the magnesium ribbon and the zinc powder. And how many acids? The same acid. The same dilute acid. No, I just wanted to know which one. Hydrochloric acid. Then we'll supply you with splints for you to test for hydrogen gas using a lighted splint. Sorry? So how many groups are we doing? 4 groups? So let's divide ourselves into 4 groups, 4 stations. Let's have our lab coats on. Your lab coat is there. Ensure you have magnesium ribbon and zinc . Give them the splints. Sharp, eh? Oh. Wanawasha aje? But these are skewers. Why can't we use the normal? Now use the test tube holders to hold the boiling tube. And then I'd like you to also take initial temperature. Okay? I'd like you to also take initial temperature and final temperature. So just have the thermometer inside. Actually, take the temperature of the acid. It's okay, it's okay. Take the temperature of the acid, have somebody record somewhere. There's something I'm checking after this investigation. Yeah, the powder is zinc powder. please put the thermometer inside the liquid and read while it is there. You've been provided with one acid. You've just been provided with one acid. Isn't this just hydrochloric acid? Gideon, what's the question again? Yeah, for both. Yeah. Initial temperature of hydrochloric acid. I feel like the zinc is too little. Will it give us enough hydrogen for us to test?
Can add more.
Yeah, let's add some more zinc metal so that we get enough hydrogen. I'm not okay. I'm not okay. Now, as soon as you put the acid in the metal, somebody else has to be ready with a lighted splint. At the same time, somebody else should be checking the temperature. It's okay, it's okay.
If the splint lights up,
It's lighted. It's a lighted splint, but when it just has a glow, kama makaa, like it's on, but it doesn't have a flame. Yeah, that is a glowing splint. Yeah. Guys, can we hurry up a bit, please? Tuharakishe kidogo. Lighter. Okay. I'm waiting to hear the pop sounds of hydrogen gas. Have you measured the initial temperature of the acid?
Yes.
And it's recorded?
Yes.
We are just putting approximate volumes slowly. Yeah, you can shake a little. Now, you can seal the boiling tube with your hand to allow the hydrogen to accumulate. Now, the hydrogen is escaping. You need to test for it. Try and shake it so that it can continue. There is no sufficient. Let me have a splint. Have you been able to make the pop sound? Carbon 4 oxide. Can we try with a more concentrated acid? It's the acid that matters. Now, can you rinse the boiling tube? And then we'll supply you with let's not take so much time on that investigation. So this is a simple chemical reaction involving a metal and an acid. So case study, magnesium and zinc. Now, instead of sulfuric acid, we are still using hydrochloric acid, okay? But the equation I'll expect you to write is for sulfuric acid. Okay, please get the acid. It had the previous acid. It's still okay. Still dilute. It's here. Now please have a ready splint. Can we just light one Bunsen burner? It's okay, it's okay. Relax. There's a pay bill there. Lipa na M-Pesa. What has happened? What has happened? I need everyone to be able to, every group to be able to get the pop sound. We must test for hydrogen successfully. Please come. Kuja.
Are they going to endure the fight considering today is winning? Yeah, no, we didn't.
Oh, I've heard the pop sound. I've heard the pop sound. So now let's do the last one with magnesium so that we move on to something else.
You will measure the temperature, right? The measuring temperature?
Yeah, have you measured the final temperature for zinc? For zinc and hydrochloric acid. Now 2 minutes to conclude that investigation. Now what have you noticed about the temperatures? The temperatures are rising. What's the range? Initial temperature? 26 degrees. I'm just hearing pop sounds. Huh? Is that magnesium that is cooperating like that? Amazing. Magnesium is a darling, especially when it meets with hydrochloric acid. It's okay, I think. At least you've heard it, yeah? That's nice. So once it finishes reacting, take the final temperature and let me know. You rinse this.
46.
What does that tell you about the reaction? Is it exothermic or endothermic? Exothermic because it is releasing heat, energy to the environment. And that is why the temperature is rising. Now, the next thing I want us to do is to make a simple oxide by burning magnesium ribbon. Remember, we are looking at simple chemical reactions that we can even do with a 12-year-old. Are we together? Exciting chemical reactions. So I want us to make an oxide. So let's finish up with the acids and the thermometers. Sorry?
You know, when they sent me the list of what they don't have, I made the workbook. So the best document to refer to now is the workbook, not even the lesson plan.
Says exams. Use this. So we are burning magnesium ribbon. We are burning magnesium ribbon. We actually have a series of simple chemical reactions ahead of us. We are burning magnesium ribbon next, and then we are reacting a metal carbonate with an acid, and then we are reacting an acid with an alkali. In fact, if we can get sodium hydroxide granules to just dissolve them in dilute hydrochloric acid and check the temperature differences during neutralization, that would be sufficient. So at least make sure you have a point of fire. A point of fire. Now I'd like us to have the safety glasses at this point if we are burning magnesium. Safety glasses. Now remember, for every simple chemical reaction, we are writing observations. The method we are using, like the method we used to test for hydrogen gas, the observation, and then up there we are writing a balanced symbol equation for the same. Yeah, they can use the same acid for the carbonate. It's just that I don't know if we are able to deliver the carbon dioxide into the lime water. I don't think. So we'll use straws. And straws. Yeah, we'll just bubble. No, we'll just test using our exhaled air.
Oh,
yeah. For the pellets, just directly. All right, are we ready? I hope it's going to be exciting. So I want to see magnesium burning to form magnesium oxide. At least every group has a... there you go. There you go, there you go. Lift it, lift it. No, you should just hold it tightly. Okay, you can get another. Okay, interesting. Just place the oxide on the bench. Has everybody been able to see that? So that's formation of magnesium oxide by combustion of magnesium metal in air. What's the color of the magnesium oxide? White. It's a white powder.
Camera, lights. Cut.
It's taking so long. It has a layer of oxide, that's why it's taking long, but nice. Yay!
the smoke is white.
Okay, I think enough of that. I know it's fun, but let's move on to the next thing. Please return the safety glasses. Now, the next simple chemical reaction will be reaction of a metal carbonate with a dilute acid. We are just following the workbook, okay? And then we are going to write, careful, 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 careful. Can you return the safety glasses to the front? You don't need them. Actually, put them in here. Let me have all the safety glasses. Really? Where have all the safety glasses? Okay. Now, the next one, the next investigation involves a metal carbonate and a dilute acid. Now, we are going to test for the carbon dioxide differently. Carbon dioxide is one of the products formed when a metal carbonate reacts with a dilute acid. But we are going to test for it differently using lime water, and we are going to test for it with our exhaled air. Our own exhaled air. So we will bubble into lime water and see the changes that will occur. Now, what do we call the production of a gas within a solution in a reaction? Effervescence or fizzing or effervescence, fizzing or yes? Bubbling. Bubbling. So the method, we used a lighted splint, and then the observation, it goes off with a pop sound. Can we go back and finish writing the findings for reaction of acid and metal, considering magnesium and hydrochloric acid? Now, we are finished on the chemical symbol, or the chemical equation, balanced chemical equation that includes the state symbols. Now, what is the method we used to test for hydrogen gas? We introduced just a splint, a burning splint or a lighted splint. That's the method. Introduce a burning splint or a lighted splint. Burning splint or lighted splint. Now, what was the observation? It goes off with a pop sound. Goes off with a pop sound. Now, I'd like someone to go up to the board and write a balanced symbol equation for the reaction between zinc and dilute sulfuric acid. Balanced equation inserting state symbols. Yes, Bilha, you can do this. I'll give you an opportunity. The next one. So, anything that is dissolved in water is in which state? Aqueous state. Is it balanced? Yes. Now, one of the secrets, and I was telling one of us here, to balancing these equations that have complex ions is treating the complex ion as a unit. Okay? So you treat sulfate ion as a unit, not as sulfur and 4 oxygen atoms. Okay? So you treat it as a unit with a valency of -2, for example, for sulfate. Then we have nitrate, which has a valency of -1. All right? Yeah, so you treat it as a unit. So when you're balancing, you count. How many sulfate units do I have in this compound or in the whole chemical equation? All right? All right. So I think we are together so far. Haron, you wanted to do something on the board. Can you write for us a balanced symbol equation for reaction of calcium and nitric acid? To give us calcium nitrate and hydrogen. Nitrate and hydrogen. Balance it as well. All right. We are moving together systematically. Unyo kwa unyo. Now, what observation did we make after burning magnesium ribbon? We are now writing our findings as we organize on the carbonate and acid. What observation did we make when burning magnesium ribbon? Sorry? Burns with a brilliant, blinding white flame. How many looked at that flame directly? It's a blinding flame, yeah? Brilliant, blinding flame. Unfortunately, we were not able to collect the oxide or sufficient amount of oxide to dissolve in water and test using red litmus paper. But are you able to predict? Are you able to predict? What prediction do you make if we are to take the magnesium oxide, dissolve it in water and test using litmus paper? Blue or red litmus paper, what would happen? Red litmus paper turns blue. Why? One person. Oh, we are all singing. I want someone I've not heard their voice for some time. Oh, Cynthia, why are we saying that the red litmus paper will turn blue? Because it's a metal oxide, and metal oxides are basic. Non-metal oxides are acidic. All right? All right. So you can write the observation as the prediction we have just made, and then we conclude by saying that metal oxides are basic and non-metal oxides are acidic. So the observation is red litmus paper turns blue. And then the reason is that metal oxides are basic. Okay, other examples of word and chemical equations using calcium and iron. Now, I want us to look at the corrosion equation, basically how rust is formed, okay? If you look around, you will see rust somewhere. Who cannot spot a corroded metal? There's definitely something that has corroded somewhere, yeah? So I want us to write the equation for corrosion. Now, what are the conditions for corrosion of iron? There are two conditions. Yes, Imelda? Sorry? Presence of oxygen. Yes, because it's an oxidation reaction. Lenny? Moisture, or rather water. So what is the word equation for this then? Word equation for corrosion. We have iron plus oxygen plus water to give you hydrated iron 3 oxide. So this hydrated iron 3 oxide actually is the rust. This is the rust we talk about. So I want to invite a real chemist to come and write for us the equation for corrosion. Hey, Clarice. I can see you're ready. Take us away. Sorry? Yeah, but how many water molecules are there? Clarice, please tell us what you're doing. Mm-hmm. All right. Now, depending on the availability of water molecules, we can have as many water molecules within that structure as possible. Okay? Now, why is it that there is a dot here and we have the water here? What does that mean? Sorry? What does it mean? It is a hydrated salt. Are we together? Yeah, so that shows that within the structure of that salt, we have water molecules. So that is basically the corrosion reaction, formation of rust. Please copy that. Yes. You can put 4 here. You can put 4 here. And then put a 2 here. And then a 3 here. Then 2. Then water will have 2. Okay? So 4, 4, 6, 6. No. Treat it as water, as a unit. Okay?
Yeah.
The moment you start balancing one by one, if you can treat it as a unit, if it exists as a unit on the left, on the reactant side, and it also appears as a unit on the right, that's the product side, then you can balance it as a unit. So here I'm looking at water molecules. I have 2 here. I have 4 here. So you can do a 4 here then.
No, it's actually 2.
Yeah, there's a 2 here that has affected everything. So you can do 4.
No, but 2 times 2 is 4. This is 4 iron.
No, I'm talking about water. Everything else has balanced. You're just remaining with water. The water are 4, so you need to put 4 here. You see? So 4 iron, 4 iron. 6 oxygen, 6 oxygen. 4 water, 4 water. Now please note the balancing of the equation. Now since water appears as a unit on the left and as a unit on the right, balance it as a unit. Balance it as a water molecule. Okay? It makes it easier. Makes it easier for everybody. Don't forget to insert the state symbols. Now, this rust is a solid. This is a solid. Now, we'd like to react metals with carbonates. We'd like to react metals with carbonates. And we are going to use calcium. Sorry? It's balanced.
Really?
It's 6. Both sides.
This
Yes.
is 10.
Sorry?
This is 6 times. This is almost 10. Because it is 4 plus 6 is 10.
That's why I said if water exists as a unit on the left and as a unit on the right, balance it as a water molecule. How many water molecules do we have on the left?
4.
On the right?
4.
Yes. Balance it as a unit because it's not changing. Yeah. All right. Now, if the carbonate and the acid is not yet ready, we can go to simple masses of atoms and molecules. Simple masses of atoms and molecules. Now, AR refers to atomic mass and MR refers to molecular mass. AR refers to atomic mass and MR refers to molecular mass. Now, in terms of showing representation from the periodic table, we had looked at this when we were calculating masses. Let's go back to 1.82, where the periodic table is. Where the periodic table is. So it depends on the author of the periodic table. Some of the authors put the atomic number on top and the mass number below. But for this particular one, the mass number is above the atomic number. So when you're calculating masses, it is not obvious that you will find the mass number at the top. So you have to check and see the number that is lower. The number that is lower is the what number? Atomic number. All right? And then the number that is higher, the one that is above, is the atomic mass. So I think that's it for that one. We can go to the calculations now. 2.22. Sorry? Okay. Group 1, you can go for your health break. Is there any other group outside? Okay, group 1, please go for your health break. Please move quickly so that we proceed seamlessly. Now let's look at the worked example. Let's look at the worked example in 2.22. Now we are calculating the mass of magnesium oxide that can be made by completely burning 6.0 grams of magnesium in oxygen in the following reaction. Now, why do you think it's important to write the balanced chemical equation before even starting to do this calculation? What does the balanced chemical equation give us? Yes? Yeah, the balanced chemical equation gives us the number of moles. Are we together? Now, in this case, how many moles of magnesium are reacting? 2 moles. And how many moles of oxygen? 1. So what is the mole ratio for this reaction? 2:1. We are doing well. Now, we need to find out how many moles 6 grams is. Now, checking the periodic table, what is the mass of magnesium? 24. All right? This is a worked example. We are just going through it. Then we'll do our own problems. So the mass of 1 mole of magnesium is 24. What about 6 grams of magnesium? How many moles are those? You cross multiply. Okay? 6 over 24 to give you 0.25 if I'm not wrong. So that means that this 0.25, the 0.25 moles, okay, represents ratio 2, isn't it? Now, please tell me if you understand what I'm saying. Do you understand what I'm saying? Yeah, because the mole ratio is 2:1. And we have already found the number of moles of magnesium, then that means that that number of moles is equivalent to that ratio. So then, how many moles of oxygen have we used? How many ratios is oxygen? 1. So we are basically using half the number of moles of oxygen as the number of moles we used for magnesium. Because magnesium is 2 ratios and oxygen is 1 ratio. Are we together up to that point? Yes, so it's 0.25 divided by 2 to give you 0.125 moles of oxygen. All right? So what is the question asking us? The mass of magnesium oxide that can be made. So how many moles of magnesium oxide are we going to make? Considering magnesium oxide is also 2 ratios. 0.25. Now calculate for me. You don't even have to check, by the way. Because the answer is there, but I just want us to flow with it, yeah? Calculate for me the mass of magnesium oxide. 1 mole of magnesium oxide. 1 mole of magnesium oxide. 40. So if 1 mole is 40 grams, what about 0.25 moles? That's like asking, what about a quarter of that? Yeah? What's a quarter of 40? 10 grams. And that's it. So being able to connect the information you have to derive information you don't have and do your calculations. Now let's look at example 1. We'll do example 1 together. And example 1 and example 2. Then I'll let you do problem 1 and problem 2. Examples are mine, problems are yours. Okay? I mean the problems in the book. Yeah. Now look at example 1. Calculate the mass of aluminium in tons that can be produced from 51 tons of aluminium oxide. Calculate the mass of aluminium in tons that can be produced from 51 tons of aluminium oxide. Now, let's look at the equation first. So, 2 moles of aluminium oxide decomposes to give you 4 moles of aluminium metal plus 3 moles of oxygen. So what is the mole ratio? 2 is to 4 is to 3. Now, in this case, we are using 51 tons. 51 tons is how many grams? 51 million grams. Great. All right. Now, what is the number of moles that we can get from this? Number of moles we can get from this. First of all, I want you to get for me the mass of aluminium oxide, and I'll challenge Damaris to come and do this one. Come on the board and do for us the mass of aluminium oxide, Al2O3, using the periodic table. Please use the microphone. So where are we getting 54? No, don't look at the first two. Just get the mass of aluminium oxide. Aluminium oxide is Al2O3. Now, there are two, let me give you a hint. In aluminium oxide, we have 2 aluminium atoms and 3 oxygen atoms. So use that now to calculate the mass of, so it's 27 plus 27 plus 48. Sorry? Oh, okay, you had put 54? That's still okay? Okay. So 54 plus 48. 54 plus 48 is 102. So the mass of aluminium oxide is 102. The mass of 1 mole. Let's appreciate her. Thank you. Okay, thank you. So if 1 mole is 102 grams. What about 51 million grams? How many moles are those? Divide by 102. 500,000 because this goes here 5 times. So 100,000. In terms of moles, here we reacted 500,000 moles, and here 1 ratio. If 2 ratios is 500,000 moles, what about 4 ratios here? 1 million. So here we have 1 million moles, and here moles. How many moles of oxygen were produced? 3 ratios is 750,000. If you honestly don't know where we are getting these figures, raise your hand. Bramwell. Is when you've decided to scratch your head. Huh? Are we all together in terms of the number of moles? Yes. Yeah, let me just explain again. Maybe for purposes of even those who are watching. 2 ratios is what we have here, and we have seen that 2 ratios is 500,000. So what is 1 ratio? What's 1 ratio? 250,000. So here we have 2 ratios, 500,000. 4 ratios is coming to 1 million because it's 250,000 times 4. And then here it's coming to 750,000 because it's 3 ratios, 250,000 times 3. Are we together in terms of moles? Back to the question. Back to the question. The mass of aluminium in tons. You want to do the question? Okay. The mass of aluminium in tons that can be produced from 51 tons. Now, we have already seen that how many moles of aluminium are we producing? Moles of aluminium. 1 million moles. And what is 1 mole? 1 mole is how many grams? 27 grams. What about 1 million moles? 27? 27 million grams. Okay? To change them into kgs, we divide by 1,000. To change them into tons, we divide further by 1,000. So we remain with 27 tons. That's example 1. Can we fill out example 1? That one is good for that one is good for concentration. For concentration. When we get to concentration, we are going to do the concentration triangle, where we have moles over concentration times volume. Mm-hmm.
Electric molecular, which is this is moles. This will remain very because wakati unataka moles ni mass over area utakumbuka haraka.
Now Haron is teaching me chemistry here, and I want him to share something that he has just shared with me that is quite interesting. I've seen it before, but I've never used it. It's a formula that has a triangle in it. Who is aware about it? The one that has a triangle for calculating moles? You know about it? Okay. Haron, go and enlighten us about it. Rub the board. So he says that with this formula, it is easier to calculate number of moles. Haron, please use this. Rub the board and then use the microphone. Sorry? The microphone. They are bringing it.
Okay. You can use this triangle to calculate the number of moles and their relative atomic number. So we use this triangle. At the top, we place mass. This is mass. Then this you divide into 2. This is a relative molecular mass, relative molecular mass and moles. So when you are calculating number of moles, it is mass over RAM. So moles.
So basically you're saying that out of that you can extract 3 formulas.
Yes.
Give us the formulas.
So moles equals to mass, the top one, over relative
molecular or atomic?
Relative molecular or atomic.
Okay.
So when you are calculating, to calculate the relative molecular mass or RAM is equals to the top one, which is mass, divided by the number of moles.
Amazing.
Divided by the number of moles. So when we are calculating the number is equals multiply relative atomic mass times moles. Relative atomic mass times the number of moles. So I think this is the simplest way that you can do all three.
He has made it easier than the teacher made it, right? Yes. Can we appreciate him? Amazing. Thank you so much. Now, go to your problems in the workbook. Start with problem 1. Actually do problem 1 and problem 2. When you're stuck, come to me for consultation. Otherwise, I want to see the problems done so that we go to calculating percentage yield. Can you do this? This one. This one. So they're following this now. Okay. How long are you taking to solve their problems? It should not take more than 3 minutes, please. Problem 1. Have we been able to solve the problem? Now remember, if you get it wrong at A, I wish you well at B. You understand what I mean? If you get it wrong at A, the balancing, that means you get the mole ratio wrong. there are high chances that B might really harass you. There's a very interesting activity ahead of us. Please, let's finish the problems. How many moles of water? How many moles of water for A? 2 moles of water. And how many moles of sodium hydroxide? 2 as well. So the mole ratio is 1, 2, 2, 1. 1, 2, 2, 1. Amazing. Now, relative formula mass of sodium carbide. What's the relative formula mass of sodium carbide? That's 46 plus 24, giving you 70 grams. Is that where the question ends? Interesting. Let's go to problem 2 then. Oh, nice. Problem 2 is on empirical formula. Wait, that should take you less than 2 minutes, kindly. Wow. Hey. Simunai attack. Are we already done? What is the empirical formula for that compound? H3? Umetoa wako wapi PO2? Jameni, please. It's 54. You reduced 34? Ah. So umeiva kem. 6, 6, because 8. That 3:1:3. Because this goes 3 times into 3.66. Do we all have H3PO3? Are we sure? Please, I want to see. I don't know why I've started suspecting some of you. Ah, H3. You see? Problem 2. Okay. Now let's go to calculating percentage yield. Remember, we are still in stoichiometry. Are we together? The mole concept, we are still there. Now, Jessica, please read for us what yield is from the notes. Wait.
Yield is a term. Yield is a term used to describe the amount of product you get from a reaction.
What's the other name you can use for yield? Product. All right? So basically it means that some reactions will not give you 100% the product you expect. Are we together? Yeah, so there are analytical ways of calculating the percentage yield when a reaction does not give you the required or the expected product, okay, amount of product. Now, there are several reasons why you may never get 100% yield in a chemical process. Can we read the reasons why? These include nice. Are you fighting with the sentence? The reaction may be reversible, and in these reactions, a high yield is never possible as the products are continually turning back into the reactant. We know, or rather, if a reaction is reversible, it means as the product is being formed, the product is also being broken down back into the reactants. So it's very hard for you to collect 100% yield for that particular reaction, right? Now, another reason for not getting 100% yield is that some products may also be lost during separation and purification. Now, have you used a filter paper before? Are you able to collect 100% filtrate? No. A lot of the filtrate is left within the residue, and some of it is even absorbed by the filter paper. So those are instances that actually lead to the loss of some of the products. And then there may be side reactions occurring where a substance reacts with a gas in the air or an impurity in one of the reactants, and then products can also be lost during transfer. So what is the actual yield? What is the actual yield? Lenny, what is the actual yield? We have several yields. We have 3 of them. So what is the actual yield?
It is the recorded amount of product obtained.
Perfect. The recorded amount of product obtained. Clarice, what is the theoretical yield?
It is the amount of product that will be obtained under perfect practical and chemical conditions.
Yes. So if the conditions were to be ideal and perfect, we would get the theoretical yield, which ideally should be 100%. All right? With no loss. All right. And what is the percentage yield? Percentage yield?
The percentage yield compares the actual yield to the theoretical yield.
Okay. Percentage yield compares the actual yield to the theoretical yield. So you basically calculate percentage yield by taking actual over theoretical times 100. Can we write that formula quickly? Percentage yield, it's actually on the next page. No need to write, please. No need to write. Group 2, please take your health break. Take your health break, group 2. Now, the higher the percentage yield, the more successful a chemical process is. So if I perform an experiment and I get my percentage yield to be 40%, and another scientist performs theirs and they get 60%, there are high chances that the one with a higher percentage yield has had a more successful investigation. Are we together? Now, the aim is to ensure that the reactions that we perform in the lab or wherever give us 100, not 100%, but give us the highest percentage yield, okay? Now, let us look at worked example 1. Let's look at worked example 1. Then I do example 2 with you, and then you do problem 1. Okay? Now, worked example 1, let's all go there. Please read for us the question. Is this microphone working? It's working. Read for us worked example 1.
Copper 2 sulfate may be prepared by the reaction of dilute sulfuric acid on copper 2 oxide. A student prepared 1.6 gram of dry copper 2 sulfate crystals. Calculate the percentage yield if the theoretical yield is 2.0 grams.
Wow, this has really been simplified for you. So, using the formula above, all right? Ideally, the theoretical yield is supposed to be 2.0 grams. Are we together? If we are together, please do this. Let me see. Thank you. So, this student only got 1.6 instead of getting 2.0. So, it's very simple mathematics. The one that the student collected, the actual yield over theoretical yield times 100. That gives you 80%. Now, example 1. A reaction with a calculated yield of 9.23 grams produced 7.89. We can calculate this quickly. 7.89 over 9.23 times 100. 7.89, that is the actual yield, over 9.23 times 100%, 85.48%, good. And then example 2, 5.96 grams of ammonia react completely according to the following reaction. Please draw an arrow where there is a space. That is a chemical equation. So draw an arrow connecting the reactants and the products. Now, what is the theoretical yield of urea for this reaction? Can you try and crack that? Uh-huh. Please attempt example 2 and problem 1. considered as time-lapse. But you see, it's not indicated that I'm supposed to pay for them. It's okay. So we don't consider them. No, when you're giving any other additional information, you will see that there were time lapses when a tempter was walking to the venue. Show us. Now you see that, I know where you're getting confused. You see that 17.031 grams per mole is trying to tell you that 1 mole of ammonia is the mass of 17.031. Are we together? That's the mass they want you to use. So that now you can be able to calculate the number of moles by dividing 5.96 over 17.031. I don't know if we are together. I feel like some people are lost and they are not saying it. How do I find out if you are lost? Yes, Martin? Now, you see this information they have given to you in brackets, okay? They are simply they are hinting to you the mass of 1 mole of ammonia. That's why they are telling you ammonia is 17.031 grams per mole. Okay, so then if 17.031 grams is 1 mole, what about 5.96? So now when you get the number of moles, you will equate them to this ratio. And then you will get half this number of moles for this one. What is the question asking? Theoretical yield for this reaction. Theoretical means that we collect 100%. So that means that they are telling you that 1 mole of this is 60.056. Let's do example 2 on the board. I can see what it is doing to you. Yes? You've been able to crack it? Okay, go ahead. You've also been able to crack it. Hey, Bill, we are waiting. Save us. Let me rub for you. This one needs a black pen so that we see properly. Who else has been able to crack this? You've been able? Great. Okay. Guide us step by step.
So you all have the chemical equation, so I'm not going to repeat it.
Repeat it, please. Thank you. Can I write it for you?
It's okay.
Let me write it for you. So it's 2 moles of urea. Okay, proceed. I've saved you some energy there.
So, the question is asking 5.96 grams of ammonia react completely according to the following reaction. What is the theoretical yield of urea for this reaction? So, because the question is focusing on ammonia and urea, we are going to do the same. So, according to the equation, 2 moles of ammonia react to produce 1 mole of urea, so the mole ratio is 2:1 of ammonia to urea. And in the question, we have been given the mass of ammonia that reacted and the relative molecular mass of ammonia. And according to what Haron said, moles is equals to the mass over R mm. So we have 5.96. We divide by 17.031. You get 0.35 moles after rounding off. So
in short, we reacted 0.35 moles of ammonia, right?
Yes.
Good. Are we together up to there? Okay.
So we reacted 0.35 moles of ammonia, and according to the equation, this is the mole ratio. So we can use the mole ratio to get the number of moles of urea that were produced. So if 2:1, what about 0.35? This side is for ammonia, this one urea. So you cross multiply, you have 0.35. You divide by 2, you get 0.175 moles. So in that reaction, 0.175 moles of urea were produced. So, and in the question, you have been given the relative molecular mass of urea,
which is
60.056. And the mass is given by the relative molecular mass multiplied by moles. So we have 0.175. You multiply by 60.056. You get 10.51 grams. Any question?
Any question? Is that the same method you used? So probably the most confusing part was when they started putting those things in brackets, figures that you did not know how to apply, isn't it? Yes. But all the information for you to answer that question had been sufficiently provided. Now, can we quickly do problem 1? Let's quickly do problem 1. For problem 1, we are looking for percentage yield. Again, draw an arrow connecting the reactants and the products to complete that as a chemical equation. So you can take a photo of this as well. Now you're masters of percentage yield. Are we done? I'll be able to take some of these nice ones. Yeah, okay. Just show you now. Also them in class. Now, for the interest of time, we have a lot of time. How many hours do we have? 66 minus 20. Imagine just 40-something hours remaining. Are there many? Okay. Yeah, next is the quantitative measurements. Okay, let's do problem 1 together. Okay. 85.4 grams of chlorine reacts completely according to the following equation. If 104 grams of phosphorus trichloride is produced, what is the percentage yield? So the actual yield is, for phosphorus trichloride, 104 grams. Are we together? So ideally, what are we supposed to produce? Or theoretically, yes. Theoretically, we are supposed to produce the number of moles for chlorine in this case to the number of moles of phosphorus trichloride is 6:4. Do we all have that? Right? Then we have 85.4 grams divided by 70.91 grams to show us the number of moles of chlorine that we have reacted. If you are lost in any step, please say. What do you get when you calculate using your scientific calculator? 1.204. Perfect. So ratio 6 is 1.204. What about ratio 4? Are we together? Cross multiply to get the number of moles of phosphorus trichloride. So we have 1.204 times 4/6. Speak up. 0.80. Okay, I'll need to calculate this for myself now. Why am I not trusting your... Let me do it. Why are we getting different answers and we are using the same calculators? 0.803. 0.803. So 0.803 what? Moles. All right? Now, theoretically, we are supposed to collect this if we convert it into grams, okay? Now, 1 mole of phosphorus trichloride is 137.3 grams. What about 0.803 moles? You know why I prefer cross-multiplying? Because even a 12-year-old with this kind of explanation can be able to handle these problems, okay? The moment you start introducing relative molecular mass, relative atomic mass, if you are explaining to someone with probably a poor foundation in science, they may not understand. You lose them by using those terms. Are we together? And we said that the aim of this class is to show the doable side of science. Are we together? Not the hard side. All right? Yeah, so let's calculate this. 0.803 times 137.3. That gives you? Sorry? 110.25. This is the theoretical yield. And what is the actual yield? 104 grams. So 104 over 110.25 times 100. What percentage did we get now? Sorry? 94? Okay, 94.35%. We said on Friday we are doing an exam in the morning. Okay. I know you are ready. Yeah, so now we do away with theory. Let's do some bit of practical, okay? Now, the following activity that we are going to do, we are going to make careful quantitative measurements. You know those measurements that you don't want it to exceed even by 1 gram? Because we've been doing a lot of approximations in the previous investigations. So this particular one, we are going to make careful quantitative measurements. Are you ready? So let's put on our lab coats for those who don't have, like myself. I don't know which one is mine. Okay. And we are using a very friendly salt called copper sulfate. Please advise the number of groups we are working in. Miss Marlin? 4 groups. 5 grams. Okay, careful, please. Musiumie. Or they're not measuring the mass themselves. Okay. Oh, okay. So where is the 5 grams? Oh, okay. Now, the investigation has clearly been described in the workbook. Is this my book? Now, the aim of this investigation is to determine the formula of hydrated copper 2 sulfate experimentally by taking very accurate measurements. All right? Now, you can see the kind of setup that we are supposed to use. All right? Now, we need to follow these instructions and the procedure to the letter so that we get almost similar outcome. So are we in groups of 4? Thank you. Now, at least we should have a weighing scale on each side, the left and the right. Now, step number 1. Now, for every measurement, just write it next to the instruction. Like you see this one, measure the mass of evaporating dish. Write the mass of the evaporating dish next to that instruction. All right? So, step number 1, measure the mass of evaporating dish using the weighing scale provided. Okay, please write, yes? Okay, we should be done with the first. We should be done with the first instruction, that is measuring the mass of the evaporating dish. Okay? Now, we have provided you with 5 grams accurately measured copper 2 sulfate. All right? So that is the known mass of a hydrated salt. So please record there, next to that instruction, that the hydrated salt is 5 grams. Now, I believe you have the glass rods to stir, because the third instruction indicates that as you're heating over the Bunsen burner, you should gently stir until the blue salt turns completely white, indicating that all the water has been lost. So we need the glass rods, just four because we have 4 groups. So it is just water of crystallization that is evaporating. So don't feel like you need the gas masks or anything or safety goggles. You're safe. There is no hazard. So have you been given the glass rod? Okay. Be patient until the salt turns completely white. The independent witness. Imagine. I don't know, Rose. My mind is tired. No one wants to cooperate. Hata ndio nimerushwa hapa. Because the students are refusing to listen to me. Trish is frustrated. The independent guys, the witness guys, I don't know where they've gone. Miruka sijui ako wapi. Kelvin sijui ako wapi. Tell me what to do. I don't know what to do. And by the way, you see those granules that are on the stirring, you have to make sure they go back in there because they'll affect your measurements. Remember, we are saying that we are taking precise and accurate measurements to help us see the number of water molecules of crystallization, yes?
Keep something. Yes,
okay. Please let's have lab coats on if we are doing a practical. Now, if some salt remains on your stirring rod, you have to make sure it goes back to the evaporating dish, because it's going to affect your measurements, final measurements. Remember, we are taking precise quantitative measurements. So we want accuracy. For this one, we are not estimating anything or approximating. Okay. But I feel like the wind is also sabotaging your process. Maybe. By the way, you can lift the Bunsen burner to be closer to the evaporating dish for quicker results. Hey, science students, you're very creative, eh? Please try and shield your Bunsen burner. I can see some people getting very creative here, just to ensure that there is no heat that escapes. Let me go and help others. Yes? Ah, okay, okay. All right. You just set it up and then you set it up and then you let me know. Okay. All right. Yeah. Wow, due to public demand, we'll use another salt. Okay. So actually, from this activity, I'm going to skip the mole and Avogadro constant, so that I attack you with it at midnight. It will keep you alert. And then now after that we do an activity. It will work, trust me. Yes, I'm sure. I'm very sure.
What can you do? Just to completely turn it.
Now, is there any group that has managed to get the white salt? Totally? Uh-huh. Miss, can it be whiter than this? It can be whiter than that. Okay, keep heating. As long as it's blue, it means it still has some water. Let me seek a second opinion. Miss, second opinion here, please. Hi. How are you? Are you okay? Yeah. Okay. Ah, nice.
Yeah, yes.
That's okay? Okay. Now we move on to the next step. You just follow the steps. I can smell some gas.
But I think these ones are leaking because they're not so tight.
It has actually stopped smelling. There was. Let me see. Maybe for 1 more minute. Oh, yours is taking long. Lime water and the straws.
You want to smell?
Yeah, we can test for carbon dioxide. When we finish this, just hit a little bit more. We see whether we'll be able to achieve. Yeah. Now, if you cannot see any more change despite continuous heating, move to the next step. If you can't see any more change despite continuous heating, move to the next step. And the next step is you measure the mass of the white anhydrous salt remaining. Now, how will you know the white anhydrous salt remaining? Excuse me, attention. The next step, let me have your attention. The next step says that we should measure the mass of the white anhydrous salt remaining. How do you hope to achieve this? All right. Sarah.
You measure the petri dish and then you subtract from the... And what's petri dish?
Evaporating dish. But I remember we measured the evaporating dish. So just measure as it is, then subtract the mass of the empty evaporating dish. All right? Please, let's do that in a few. Yes. Are you okay? Oh. Okay. Is there another group outside? That's 0.5. That's amylase 0.5. Okay. Okay. Now, can we please finish that investigation now? Now, there is a table in the next page. There is a table in the next page to help guide you. To help guide you in how you're going to get the ratio that will give you the salt and the water of crystallization. Please refer to that table. So mass of the anhydrous salt, and then how do you get mass of water? Mass of the hydrated salt minus mass of anhydrous salt. So it's 5 grams minus, what's the mass of the anhydrous salt? 48? 48? 48? Mass of the evaporating dish alone is 66. And then mass of evaporating dish and contents? You didn't measure yours. And I suspected you people will do that. Now you guys, can you measure can you put the salt like on a filter paper and measure your own? Or just use a filter paper. So some people used the mass of somebody else's evaporating dish. Are we really serious? You are supposed to measure your own. Now let me see the figures you've got. Evaporating dish alone, and then?
Then this is the combined hydrated, then this is the unhydrated.
Okay, so then, using this, can you feel now the mass of anhydrous salt and the mass of water? And then now divide those masses with the molecular masses. Can we fill the table in the next page? Step 2, step 2. I want to use sample results. Sample results for anhydrous salt. So I'm using sample results from this group, and from their sample results, the mass of their anhydrous salt is 3.28. All right? So let's fill this here. 3.28. And so that means the mass of water is 5.0 minus 3.28, which is 1.72. Okay, based on this data for this group, mass for the anhydrous salt is 3.28 grams, and the mass of water is 1.72. Now, what is the molecular mass or formula mass of copper sulfate? Copper sulfate. Copper is 64 plus 32 plus 64. 160. Group, is there another group outside? Okay, group 3 you can go for. So we are doing 3.28 over 160, and then we are also doing 1.72 over 18. Please calculate that. This is 0.0205. And then for water, 1.72 divided by 18. 0.096. So the smaller one is this one, yeah? So we divide to get 1, and then divide by 0.0205. What will you get? That's, oh, so when you round off, it's 5 actually, and it's normally 5. Yeah, it's normally 5. So it's 0.468. So we round it off to the nearest whole number. So it's 1:5. So the ratio in form of a salt is copper sulfate with 5. Hey, Well, you guys, this group has got accurate and excellent results. The ratio in form of salt is to water is 1:5, which is the way it is normally. 1:5. 5 molecules of water of crystallization. So I'm waiting for the rest of you to finish. I'm waiting for the rest of you to finish. 2. What is your salt? 1 to 5. Oh, wow. Let's appreciate these two groups. Let's appreciate them. As you also finish working on yours. Maybe I can award you 2 more minutes to finish what you're doing. I really want to know your ratios for those two groups.
See you.
See you.
Now that we have got them, I'm not good with this biology practical. So just So I just want us to investigate enzyme action. Okay?
Sorry?
On a substrate. Enzyme action on a substrate. So we have amylase, then we have starch. So when amylase breaks down starch, what do we expect? Expect to have glucose.
Yes.
So then we will start by testing the starch for the starch test.
Yes,
Then
using iodine.
Yes, then after that, tunaweza changanya maziwa. But I wanted us to put in a whisking tube starch pamoja na amylase in a whisking tube. After hydrolyzation, after digestion, you know the simple sugars will diffuse into the Benedict. So to make a Benedict solution in the beaker to mass level. Whisking tube yenye iko na nini? Na iodine, so na amylase na starch. Then tunachukua Benedict na if the diffusion has taken place, we heat it.
Yes.
Tunaweka kwa maji moto. Tunajua kama imehydrolyze, so you know the simple sugars will diffuse.
Yes.
Then it will turn positive. How can we do it? But that is only possible if you are so sure that amylase is active.
Yes. Can we do a pre-test and then do it later?
Yes.
Okay.
Can I do that?
Okay, these two groups are the ones we are waiting for, please. We need to know the ratio you have found after all is said and done. Please, I'm giving you just 1 minute. You have gotten a ratio of? Unafanya nini? We are here. Or you are doing it? What ratio did you get? What ratio did you get? 1:8. So, copper sulfate to water, 1:8. How come? I even thought it would come to a less. Okay. I don't know what you did wrong. What did you get as a group? The ratio?
Divide by 18. Lazima maji 18. Hii ni 1.8 divided by
2 into 1 ya maji na 16 ya oxygen. So ratio is 1.8 divided by 18.
Then you divide
alafu 3.19 over 160 share. Then we'll divide.
Okay, I think we'll suspend that because we have taken so much time. It's unfortunate that you have not been able to give us your ratio as a group. Huh? So please finalize that in your own time. Now, this group has got a ratio that is way above what everyone else has gotten. But I highly suspect the problem started when you did not use your own. Okay, they have gotten 5. Let's appreciate them, they have gotten 5. Oh, you have also gotten? Are you sure? Okay, then we are all on the same page. Okay, now, can we put on hold topic 1.33? Now, before we move on to the next thing, I was saying that I have suspended topic 1.33 until midnight today. Tonight. Who will remind me? Thank you. Sorry? No, yeah, we give them. We give them. Yes. So now we want to test for carbon dioxide using exhaled air. Now there's a lot of murmuring going on, and it's making it difficult for me to communicate to you. Okay? So you're being provided with lime water and a straw. How many are doing per group? 2. All blowing in the same or different? Okay. So select 2 of you to do the activity. It's not there, so we'll note it down in the exercise book. Now, I've decided to test for this so that when we are doing the carbonate and acid reaction, we'll have already tested for the gas prior. So don't wonder why I'm jumping again to testing for gases. Anyway. Now, please exhale into the lime water through the straw. Until you see an obvious change. That is the test for carbon dioxide using lime water. Exhale into the, yes. No, just do it slowly.
I'm doing it slowly.
I don't think you're doing. Just do slowly. Keep doing it until it turns milky. Now, note in your, kuna straw ingine? Miss, tunaweza pata straw?
Straw?
1 straw. Thank you. Please don't, you know students are funny. Or you don't know you are funny? You can decide to do the other way around. Instead of exhaling, you decide to ingest. Lift up and show them yours. That's nice. So that's a positive test for carbon dioxide. That's a positive test for carbon dioxide. Now, do you have the energy to do the mole and Avogadro constant now? No? Okay, we can go to enzymes. I know where I'll catch you. Let me write it on top of the board so that and nobody should rub. It's for my own reminder. Now, let's go to enzymes. So flip your pages until you get to enzymes. Now, if you'd like, I don't know, are we doing the experiment now or so we don't remove our lab coats? So we do theory first? Okay. Okay, define enzymes. Define enzymes. Tell us what, yes, Clarice? Catalysts that are protein in nature, I agree. There's just something missing. Catalysts that are protein in nature, yes? That speed up the rate of reaction but does not change, I agree. Combined with her answer, there's still something missing. Yes, Bramwell?
Does not take part in the
Hey. Thank you. There's something that is still missing. Huh? But that's what he just said.
He said in the chemical.
And they are naturally found in the body.
Naturally found in the body. Master. Hey, thank you guys, but there's something missing. Are we together? There's something missing. Now, enzymes are biological catalysts that speed up the rate of metabolic reactions and are protein in nature. Biological catalysts that speed up the rate or that increase the rate of chemical reactions, and are protein in nature. All right. Now, I don't know, but have you ever noticed that when you put a piece of bread in your mouth, it doesn't taste sweet instantly? Kweli rongo. But when it stays in the mouth a bit, the taste changes and it becomes sweeter. If you have ever honestly observed that, please raise your hand.
Biscuit.
For biscuit? Yeah. Now, what do you think happens? What do you think happens? I did this experiment with my students and they could not believe it. I bought for them bread. And I told them, put the bread in your mouth. We just did it in class. We didn't even go to the lab. I try here. Now, what happens is, by the time the bread is getting into your mouth, it is starch. Okay? Now, starch does not have a sweet taste. Okay? But because of enzymes in the mouth called salivary amylase, it changes the starch into a disaccharide. So we move from polysaccharide to disaccharide. So we reduce the complexity of that molecule. And we end up with maltose. Are we together? Yes. Which has a sweet taste. So that is why when you put bread in your mouth and you stay with it for a few seconds or minutes, minutes are many? Seconds. A few seconds, you realize the taste is changing because there is an enzyme in your saliva. Are we together? So that basically now gives you an idea. Now, a catalyst alters the rate of a chemical reaction without being changed itself. We have been told that repeatedly by the three gentlemen here, that a catalyst alters the rate of a chemical reaction without being changed itself. Now, chemical reactions, we are writing as I talk, isn't it? Chemical reactions that take place in the body are called metabolic reactions. Now, we can survive without enzymes. Are we together? Yeah, it's not a must for us to have enzymes, but that would mean the food you eat on Monday will leave your system. Something is burning outside. Okay. Okay. So that would mean that the food you eat on Monday would leave your system after 3 weeks. Are we together? Yeah. Yes. So the work of catalysts is to speed up the rate of some of these metabolic reactions like digestion. Now, starch is digested by amylase, and protein is digested to amino acids by protease. 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 Now, do you believe that teachers can forget? Yes. So if I say I forgot, I'm not being judged, eh? No one is judging me. I forgot something. When we were looking at chemicals of life, yeah, we did not say that when you break down proteins to the simplest molecules, you end up with amino acids. We just talked about the chemical elements that make up the three main chemicals of life, okay? So is it okay if I give you that table now, quickly? Thank you. So I'll give you the biological molecule, okay? And the simplest molecule it can be broken down to. For example, let me just do it in form of a table. Biological. Okay, so give me the three main ones. Give me the three main chemicals of life. We already looked at them earlier. Now, when we break down carbohydrates, what do we get? Simple sugars, okay? Simple sugars. And an example of a simple sugar is glucose. Now, what do we get when we break down proteins? Sorry? Amino acids. So amino acids are the simplest particles of proteins. And then what are the simplest particles of lipids? Yes? You're just saying, can I have somebody giving me? Alvin, how are you? One of the simplest particles of lipids? Fats? Not fats and oils. Glycerol is one, and then the other one is fatty acids. Okay? Now that we are looking at enzymes, if we need to break down these complex particles like proteins into simple particles like amino acids, we would need a protease. Are we together? So the enzyme that we would use here is protease. All right? Now, which one would we use for lipids? Lipase. Now, it doesn't mean that there is just that one enzyme that can break down lipids. It's a group of enzymes that are able to break down lipids into fatty acids and glycerol, okay? And then for carbohydrates, we have carbohydrases. Carbohydrases. For example, what? Salivary amylase. Finish writing that. Okay, now that I've named a few enzymes there, it's important for me to mention that enzymes are named according to the reaction that they catalyze, okay? So if they catalyze the breakdown of carbohydrates, they are called carbohydrases. If they break down proteins, they are called proteases. And if they break down fats or lipids, they are called what? Lipases. Good. And the one that breaks down sucrose? Sucrase. Yeah, hii ni vitu rahisi tu. Now let's look at part of an enzyme. I don't know if I've given you that diagram. Now, in your own words, please write why enzymes are important to living organisms. Why are enzymes important to living organisms? Is there anyone I've misplaced here? You're back? Okay. Let me go back to where I belong. Now, before I even go to enzyme action, I would want us to draw an enzyme and label it. Now, the main thing that I want us to label is the active site. Are we together? Now, if you feel that you can label the enzyme that is already there, it's still okay. I am not under pressure to make you draw. So you can actually label the active site in the enzyme that is already provided for in your workbook. Now, the active site is the dent. Is the dent, or what else can you use to describe a dent? Depression. Yeah, let's use dent. Even my textbook here says dent. So we can stick with that. Okay? Now the dent, where the substrate fits, is called the active site. Now the active site has a complementary shape to the substrate. So the substrate is the substance being acted upon by the enzyme. All right? So the active site of the enzyme and the substrate have a complementary shape, just like the lock and key. So the lock of room 130 can only be opened by its specific key. And even if I have 5 similar keys that are not for that lock, I cannot use it. Have you ever tried opening? I'm sure you've done these things. Huh? Have you ever tried opening a lock with a key that does not belong to it, but that key looks like it's for that lock? That is how specific enzymes are. So that is what I've just explained is the lock and key hypothesis. Now, the moment the active site combines with the substrate, we call it enzyme substrate complex. Can you say that?
Enzyme substrate complex.
Thank you. The enzyme substrate complex is a combination of the enzyme, or rather it is when the enzyme binds to the substrate. Let me confirm if we had a model of lock and key. Did we have a model of lock and key? Can we proceed? Or what are you doing? You're resting. From what? From 21 hours. Now, I want us to look at factors that affect enzyme action. And before I go to the first factor, I have a question for you. When is enzyme action at its optimum? When is enzyme action at its optimum? When is enzyme action at its optimum? Sorry? When the conditions are favorable for its action. Good. What else? Go and wash your face. Go and wash your face. When is enzyme action at its optimum? What does optimum mean? When it is performing at its peak or to its maximum, all right? Yeah, so when does an enzyme perform at its optimum? Now, as you're giving me the answer, you could consider using enzyme substrate complex as one of your jargons in your answer. I'm saying, as you're, who is it, sorry? Okay. I'm asking one last time, when is an enzyme considered to be at its optimum performance? And I'm saying that in your answer, use the words enzyme substrate complex. There's a group outside, right? Okay. So group 4, you can go for your health break. What's the time? Bye. What's the exact time? What's the time? Now, an enzyme, or rather enzyme action, is at its optimum when there is the highest number of enzyme substrate complexes being made per unit time. I repeat, you're scientists, right? Yeah, so enzyme action is said to be at its optimum when we have the most enzyme substrate complexes being formed per unit time. Now remember, you can't measure rate without including time. It's like saying that a car is moving at a speed of 20 kilometers. Is that a good way to describe speed? It has to be per unit time. So these factors that we have listed here could either increase the number of enzyme substrate complexes being formed or reduce the number of enzyme substrate complexes being formed. So let's look at temperature, and I want us to describe graphically. Excuse. When it's high, when it's low. I'd like to hear what you've gathered about temperature in particles, and especially biological particles like proteins. Yes?
When temperature affects these particles, it will increase the activation energy. And when the activation energy increases, there will be more coalition of the particles. But for the enzymes, it will raise the enzyme activity to an optimum point. And if the temperature goes higher than that, it will force the enzyme activity to drop.
Are you able to repeat what you have said?
Yes.
Let me have the microphone. I want everyone to hear what he's saying. It's here. Thank you. You know, science students, you are nice, all of you.
So for the temperature, when temperature increases to a substance, it will increase the activation energy. And after the activation energy increases, there will be more particles bombarding each other. And for the case of the enzymes, the temperature will increase the enzyme activity to a certain optimum point where a temperature to which that enzyme won't work again. So after that point, the enzyme activity will drop.
I have a question for you.
Yes.
What is activation energy?
Activation energy, this is the energy that happens between molecules.
No, let him finish. You, Haron, leave him alone. Continue with your answer.
Activation energy, this is the energy that is found between molecules. Molecules have bonds. So for these bonds to be broken, there will be needed some energy to break the bonds and make them active to other reactions. So you can call it activation energy.
You know why his answer is stressing me? It's because he's saying a wrong statement and then qualifying it with a right statement. Now, let me talk about activation energy because it is still in science. Activation energy is the minimum amount of energy required for a reaction to take place. Is that what you meant? But then, how come you're saying that increasing temperature will increase activation energy, which will make the reaction to happen faster? Wacha kukaana, si hivyo ndivyo umesema?
Until its optimum point, then you drop.
Activation energy. Ideally, that's what you're saying, that when activation energy is increased, the rate of enzyme action, or rather the enzyme action, increases as well. Because as far as I'm concerned, if I increase activation energy, I'm making it difficult for that reaction to take place. Okay? So I should actually look for ways, don't close your eyes. I should actually look for ways to do what? I should look for ways to lower activation energy. So you're saying the wrong thing and then you say the right thing. Then you say the wrong thing, then you say another right thing. Huh? Okay, anyway. Now, for us to increase enzyme action, we should make sure that we have as many enzyme substrate complexes as possible. Do we agree up to there? So how can we make them as many enzyme substrate complexes as possible? Number one, by increasing the temperature, okay? By increasing the temperature. So this makes both the enzyme and the substrate to gain kinetic energy. This makes both the substrate and the enzyme to gain kinetic energy. So they move more and they move faster. So they collide with each other more. Those are the words, you know, that best explain what happens. They move more vigorously and faster. So they collide. They collide? What are we saying? They collide more, okay? And so the chances of an enzyme substrate complex to be formed increases by this process, okay? So that way, have we achieved better enzyme action? Have we? Yes. Now, when it comes to temperature, we should be very careful because beyond the optimum temperature, what happens to the enzyme? It becomes denatured, or basically denatured means that the nature of that enzyme is destroyed. All right? So if the nature of that enzyme is destroyed, then the active site can no longer fit with the substrate, and so we are unable to make enzyme substrate complexes. All right? So that's what we are discussing, temperature. Now, on the other hand, think about low temperatures. Now, we need the enzyme to move, we need the substrate to move so that they collide with each other. And as they collide, chances are we will form an enzyme substrate complex. All right? So what happens then when the temperature is low? Marston. Marston. What happens when the temperature is low? Take this to me.
When temperature is low, the kinetic energy, when temperature is low, it makes the subject to the substrate to reduce the kinetic energy, thereby it reduces the chances of a new substrate being formed.
Yeah, so when the temperature is low, it means that the particles lose kinetic energy or lose energy for movement. And so they move slower and they collide less, so reducing the chances of an enzyme substrate complex. Sawa sawa? Can we write that down? Now, I had a question on temperature as a factor that affects enzyme action. Let me see. So guys, the best temperature at which the enzyme works, you see at the peak of its performance, that is the optimum temperature. So like for humans, The optimum temperature is 36.8 degrees to 37.2 degrees because that's the normal human temperature. Now, Miss, are we able to do effect of catalase on hydrogen peroxide? Yes. All right. Yeah, so we have an activity here. We are going to supply you with hydrogen peroxide, okay? Then we will give you catalase. Now, catalase is found in almost every kind of living cell. So I don't know if we are using potato or liver. We are using liver, okay? Can we write the procedures? They help us set up. So the activity ahead is effect of catalase on hydrogen peroxide. Effect of catalase on hydrogen peroxide. Now there are three things that we are testing here, or rather we are aiming to achieve by this investigation. And the first one is we want to be able to use techniques, apparatus, and materials, all right? We want to be able to use techniques, apparatus, and materials. The other thing, we should be able to observe, measure, and record. We've been doing that ever since this lesson began, yeah? Then lastly, which is most important, other than just collecting data, is interpreting and evaluating observations and data. Interpreting and evaluating observations and data. All right? Now, I must give you a few warnings concerning the task ahead of us. Now, we need to wear eye protection because hydrogen peroxide is a powerful bleach. And if it falls on your skin, you need to wash it off with plenty water. Or plenty water. I'm repeating so that you don't say you didn't hear. So, you're being provided with a white tile. Are they working in pairs or? Pairs. 6 groups. Mm-hmm. Yes, to test for oxygen, yes. Now, do you remember we were testing for hydrogen using a burning splint? Now, in this one, we will test for oxygen using a glowing splint. So, how do you get a glowing splint? You light it, after it burns a bit, you blow it off, and then you leave it glowing. You introduce it where you suspect there is oxygen. If there is oxygen, it will relight. Okay? So, Miss, are they measuring the hydrogen peroxide for themselves? Okay, please, can you send a group member to take safety goggles? The one who has gloves should also take safety goggles. That's the one who will handle the hydrogen peroxide. Okay. Now, we are only using 10 centimeters cubed. Have you sent your representative for your... Miss, can I just measure for them? Because I'm afraid of spillage. Where is my laptop? Naogopa spillage. Naogopa spillage. Yeah, I'm biting. How many minutes? 4. I need to go to the washroom in 4 minutes. Where are those small measuring cylinders? Please come for hydrogen peroxide. Come for 10 ml hydrogen peroxide. That's for group 1. Quickly for group 2. That's dirty. Come with your measuring cylinder, quickly. Any other? Hi, was this clean? Because I've just put some. This one has already started bubbling. I don't know what was inside there. You go try. Do we all have? Harold, you are coming here and you're not saying anything. You are just standing here. I want you to say what you have come for. Is it time? It's time. I'm coming. I'm back with a bang. Let's continue. Do we all have hydrogen peroxide? Now, do we all have the live matter? Yes. So let me guide you on the next procedure. Okay, now, we already measured 10 centimeters cubed of hydrogen peroxide, all right? Now, to that boiling tube, please add a small piece of liver. Now, do not handle the liver without gloves, kindly. You've already done that? So you crushed the liver using a mortar and a pestle. Or you can slice it. You can slice it into small pieces. You can chop it. Chop the liver into small pieces. Not really, because the hydrogen peroxide is not much. So just maybe an amount, a small piece of liver. Yeah, so you're being given the mortars and the pestle to crush to crush the liver. To crush the liver. Now, why do you think we need to crush the liver? Why do you think we need to crush the liver? Yeah, to increase surface area so that we can have quicker and better results. Now, we need to have a source of fire. Now, remember, we are testing for oxygen using a glowing splint. So nitachukua tu bado. I'll still take at 3:17. What time is it now? What time have I come back?
You came back at 2:30.
2:30. So then I can only go at 3:30 for the 17 minutes. Yeah, they are mine. I can even take every 1 hour. I can even take every 1 hour. 5, it doesn't matter the number. Now I want to see glowing splints relighting. That's all I want to see. How you do it is up to you, but I want to see glowing splints relighting. Now, if you manage to make a glowing splint relight after that reaction, please let me know. I want to come and celebrate with you. Yes?
units of hydrogen peroxide.
You're using this is?
Hydrogen peroxide.
Yeah, so you're putting it, or you did the other way around?
What?
You did the other way around?
And to make hash to Rebecca the small piece one.
It's okay, you can put the hydrogen peroxide after you've put the liver. All of it, the 10 centimeters cubed. Just have do you have a splint? Light it, make sure it's glowing. Maybe the camera people can come and capture this moment when you know it doesn't glow for long, so maybe you can just put the hydrogen peroxide. Okay.
Anja, nani simnisaidie hii nini?
How come? I wish probably we would have maybe blocked the gas from getting out, and then when it accumulates, can we redo? I can give you more hydrogen peroxide since you already have the liver. Dispose that. Let me measure for you hydrogen peroxide.
Thank you.
Please do not miss the excitement of, or you can put, yeah, for their group, 10 centimeters cubed. It's still okay. Don't worry, because it's already contaminated. It's just leave it as it is. Any other group that wants to repeat so that I am committed to making us relight a splint. Kuleta gloves zingine. Sawa, toa hizo udispose. Yes? Eka yote. Now, nikupe siri. Ukishaweka funika kwanza. Let the gas accumulate. Then once it's a lot, introduce the lighted splint. No, glowing splint. Yes, you can. Where is your splint? Where is your source of fire? Oh,
It is going off.
make sure it's glowing. Darlene, kosa? It's too late, it's okay. No problem. Yeah, that's what I also thought. So maybe the catalase was not enough. Yeah. So this is officially my group.
Wow.
Until we relight the splints.
Hallelujah.
Imechukuliwa na Marlon.
Pistol has been about, so to me.
I use a little bit more blue.
And then how will we prevent the gas from escaping? I will
Cover it like this. Just light yours.
Light yours.
Lighter.
1, 2,
3, yes,
ngoja gasi accumulate.
Unasikia inakusukuma? Oh no. Not again.
Power imewaka because the nini inachomeka.
But it's always exciting when it's
burning.
Oh, it's burning.
Yeah, it's burning.
It's actually glowing. The splint is actually still glowing.
So ni kama inapuliziwa. Please call me to your group when you successfully relight a glowing splint. Oh, nice. Okay, so what do you think we should do? Because that's not a real positive result, eh? We do one more time?
Yeah.
Dispose. My work is to bring hydrogen peroxide. And gloves. Gloves are here. Have you managed? Oh no, you can't use such a short splint. Come, I give you a splint. Hey. Hey. She's using a splint that is the size of my finger. This kagal here. Wewe ni wa group yetu? Now, are you telling me that none of us has successfully relighted a glowing splint? Splint. Miss, are we using these skewers as the splints? Well, actual splints. He needs, I also need. He needs and I also need. Okay, where are the splints? No.
We've been using this.
Yeah, the skewers do not relight. Huh? Let me look for the splint. Eh? Ni rada nakuja. No, I bought it yesterday, the day before. It's concentrated. We have actually unsealed it today. Yes. Where are the splints, kindly? Splints. Ah, thank you. Crashing on the tile, it's not possible.
No, I'm not saying that. The outer membrane.
Yeah.
Huku sichinjangi, ndio zinitolee macho.
Sorry? Now, please, we need to go on to something different. I just want one group that will succeed. and we call it a day. Now, when you succeed, let us know. Is this hydrogen peroxide out of order? 20 volume. Yeah. I'm the one who bought this.
We have to try until
There's 100 volume. Because this is mild, if it's the one for first aid.
Yeah, 50%. Let us try with this.
Okay, we are changing our tactics. Okay. Can you come for more concentrated hydrogen peroxide?
This way, we have to return.
Just discard it. We can't return. We must get results by all means. Now, Mr. Mark will give you the hydrogen peroxide. Come for it. 10 ml, 10 ml. Sorry, I didn't hear. Please rinse your measuring cylinder before coming for more hydrogen peroxide. Don't finish the liver. Musimalize hiyo tissue. Mshamaliza. Now, they have brought more concentrated hydrogen peroxide.
We have depleted our liver.
Could we get more liver, miss? Excuse me, miss. Could we get more liver? Miss, yeah. Could we get more? Oh, it's okay. All right, thank you. Okay, let me see yours.
There is more liver here.
Zima.
Huku zima.
No, relax. You have gloves.
Ukitumia 2 centimeter cube of it, ndio reaction place kidogo to concentration.
Yeah, you could try using 5 ml. Not 2 ml is too little. 5 ml is okay. Excuse, could we try using 5 ml instead of 10 because the 10 is frothing too much and we are unable to get the this group. Where are we? Why don't you go for the more concentrated hydrogen peroxide? The 50% volume. It's there. It's going to relight. Finally! Yay! Yeah? 5 seconds. This is their secret. Can I say their secret? Reduce the volume of hydrogen peroxide from 10 centimeters cubed to 5 to give it time to accumulate the gas. Yes. When I saw that it is the one for first aid, I knew then it is mine. Oh, but we started yesterday past 4.
So it's for 50.
4:50.
Yeah. So we can
Now, for those who have managed to get the results, and even those who have not managed, we need to move to something else. I'm writing for you the chemical equation for the breakdown of hydrogen peroxide to water and oxygen. Are we together? Okay. Now, nice. Concentration, okay. You're just passionate about biology.
Oh my God. Me, I really don't. You see that's why I'm supposed to go for lunch and I'm not going. When it's not chemistry, I disagree. I love chemistry. I mean, I love biology. No, I love chemistry, but I love biology.
Okay, let's sit down and do something different. Let's sit down and do something different. Have you managed to relight?
Yeah.
Have you managed? I'm sorry. Hugs. Poleni. Okay, let's sit down. Let's sit down and do something different. Now, please get rid of any hydrogen peroxide that is remaining. Switch off the Bunsen burners. Switch off the Bunsen burners. You're lying. How will it even light and you have inserted it inside? Dennis. Now, please do not move to the next thing without copying that chemical equation. Okay? Promise? Okay. Breakdown of, oops. I'm sorry, there was a mistake. Hydrogen peroxide, H2O2. Okay, now I want us to conclude on enzymes. Okay?
Yes.
I want us to conclude on enzymes by looking at temperature, effect of temperature on enzymes graphically, effect of pH on enzymes graphically, and the other one is? Okay, enzyme concentration versus substrate concentration. That one we'll discuss and write. And then presence of inhibitors and activators. So let's go to that quickly. Now, have you all written that chemical equation? I insist. I insist. Now, I'm drawing for you a simple graphical representation of, attention kindly, I'm drawing for you a simple graphical representation of an enzyme that works best in a human body. So we expect the optimum temperature to be at what temperature? Between 36.8 and 37.2. So the y-axis indicates the rate of reaction. So I expect you to draw this under that part for temperature. If you have depleted your space, do it in the exercise book as long as you write the subtopic. And then we have the x-axis, which is temperature. Now remember, this is in a human body. Okay? So definitely anything between 0 and 20 something is inactive until it gets denatured there. Don't mind my proportions as long as you understand what I'm saying. Now temperature is in degrees Celsius, and you can see that the optimum, this optimum is around 37 degrees Celsius. Now, this is in a human body. Now, for plants, the optimum temperature for enzyme action is between 15 degrees and 20 degrees. So if I'm talking about, or I'm drawing a graph for plants, it will be something like that. Are we together? Yeah. So this one is for human body and this one is for plant. So you can see that it is somewhere between 15 and 20 degrees. So that's a graphical representation. Now you may be wondering, why is this graph like this here? Because the enzyme is still inactive. So you can say enzyme inactive. Enzyme inactive. And then why is it going down after the optimum? Enzyme denatured. Are we together? Yes. So enzyme denatured. So denaturing, I've explained that in terms of enzyme substrate complex formation. Now let's go to pH. pH. Let's discuss pH briefly. Now enzymes are specific. Enzymes are specific on the pH they work best in. On the pH they work best in. Enzymes are specific on the pH they work best in. Enzymes are specific on the pH they work best in. Now let's discuss this before we write. Now, is your mouth acidic, alkaline, or neutral? Hey, you sound sure. You sound very sure. Sorry? Now, in an ideal situation, the mouth is supposed to be neutral. Yes. Are we together? Yes. In an ideal situation, the mouth is supposed to be neutral. And that tells you that salivary amylase works at an optimum pH of, what's the pH for neutral? 7. Good. That's salivary amylase. Now what would happen if you took salivary amylase to the stomach? Just use any prior knowledge you know about the stomach, even if you did it in class 5. Huh? It will be denatured. Why? Because in the stomach, the pH is, the environment is acidic and the pH is very low. The lower the pH, the more the acidity. Are we together? So you find that different enzymes work at different pH. So enzymes that digest proteins, they work best in an acidic condition, and that is why they are only able to digest proteins in the stomach. So, diagrammatic or graphical representation of that. Now, I'm going to use two enzymes, and I'll use pepsin within the same graph, and I'll also use salivary amylase. I'll use pepsin and salivary amylase to illustrate effect of pH on enzyme action. Are we done drawing this? Who is not done? So, y-axis again, rate of reaction. And then the x-axis is the pH. Are you together? So we have 0, 1, 2, 3, 4, 5, 6, 7, 8, 9. Let's go up to 10. For purposes of illustration. Now, pepsin. Pepsin is an enzyme that digests proteins. Where do you expect it to thrive most? Optimum temperature of? Sorry, not temperature. Optimum pH of 3, yeah? So, that one should be like that. So that the optimum pH is 3. So this is pepsin. Pepsin. Now let's talk about salivary amylase, which I will draw using a different color of pen. Let me use red. Salivary amylase works best in neutral conditions. So starting from 0, it will be inactive and then peaks at neutral. Are we together? So this is salivary amylase. So it's only one group that has been able to relight a glowing splint? Oh no. After all the effort. Now the rest, the two that are remaining, let's discuss them. Let's discuss them. Now let's talk about enzyme concentration versus substrate concentration. Comment about that referring to enzyme substrate complex formation. Musiniangushe. Jalimo. Martin. Comment about how enzyme concentration versus substrate concentration affect enzyme action, referring to formation of enzyme substrate complexes. Yes, yes.
The more the enzyme, the more the
there will be more formation of enzyme substrate complexes, therefore raising the enzyme activity.
Ah, good. So that means that the higher the enzyme concentration, the more active sites available. Are we together? The more active sites available for substrates to do what? To bind with. So this definitely increases enzyme activity. Are we together? Now, the substrate could be highly concentrated also. So the higher the concentration of substrates, the higher the chances also of a substrate binding to what? To an active site, forming an enzyme substrate complex. Please, I hope you're writing C, because I can see some people looking at me without blinking. Now, who can tell us about inhibitors and activators? What are inhibitors? Inhibitors. What is to inhibit something? Huh? To prevent, yeah? So if there are inhibitors, what would they be inhibiting? There? Enzymatic action, or rather they would be stopping the formation of enzyme substrate complexes. Please, let's note that down. Do not be distracted. Let's note that down, that inhibitors prevent enzyme substrate complex formation. And then we have, can we reduce the movements, please? And then we have activators. Activators encourage the binding of the substrate to the active site of the enzyme, increasing enzyme action. So basically, enzyme action is increased by making sure there are as many enzyme substrate complexes formed as possible. So let's go to part 1.35. Sorry? Activators encourage the binding of the substrate with the active site. Pay attention, be in class. Okay, now next is plant nutrition. Plant nutrition. Now, the first thing we do in this topic is to define photosynthesis. Some people say photosynthesis. Huh? Photosynthesis. Are we done with these graphs? Okay. Now, how many are we currently? Huh? Can I have 5 volunteers to go and collect specimen for us? Nice leaves. Don't bring dusty leaves and dirty leaves. We want to stick those. Listen first. Aye. Hey, Damaris. Now, I want you to bring leaves, okay, that have all the, we can clearly label, okay? And I want you to bring leaves that we can stick in our books using tape. So you're bringing leaves for everyone. So, Marston, Bramwell, Damaris, Marlon, or is it Malon? And Alvin. Alvin, go and get leaves. I know you are catching up with notes, but... Huh? Are you ready? You're ready? Okay, it's okay, don't worry. Don't worry, we are doing it. Immediately they come back. Yes, yes. We can. We can. We can. Okay, we can be defining photosynthesis as we wait for them. Now, give me the definition of photosynthesis. Jessica,
photosynthesis is a process by which plants, green plants, make food.
Now, you have answered me, but like a young baby. You know the way children say, plants make their own food.
It's the process by which plants make their own food in the presence of light.
Using?
Green color in mass.
Okay, now photosynthesis is a metabolic reaction. We start there. Because it's a chemical reaction that takes place in what? A living thing. Yeah, so photosynthesis is a metabolic reaction that takes place in green leaves of plants where they make their own food using water and carbon dioxide in the presence of sunlight or using light energy. By the way, there is no specific definition for photosynthesis. As long as you know how the process goes and what the process uses and what it produces, you can just write the definition in your own words, capturing everything. Are we done defining photosynthesis? Yes. Now I'd like you to write the word equation for photosynthesis and the balanced chemical equation for the same. We are not playing here. Taking you back to the basics. Write the word equation for photosynthesis. Distribute the leaves to everyone. I need everyone to stick a leaf in their book using tape. And take 22 minutes or 5:30 and take 20. Okay, I'd like to see your word equation and your balanced chemical equation. Photosynthesis is a metabolic reaction Yes, are we ready for the amylase? Let them stick the leaves and then we revert to amylase.
They have even the regions already. Imagine
Okay, now the balanced word equation. Okay, sorry. The word equation for photosynthesis is water plus carbon dioxide. Now, what do you write on top of the arrow and below the arrow? Light and chlorophyll. Now, why can't you write them on the reactant side? Yeah, because they are not reactants, they are not reacting. They are conditions necessary for the reaction to happen, okay? And then what do we get? Glucose. And we don't get energy from photosynthesis, we get energy from respiration. Photosynthesis gives us the food, or rather the glucose, and then this glucose is broken down in respiration to give us energy. So energy is not a product of photosynthesis, but a product of what? Respiration. Tuko pamoja? Okay. So glucose plus oxygen. Now, balanced chemical equation, how many moles of water? Hey, people have forgotten Form 2 work. 6 moles of water plus 6 moles of carbon dioxide to give you a mole of glucose. Glucose is C6H12O6 plus 6 moles of water. Wow. The confidence. It's the confidence for me. Oxygen. Okay? Yeah, now before I go to anything else, you see where there is parts of a leaf? Stick your leaf there using tape. And then now we go to one practical, then we revert to plant nutrition. We had tape somewhere. He has tape, and there is another one. It's here. Now here's the tape. So how you will use the tape to stick your leaf? So stick your leaf and label it based on the leaf above. Stick your leaf and label it based on the leaf above. I put it in there. You know, I stopped following that because you are not getting. It's there, 1.58. Yes. Mercy, where are the 3 books we sent for? They are all there. Please, they are there. 3. So. Please, let's stick that leaf quickly and label its parts. Please, let's be taking less than 2 minutes to do most of these things. Eh? Surely. Harakisheni, harakisheni. So that we do an investigation involving amylase and starch. At what time? Tomorrow. Tomorrow daytime. Just for starch. It gave us a negative. We'll do that sometime in this experiment, in this topic. Now let's proceed. Let's proceed. We are doing now the experiment. We are investigating the effect of amylase enzyme on starch. Now we go to the exercise book, please. We go to the exercise book. Investigating the effect of amylase on effect of amylase on starch. So I can see you have starch, you have Benedict solution, you have amylase, okay? Now, we are going to do a control first. Are we together? Can we get hot water just for one control?
Yes.
Okay? Sorry? We all do control. So first of all, we are going to do a Benedict's test on the starch before we add amylase. Are we together? Yes. So that we can clearly see the effect. Now, approximate about half of. Do you have more starch? We do. So we can use all this and then get more. All right. My group.
Yes.
Half. Okay, no problem. So we use half. We are approximating. Are we together? We use half this volume and half the volume of Benedict solution and test to see if there are reducing sugars here. Oopsie. Sorry. And then now later on, we are going to add amylase, okay, to see whether now when we test again, whether the amylase has broken this down to reducing sugars. Are you understanding the investigation? Yes. Now I won't even demonstrate. I think I've explained. No, demonstrating means doing for you. No. Now let's do that. So half the starch in a boiling tube, half the Benedict solution, dip in hot water.
Yeah, that's when has
So you're timekeeping with who? And the other guy? The other tall guy?
Duncan. So when you talk about the shift,
oh, him is not your partner. Who was your partner? And where is she?
And they are doing nurse today.
Wanafanya?
Walikuwa nafaa kwanza kwenda lunch. So ameshakula. So the next
Deal now, so they are student managers.
Yeah, she's with Jane. So I was with Jane.
You see, vitu kama timekeeping, you know they are very specific. Afadhali tu angeendelea vizuri. Not akina Duncan. I'm saying timekeeping is not a department we can keep for a crew. You see, like Liz, she should not have stopped timekeeping. It's more important than students eating. Because all these things, women go such a manage kabisa. I'm not at peace. I'm not at peace. Naona something will go wrong. Now, what have you found? Okay, okay. So you'll receive a water bath. Now, once you get hot water, you're timing for 5 minutes. Okay? You're timing for 5 minutes to see. No, 3 minutes are enough if the water is hot enough. It's not hot enough. I wish we got very hot water so that we get results faster. Then after that, now, the rest of the half that is remaining, you mix it with amylase, half the amylase, and leave it to settle and react, and then now test for reducing sugars again, using the same procedure. All right? Yeah. Okay. Okay, now what have you found after the first investigation? Positive or negative? After the first investigation, testing for reducing sugars in starch. Testing for reducing sugars in starch. Now, how long has it been in hot water? Okay, have you seen any change? So is it a positive or a negative result? A negative result. Dispose, rinse the boiling tube. Now put starch, amylase. Leave it for around 1 minute and then add Benedict solution and test again. Maybe we can get hotter water. Sorry? We are going to test after the enzyme has already broken down the starch. Okay. Now, what is that you're putting in hot water? That's starch and amylase. You're putting in hot water. Amylase is an enzyme. Just wait to be given water of the right temperature. in the warm water for around 3 minutes. And then now you can do the Benedict's test. Now, why are we putting it in warm water? Sorry? Not to denature the enzyme, and also to give the enzyme the optimum temperature for enzyme action. So after, oh wow, you already have a positive Benedict's test. Amazing. So I can see some people have already started having positive Benedict's tests. Okay, if you have had your positive Benedict's test, now we can move on. Have you allowed it to stay in hot water enough? Have you allowed it to stay in hot water enough? Yes? Yeah, as long as you're getting yellow, green, orange, that's positive. Okay, let's go back to photosynthesis. Now we left it at writing the word and balanced symbol equation for photosynthesis. So now I want us to look at importance of chlorophyll, nitrate, and magnesium ions. Importance of chlorophyll, nitrate, and magnesium ions. It would have been nice if we could get a variegated leaf or variegated leaves. Now you remember the experiment we did at night? Yes. And it gave us a negative test? Yes. And we said we would try it during the day. Yes. Yeah, so we are going to investigate. We are going to investigate photosynthesis in a variegated leaf by testing for starch. All right? Yes. But for now, we are looking at the importance of chlorophyll. We start with chlorophyll. Now, why do you think chlorophyll is important in photosynthesis? What is the role of chlorophyll? Someone is raising their hand? Imelda? Imelda, what is the purpose of chlorophyll in photosynthesis? By? Okay, but you have not said the specific purpose of chlorophyll. Now, chlorophyll traps the sunlight energy. Chlorophyll traps the sunlight energy. And contain catalysts that speed up the process of photosynthesis. Contains catalysts that speed up the process of photosynthesis. Again, chlorophyll traps sunlight energy. and also contains catalysts that speed up the process of photosynthesis. Now, in which organelle do we find chlorophyll? We find it in the leaf. In chloroplasts. All right? Now, the other thing I want us to look at is the usefulness of nitrates. Is it nitrates? Yes. Now, first of all, where do plants get nitrates? Please let's participate so that we move on without feeling tired. Yes? Nitrates, no, we don't get nitrates from the atmosphere. Nitrates are ions, remember? Yes, Alvin? From the soil, and they are absorbed through two processes. Which one and which one? Now, when the concentration gradient, okay, when the difference in concentration of the nitrate ions in the soil, when the soil is more concentrated than the inside of the plant, the nitrates are absorbed through which process? When the concentration is higher in the soil than in the plant, yes? Active transport. What about when it's the other way around? Diffusion, isn't it? Yeah, so we obtain nitrates from the soil, and nitrates are absorbed by the root hair cells and are transported to the rest of the plant through the xylem vessel. So nitrates and water are absorbed by the root hair cells and transported to the rest of the plant through the xylem vessel. Now, what do you think is the function of nitrates? What's the function of nitrates? Yes? Nitrates aid in making of proteins in plants. All right? Nitrates aid in making of proteins in plants, and proteins are vital for growth and development. Do you remember when we were looking at biological molecules, and we said that proteins are important for what? Growth and development, and also repair of worn out tissues. Yes. Same purpose in plants. And this is affected by nitrates. So how do you think a plant that has nitrates deficiency would look like, considering now the function of nitrates? It would have yellowish leaves. It will have stunted growth. And the organs of the plant will not develop fully because there are no nitrates, there are no proteins to aid in growth and development. Okay? Have you been writing that? Now, let's talk about magnesium ions. Where do we get magnesium ions? From the soil again, okay? All water and all mineral ions are absorbed from the soil by root hair cells. Now, what's the function of magnesium? What is the function of magnesium in a plant? Anyone? Hey, you guys, niwatume kwa field mkimbie 1 lap. Alafu mrudi. Amtaki? Then participate. Okay, magnesium aids in manufacture of chlorophyll. All right? Magnesium ions aid in manufacture of chlorophyll, and they're actually the ones that make a plant greener by ensuring as much chlorophyll is produced as possible. So that means that plants that have a deficiency of magnesium have yellow leaves, have leaves that are stained. Have you ever looked at a plant and the leaf has yellow dots, many, until it almost looks like it's all yellow? That is a leaf that is deficient of magnesium ions. We are moving on very well, isn't it? Yes. Any question about the importance of these three? Now, we already wrote the word and balanced. Write it here in the workbook. Write the definition of photosynthesis in the workbook. Okay? Or you have not been writing in the workbook? Okay, if there is space in the workbook, just use the workbook, okay? Okay, so what is next? Factors affecting photosynthesis. Now, some of these investigations would take days. Like some of these investigations, like, you know the way you take a plant and you cover it in a black paper and give it a few days to distarch, and then you return it outside, and then you leave others inside. Like some of these investigations for the factors affecting photosynthesis would take days to do them, but we can discuss them theoretically and how it would be and the observations that we would observe if we were to do that. Is that okay? Okay. Now, factors affecting photosynthesis. The first one is light intensity. Now, what's the importance of light in this process of photosynthesis? What's the importance of light, Lenny? Sorry? Tell us the importance of light in photosynthesis.
For the factors or importance
Sorry?
of light.
The importance of light for photosynthesis.
Okay, for me, I guess light help in breaking of, conversion of carbon 4 oxide and water through chlorophyll to glucose and oxygen.
Repeat again. It helps in conversion of
Water and carbon 4 oxide in presence of chlorophyll to glucose and oxygen.
So how does it help? That's what I want to know.
Oh, to explain.
Yes. Anyone with an explanation? Yes, Clarice? It breaks down?
And oxygen gas is used in photosynthesis process.
So would you agree with me that it provides energy for bond breaking and bond making that involves formation of glucose? Yes, that's actually how you can explain that. Now, you can investigate the effect of changing light intensity, okay, by having plants different, okay, or rather the same plant exposed to different light intensities for the same amount of time, okay? So the constant variable becomes what? Time in this case. And then the variable that we are changing, the independent variable becomes the light intensity. Are we together? So there is one here that has been described that can be done by moving a lamp different distances away from the beaker containing the pond weed. So I'll go around showing you. So the more the photosynthesis that happens, the more oxygen is absorbed. Are we together? Because oxygen is a product of photosynthesis. So if you want to know that there is a high rate of photosynthesis, then you will collect more oxygen. So this is an example of a setup of an investigation. Look here. An investigation that is used to check the effect of light intensity on photosynthesis. Are we together? Yeah. Marston. So you expose the pondweed to different light intensities and measure the amount of starch and also measure the volume of oxygen collected. Okay? Now, other than what I'm showing you, are we able to design together, okay, to describe how we would measure the effect of light intensity on the rate of photosynthesis? Alvin? So let me have manila papers, like 5 of them, and the permanent markers. I can see permanent markers all over. I want you to design an investigation together showing how you would investigate the effect of different light intensities on the rate of photosynthesis of a green plant. Let me write the question. Investigating effect of changing light intensity. So this is the title of your investigation. Investigating effect of changing light intensity on rate of photosynthesis Okay, now, remember, have a question, have a hypothesis. Are we together? Design the investigation, and then name for me the variables and how you would collect data. How you would collect data. Are we together? Now, which group is supposed to go out for break? Group 1. So, group 1, how many are in group 1? Raise your hand. How many are you? 5, 6. 6 can go. Is there a group outside? Then we can't release a second one. We have to wait for them to come. We can't have 2 groups outside. So please start designing the investigation.
It's not a group that is outside, it's just...
Let's wait for them to come back. Who and who? Bilha and Cynthia. When did they leave?
2 hours ago.
Let's wait for them.
You see now, you don't have student managers. That's why we are discussing this in class.
It's the assistant teacher who is to wait.
Let's design that we should take a maximum of 5 minutes. Where have we reached? Yes? We are investigating the effect of changing light intensity. So what experiment can you do, or what investigation can you do or design, okay, actually designing, yeah, to investigate how different plants would behave when you change the light intensity for them?
Part of the leaf with foil.
Yeah, you can do that. But you see, that one is blocking light altogether. This one is talking about intensity. So you can talk about putting one in a dark room that has less light, putting another one in a room that windows have been opened, putting another one under a tree, putting another one in the open, like as long as you can clearly show that you are varying the light intensity.
Even dark polythene bag because
yeah, dark polythene bag, you've blocked the light altogether, but it is going to show that light is important for photosynthesis. Yes, Harold. And then you come back after how long?
Just 2 minutes.
See light lakini haziangazi polythene black.
Oh, ya black on kwanza.
Excuse, let me make a correction. The one for black polythene, please, that one, you're blocking the light altogether. That one is actually for investigating the need for light in photosynthesis. So you block light fully, but this one is talking about light intensity. So how can you vary light intensity? Maybe have a plant in a room where the curtains are closed, another plant in a room where curtains are open. another plant under a tree, another one outside. I mean, as long as in your investigation you can show that you are varying the light intensity. Guys, I think we are working really, really slowly. We are going to take more than 15 minutes doing this. Huh? What are the independent variables? I want to remove the lab coats. It's okay. Now we are not awarding how of a designer you are. Huh? We just want to see if you're able to bring out the science in it. Excuse me. Trish, you're in front of the camera. How is it coming along? They do it in a subtle way. Uko sawa?
Trying.
You're doing well.
Thank you for that.
You're doing well.
You're doing better.
Atukuli lunch, mom.
I'm waiting for the students who are outside to come back.
I'm not doing architecture.
Well, it seems quite interesting. I'm looking forward to seeing the final products. Now remember, there must be data that you are collecting in that investigation, yeah. Because you make observations, you collect data, and now that's when you make a conclusion. Okay? And sometimes your conclusion can actually agree with the hypothesis that you had. Are we understanding? Yeah, it's an investigation, so we must have observations, data, etc. What's your idea?
We have 5 situations where plants are growing in different places, and okay, we have the dark room, we have where there's too much sunlight, moderate conditions under a tree, and near an upper condition. So which plant is going to flourish fast, depending on where it has been kept.
So what's the timeline that you're giving them? The timeline you're giving? So you're able to detect tangible growth within 5 days?
Not 5 days, maybe a longer period, like let's say 2 weeks. Then.
Yeah. So what data are you measuring? Length before?
Yeah, length before, then time.
And
what about the leaf size?
You could measure different parameters, but for leaf size, you can't measure one leaf. You must measure many, must pick randomly 10 leaves and get the average leaf size. You can choose to use length or width, okay? So for your results to be reliable, you must take 10 leaves of the first plant, get the average leaf size. For the second plant, like that. But again, I see a lot of gaps in that investigation, but just finish and then we'll see. Okay, let me hear your ideas.
Saidia nini choroni hapo mmea ingine iko in the open.
Choranga.
Like one in a well-lit room, another in a poorly room, and one outside.
Sorry, one in a?
Well-lit room.
No? Mm-hmm. Yeah. As long as you're able to vary the light intensity, it's still okay.
Chora hapo. Na mbakishe space ya tatu. Wakati tumesema tunaiputi na dark room.
So utachora
in the open, iko kwa field.
We're doing a well-lit room, partially lit.
This one
so
is
these are well lit.
So how do you define a well-lit? How do you define a well-lit room?
like well lit ni kama like in the open air. Like jua ina penetrate.
Like a room like this where the loop
Partially lit.
Partially lit ni
like
kama
place ka store enye light inaingia like kidogo kidogo, like it's not to say
Or a room that has curtains.
Then the other one totally dark. So dark room.
To show that in the absence of light.
Dark room is
there is no photosynthesis.
Kulikuwa na
So which data are you
Sema
collecting?
na open. Which data are you collecting about
How? Through using which data? Which parameters are you measuring? Because your investigation must have a measurement. It can't just be like a story.
Time.
Time off?
Come again with the question.
Like for example, I'll just like use their example. They are going to measure the average leaf sizes of the plants they are going to use. So like they take, if this is the plant, they take 10 leaves, they measure the width, and then they get the average. Okay? After getting the average, now they record that as the starting leaf size. So when they expose this plant to high light intensity, after like 2 weeks now they come and measure again the size of the leaves. Okay, the average size again. They can even mark those leaves so that they see leaf A has widened by, you know, like you have to collect data. You understand? Then now they go to the second plant. They take 10 leaves. They measure the average leaf size. And they even label some leaves so that 3 or 4 weeks later, after putting that plant in a dark room, they will come and see whether that plant has grown or not, and they can conclude that it is because it did not have enough light for photosynthesis to take place. So when you're designing an investigation, your investigation must show what you're measuring and how now the light intensity variations has affected your measurements.
Can we use the blue mill and the sprouting of a paper blue mill? One in a light, one in a well-lit area, the other one in dark, and then we measure
the sprout. But you see here we are measuring photosynthesis, so we must be working with plants that already have leaves, that are already photosynthesizing, and plants that will be affected by different light intensities. But he's talking about sprouting. Sprouting of, yeah. I think about that I moved to another group. Okay, so are we starting to get the idea better? Oh, but I was here, I explained. What happened? Okay. Now, what parameters are you measuring to show the effect of the different light intensities? Plant exposed to light, and then?
Direct light. I receive light.
So what have you measured to show the different light intensities?
The amount of light it is being exposed, so photosynthesis takes place. And
Amount of?
light being exposed.
I'm asking, what are you measuring in this?
Growth rate.
How will you measure growth rate?
By seeing how the plant flowers.
By seeing. We are in science, we must collect data.
We're trying to print that picture on paper.
Okay. Okay. Now I think I need to guide you guys. Are we together? Now every investigation must have data being collected. Every investigation must have data being collected. If your investigation cannot clearly show which data we are collecting, then it's not a scientific investigation. Because we must observe, collect data, and represent that data in different ways, like in tables, graphs, bar charts, pie charts. That's what science is. Collecting data and representing it in different ways, and analyzing it using different tools. Time is only awarded after 60 minutes. It can't be awarded for half an hour. I have 17 plus 5, 22. Yes, I told you that I can even take the hour.
That you have to tell separately because hapa haitaingia.
Sorry?
Nakwambia hivi, let's make it simple. These papers, you do the first grade, utajaza hapa, then the second one, you take another one of this and jaza. That should be okay.
Is there something to keep time?
It's just because it's like you don't trust us.
It's not. No, I have not indicated that, but I'm also telling you that you have to record as it is. It's not about trusting, it's about being keen so that we don't get difficult. Yes, as many times as possible.
That's what I'm saying. This is
Is there any group that has managed to come up with an investigation that is measurable? I gave a few groups a hint, but I'm still seeing people stuck. I gave some people, Aaron. Mark my words, I said some people.
I have a question. Can we do like an experiment, like similar to the experiment where we covered a leaf with aluminium or something like that?
We can do that. So should I give you more time, or should we suspend that activity until we have enough time to do it? We suspend the activity, but it will still come back. So appoint somebody to keep your manila properly. Appoint someone to keep your manila properly. Now, carbon dioxide concentration. Can we talk about carbon dioxide concentration? Let's suspend the activity, please. Suspend the activity. But suspending does not mean you will not do it. So keep thinking about better ways to design your investigation. But anyway, we are going to do it together. We are going to do it together. Now, where does a plant get carbon dioxide from? Where does a plant get carbon dioxide from? From the atmosphere. And what percentage of carbon dioxide is there in the air? 0.04, all right? So that means that for a leaf to actually absorb carbon dioxide to a level that that carbon dioxide can benefit that leaf enough for photosynthesis, then it means that the leaf must have a very efficient way of absorbing it. Are we together? And that is why the leaves are exposed. They are handheld out in the air by the stem. When you look at a plant, what's the first thing you see? The leaves. Even this plant out here, what are you seeing the most? And that's why plants have numerous leaves, so that they can absorb as much carbon dioxide as possible. All right? Someone was raising their hand? So we are writing what I am saying under carbon dioxide concentration. Are you together? So point number 1, carbon dioxide is obtained from the air. Carbon dioxide is obtained from the air. The atmosphere only has 0.04% of carbon dioxide available for the plants. So the atmosphere only has 0.04% of carbon dioxide available for plants. So now, what adaptations do leaves have that help them absorb carbon dioxide efficiently?